Hello?
Speaker, can I use my own computer or should I use that one? I can both.
It would be, in principle, it would be possible to use your computer, but for instance, I can't tell.
It's better to use.
I need to use my own and I speak at 9.
You speak at 9, so in 3 minutes. Okay. Would you like to place it on here or?
The recordings were great yesterday. I was able to watch from the train. So the connection for that is here.
HD.
Yeah. We can get the sterics to work. Yeah, I think it does. Does it not?
So I will have to check upstairs. No, that is not the. Sorry.
Oh, I think you got it. There we go.
No, leave.
Sorry. Good morning. I have run here. I'm a little bit warm.
Boy. The problem is always last day, first talk.
Well, thank you so much for accommodating it, and it really is the only time I could have done. Now I see the trains, I couldn't have.
Did you share your screen already? Because
Let's
there
go.
should be similar.
Nothing happening.
What video mode do you send?
I do not know, I'm afraid.
4K or something like this.
I'm afraid I don't know. Do you want me to go into settings? System preferences.
We don't get a signal.
Okay. Right, I'm going to take the Wi-Fi off. Let's try taking it in and out.
Maybe you also need to.
Oh, this is innocent. Yeah. Anything yet? No, it didn't even glimmer, did it? Yeah. Do you have a USB and I'll take it across?
A USB.
And then I'll copy it onto a USB.
I can organize it.
And then move it on yours. Vielen Dank. Not seeing it.
You can't see the device.
Not showing. Got him. It's got him.
Yeah, okay. So it's on the stick?
Almost.
Yeah, okay, I see.
Don't worry, it's Latin America where 5 minutes is nothing. Is it on now?
I think it is, yeah. And I'm just going to stabilize by
Put it on the screen already, then we are safe.
Yeah. Okay. And if I may use my slide advancer on your machine.
Yeah, we can try it. But do I have to, you could place it down there, or I will see. Yeah.
So that should be there and safe.
Yeah, this is not, we are also the only guests here. So.
Beautiful. What a lovely place to be a guest at.
Which one is it? From today.
Goss.
Yeah, just a moment. Copy.
Okay. Okay, we are set up. Sorry for the delay. Well, you know, an easy way to distinguish a serious and dedicated scientist from the, let's say, average one is to go to the first and last lecture of a conference. The first and the last lectures of the last day are usually only visited by those really dedicated. And so I'm very thankful for all of you who are here now, after yesterday's evening, to welcome with me Rebecca Goss. Professor Goss, I'm really happy to have you here, and she came on short notice because it didn't work out originally as planned. So originally I invited her, then she couldn't come, then I invited someone else that didn't work out because it's holiday season, as you know, and then she jumped in again, and I'm really, really thankful for that. It is a big effort for her, so thank you once again to do that. But why was it so important to have her here? Because she brings in some expertise which we haven't seen so far. And the reason is that it's not only about being prepared and finding drugs. At some point you need to produce them also. And also for that we need research, we need tools in place, and that's where she is, among many other things, active. I give you a short introduction to her. So she studied at Durham with Professor O'Hagan, got a PhD there, and then she had several stages. I think the unavoidable University of Cambridge or Oxford, one of them, or Imperial College usually, to have a perfect career, which she then had, of course. I won't go through all the stages here, but she actually ended then with a full professorship for biomolecular and organic chemistry at the University of St Andrews in Scotland. And who works in natural products chemistry knows that the center, I would say the center, not one of the centers, but the center of natural product chemistry in Great Britain is in Scotland. And many colleagues working there in various universities. And of course she brings and brought new methods, new methodologies, new ideas, also of course new molecules with her, adding to what was before also only classical natural product chemistry, isolation, characterization. Of course now with all these genetic tools and so on, we go far beyond that. We will hear that from her. I don't have to mention that she got numerous prizes. I could read here another 5 minutes or something. I won't do that. I would give her the stage. Please, Rebecca. We're happy to.
So thank you so very much indeed for your very kind invitation here, and for then re-inviting me when the colleague that I suggested wasn't able to come because of summer vacations. I am so sorry, everybody. I would have loved to have been here for the very first lecture and the very last lecture, but unfortunately my diary evolved in the interim, and I was bookcased in yesterday, and I fly to Boston tomorrow from Edinburgh, so just here transiently. But it's such a pleasure to meet a number of you last night, and I look forward to meeting more of you this morning. So, the title of my talk, Tailoring Medicinally Relevant Natural Products, using that hyphenation, a blend, if you like, of synthetic biology and synthetic chemistry. And I'll explain to you in a while why I believe these are perfect partners for advancing drug discovery and enabling sustainable production of new pharmaceuticals. So I've broken my talk down into small chunks. I'm going to start with a research snapshot. I'm then going to talk about why I strongly and passionately believe that natural products and natural product analogues are very much the way to go in 2024, 2025, and for the decades to come. I'm then going to talk to you very transiently about chemical tools that we use to manipulate natural products and the bio tools. Then I'm going to talk to you about this new concept that we brought together called genochemetics, introducing a gene to enable a reactive chemically orthogonal handle onto your natural product to enable site-specific diversification. So firstly, research snapshots. This is a distillation of a few of the things that we're having fun with in our labs in Scotland. So a number of you at dinner last night asked me, where on earth is St Andrews? So I added this extra slide in. A number of you will know Norwich. So Norwich is here, very famous for the John Innes Centre. I believe one or two of you have had parts of your career there. I moved from this area up to Scotland, so Edinburgh is here. St Andrews is across the Forth Road Bridge and just on this little peninsula out here. It's an 800-year-old plus university, so similar vintage. Its origins, of course, were around St Andrew's Cathedral. And we have some beautiful old buildings, some of them in better shape than this. This wasn't ransacked or didn't have any problems with fire. It was just repurposed. The bricks from this were repurposed into new buildings. So you see St Andrew's Cathedral here, the beautiful castle overlooking the sea. And then the other thing that St Andrews is a little bit famous for, it's allegedly the home of that very vicious game where innocent little white bulls are whacked mercilessly with clubs. So St Andrews, known as the home of golf as well as science. Okay, so beyond the old buildings, here's the Biomolecular Sciences Research Complex. I love this building here. It sits in the heart of our campus. We've got computational sciences here, medicine out here, chemistry at the back. So it's nestled in the middle, and it really brings together this nexus of disciplines to enable fast drug discovery and medicinal chemistry. So our labs are in the top floor here and also the bottom floor here. We had a little problem in 2019. Some inorganic chemists on the floor below us unfortunately had a very major fire, so we lost our building until last year. But it's a nice opportunity when something burns down to start with some very nice new designs. So this is the top floor now with rather nice views out across the ocean and our 2-meter fume hoods at the top. So nice place to be. Apart from fires, plagues, of course in the season there's also been wars, and I am very delighted that we are able to host the brilliant Elena Holodeieva. She was previously head of pharmacognosy and pharmacology at the International University of Kiev, and together she and I have been driving forward an AI program for selection of enzymes. So I think this was the reason that the committee very kindly invited me here. At the beginning of the pandemic, like all of us, I was very, very worried. I was very, very worried that vaccine development was obviously a race, but who was going to benefit in time from the fruits of our labours? And I was very, very worried at how unprepared as a globe we seemed for a crisis like that, in spite of scientists across the world predicting the next pandemic to be imminent. And so what I did was collect a group of friends, and within my role as one of the editors of Chemical Society Reviews, I felt that we should put together an opinion piece. And I brought together, this was with a particular focus on not Latin America, but on Africa. And some of you will recognize this stately gentleman here. This is David Newman. David Newman, who is still working, although in retired state, at the National Cancer Institute in the US. This is Professor Gordon Cragg. So his origins are from South Africa. Rosie Dorrington, a brilliant natural product chemist and virologist from Rhodes University, South Africa. Kelly Chibali, a brilliant malarial chemist from University of Cape Town. And Marcel Jaspers, one of the very best natural product chemists developing new tools for natural product identification. And the piece that we put together was really looking at that antiviral drug discovery and preparing for the next pandemic. So we put together the basis of what we would be looking for, and we very much shone the spotlight back on the future need for small molecules and banks of small molecules. Rosie, like me, is very excited about cyanobacteria and the wealth of natural products that they produce. And cyanobacteria are subject to many viral infections, so bacteriophage infections. And so we believe that they are a great place to look for new antivirals. So there's this article, which please go and have a look at it. It will still hold some useful content for you. Meanwhile, on the other side, my lovely friends, who I've just included a picture of here. So this is Professor Norberto Perrone Lopez from University of Sao Paulo. I added this in just recently for you. And his wife, Thais, who directs the brilliant spin-out Linoflora, were facing the pandemic for Brazil. And Norberto was going into the labs, acquiring hospital samples, looking at these. And Thais was looking at other variants for vaccination. Could she produce attenuated virus? So two brilliant people who I want to shine a light on for you. Norberto is a brilliant entrepreneur. He did one of the very first spin-out companies at the University of Sao Paulo. Now, under his mentorship, I'm so excited to hear that there are 200 going forward. So a brilliant individual. But the pandemic wasn't without casualties, and Thais lost 1/3 of her family because of COVID-19. Okay, so natural products and why natural products. I promised you a quick research snapshot, a distillation of things that we're having fun with. So in my research team, we have 4 complementary and overlapping themes, which you can envision as leading one through to the other. We start top left-hand corner with looking at new discovery. We like the unusual ecological niches. We believe if you look at the environmentally unusual, there you will find new chemistries, new enzymes, new genes. Hopping on from that, we look at our heartland of biosynthetic elucidation. We like exotic molecules, and we believe that if you look at the biosynthetically exotic, there you can find new hidden treasures, new types of enzymes, new types of reactions. This leads us on to chiseling out individual enzymes for synthesis, but also being able to harness entire biosynthetic pathways. So just taking you through that one by one, novel natural product discovery. A little while back, I put this in as a potential useful tool, writing this with David Newman and an excellent PhD student from my group. We published a natural product report on some of the newer ways for unlocking novel natural products, waking up silent and cryptic clusters, as well as overviewing some of the historic approaches. It's a golden age for discovery. I am super excited about natural product discovery with the advances in sequencing, with the advances in AI, and with the advances in being able to engineer organisms. This is one of the empowering technologies. I don't know if you guys are familiar with these minions, the nanopore. It's about the size of a Snickers bar, if you haven't discovered it yet. Or Mars bar. It's very, very fast, and it gains very long read genetic information for you. So if you can get a pretty pure sample of unfragmented DNA, then this is a great device. And you literally plug it into your computer, and all you need then is a okay web connection, so you can connect data in the field, so long as there is modest Wi-Fi connection. So I've hotspotted this to my phone previously. So this is really empowering technology. and enabling our natural product, multi-omic natural product discovery efforts. Biosynthetic elucidation, I said we like the exotic. Here's one example for you. I love this compound, pasidomycin. At first sight, it's not looking very exotic to you. When you look a little bit closer, beyond the peptide nucleoside fragment, you start seeing some strangeness. There's the N-terminus, there's the C-terminus, but look what happens in the middle. There's an inversion of the sense of the peptide by the introduction of this diamino acid. We believe that this helps against peptidases, so you're getting around that peptidic instability, and then a reinversion here. You look a little bit closer and then you see that nothing is normal within this. So in terms of amino acid usage, I've said there's this diamino butyric acid. It occurs in several other of the uridile peptide antibiotics, but apart from that is very rare. Not tyrosine, but metatyrosine. And perhaps the most unusual part of all is this nucleoside. The 3' hydroxyl is missing. And so we've been finding and working on the 5 enzymes that assemble this component here, seeing whether this can be used to assemble fragments for antiviral discovery, as that chain termination unit is already in place. So those are some of the things that inspire us for our developing enzymes for synthesis, but what I'm really going to talk to you most about today is harnessing entire pathways and accessing libraries of bioactives by using that blend of synthetic biology and synthetic chemistry. I've just added in a couple of additional reviews which might be useful to this audience. People are very welcome to screenshot if they want. So with Marnix Medina, we were part of the collective that worked on a review on artificial intelligence for natural product drug discovery that came out last year. And something that I think might be useful to a number of you, we've been building libraries as well for chemical analysis, and we built up a compendium of all of the cyanobacteria and all of the published genomic data sets. Unfortunately, there's a big gap between sequenced organisms and the ones where the really cool natural products are being found. So we had to name the paper Mind the Gap. So my team and I are passionate about natural products in the broader sense. Here are a few of our muses. You've already seen pasidomycin, which is strange from a biosynthetic point of view and inspirational from a biosynthetic point of view. I also love this compound because it has an exquisitely narrow spectrum of activity. It hits only Pseudomonas aeruginosa. So that's very interesting, as aeruginosa has the greatest number of efflux pumps. There are analogues of this, though, the sansamycins, where you have a methionine at this position, and these have promising anti-tuberculosis activity, and there are groups in New Zealand and Germany working on that. Our interest in natural products is both structural guided and activity guided. So we're also very interested in the plant-derived adenosines, which we work on with Anne Osborne. We're very interested in marinomycin, a novel polyene antibiotic with very good activities against vancomycin-resistant enterococcus and also MRSA. Notably, it's also got very good activity against melanoma cell lines. And then good old compounds like rapamycin, which I'll tell you a little bit more about in a moment. So David Newman refers to rapamycin as the drug for all diseases. Good antifungal activity, but of course, a huge immunosuppressant activity and utilised clinically for kidney transplant patients and others. So why this fixation with natural products, and why should you guys be fixated or become fixated with natural products? So natural products have a very prestigious history. They've been used in medicines, traditional medicines, in Egypt, Israel, China, Japan, since the beginning of documented history. But if we look at the statistics now, in this new century, then we can see that these are very compelling. So over 80% of antibiotics entering clinical trials are still informed by or based on natural product scaffolds. And it doesn't end there. So for chemotherapeutic compounds, again, over 60% of chemotherapeutic compounds going into the clinic are based on, or their targets informed by, natural products. So a really exciting place for drug discovery. These molecules, made by biomolecules, are naturally predisposed to interact very selectively with those biomolecules. But what about the analogues? Why add and analogues? Well, if we really want to pin down the details of how these beautiful compounds work, we need to make analogues to explore structure-activity relationships or to pull down targets. We also have to think about their medical utilization. And we saw a beautiful example in a talk yesterday of a natural peptidic antibiotic and the improvement that could be made by placing those lysines in there. Of course, they have been evolved in their own systems, whether that be in microbes, man, or plants, to address their own niches. And there's great opportunity for improving bioactivity and bioavailability by dialing in to analogues. There has been a bit of a turn from this community, though, and one of the reasons behind that, and I'm quoting Scott Miller from Yale, is that natural products are misperceived to be unmedchemable. So what's meant by that? I think this is stuck back in ancient thinking, dinosaur thinking, as to our methods for making natural products. So natural products, beautiful architectures that many of us fall in love with, but they're also very, very complicated. And it's very difficult for chemists to think about how they could exchange in, for example, a methyl at this point, or how they could even selectively just displace one of the hydroxyls. Unless you can put in a chemically reactive, chemically orthogonal handle into these systems, you are really straitjacketed by the innate chemistry within here. So semi-synthesis can be limited. One of the other ancient methods which has led to this misperception is that of total synthesis. So total synthesis, where often it's compared to seeing your beautiful target, looking a little bit like a fantastic mountain range, and the aim is to reach that target in the fastest time possible and the fewest steps. Like mountain climbing, total synthesis is grueling. So for rapamycin, that immunosuppressant, Casey Nicolau's synthesis was 102 individual steps. Imagine doing that, making one compound, purifying it, then subjecting it to the next reaction, 102 times over, and that's when the route to the top has been made. But the microbes that make it can make it in 3 days. So our approach, yes, we do use some total synthesis when we need to, but our approach is, why not piggyback a ride on those microbes to reach the top of the mountain and deliver the analogues that we want? There are several ways of putting that cable car up there. There's an approach, which many of you will be very well aware of, where you feed in an alternative building block into the organism, and that building block, if it can be incorporated by the biosynthetic machinery, is added in to that natural product. So a pre-biosynthetic chemical step, extending out the biosynthetic cluster's capability, of course called precursor-directed biosynthesis. What about post-biosynthetic chemical steps? Well, of course, this is semi-synthesis, but as I've said, it's very much limited by the handles that are innately present, the chemical orthogonality that's innately present in those molecules. So we've been developing a new approach, which we call genochemetics. So with genochemetics, we take a gene from another organism. Now it could be something as unusual as a dolphin or a cabbage to complement a plant or a microbial genome, codon optimized, of course. And then that adds in a chemically reactive, chemically orthogonal handle into the natural product. And then you can do your post-biosynthetic chemical step exclusively on that handle. So approach of genochemetics. Okay, so a quick example with precursor-directed biosynthesis, mutasynthesis. Here's the building block, or an alternative fluorinated building block for the start of rapamycin biosynthesis. We're leveraging the biosynthetic machinery from Streptomyces hygroscopicus, the rapamycin producer, and feeding that into the system. Can we make these pre-rapamycins this way? So, synthesizing the building brick isn't trivial. This is 4 years of PhD. I'm skipping past that. That was a valiant effort, and some of these compounds are not so straightforward to make. But once they are made, the microorganism makes it look very, very easy, and you can very quickly dial in to 26 mg a litre of the fluororapamycin that you want. And in one step, adding this in, you can access through to that range of diversity. You, of course, get varying different levels of production, and this is, of course, due to how well that building block is accepted. But then there's always room for further engineering. So that one-step approach to making your new rapamycins. But frustrating, because each brick is so hard won. So going back to that perception of unmed chemical, what if you could CH activate anywhere? What if you could put a reactive handle on any molecule in any position that you wanted? Surely this is the holy grail that many medicinal chemists are after, because if you could do that, you could work out where to put a tag for a bioactivity assay. You could very quickly sort out structure-activity relationships. You could make quickly your modified, improved natural products. So what if we could do it on something simple, like this hydroxytryptophan? Or what if we could achieve CH activation on something mid-sized like this, or even dream of our bigger targets? So I'll show you how we're doing this at the moment and how we're converting these dreams slowly into reality. So our approach, again, leveraging enzymes. So taking our substrate and incubating it with a selected enzyme, and we're using increasing levels of AI to be able to select the right enzyme. We're looking at various different handles to install. I'll focus here on carbon-halogen bond formation, because once you can introduce in one position precisely a carbon-halogen bond, it opens up a gateway limited only by your imagination to any diversification that you could want. You can then switch this out for pretty much anything. Now we need to think about the chemistries a little bit. So if we want to do things in the same pot as the enzyme, which is often beneficial for sustainability, and ultimately it would be lovely to tag these molecules in living systems in presence of targets, then aqueous conditions need to be developed. Here are a few of the aqueous conditions. Again, you're very, very welcome to just take a screenshot of this. These are all developed so that they're pretty easy to use, and what we do on the chemistry side of the team is we make these catalytic cross-coupling mixes and we pass them to biologists in our team. So how to switch out that CX. But I want to just let us reflect for a moment on why CX, carbon-halogen bonds, are so important. If you look just solely at carbon-chlorine bonds, and you look at the top 200 pharmaceuticals, you can see that 93% of them require chlorine. So 93%. Why did they do that? So one of the reasons is that handle, Legalization of the molecule, so that you've just got one plug that you can then bolt molecules together with. But carbon-halogen bonds are also a really good way of tuning bioactivity, bioavailability, and also metabolic processes. So you can increase bioactivity, you can increase bioavailability. So let's have a look at the star of the show. I think it's amazing how these enzymes can so selectively put a halogen just to one position on a molecule. A benefit of using an enzyme like this is that it can add it in late stage. It can put in a carbon halogen right at the end of the build of a molecule or on a natural product scaffold. Having a carbon halogen bond there is, in my mind, a little bit nicer than having an alkyne or an azide there. If you have an alkyne or an azide, at the end of the day, you have a heterocyclic scar. Whereas if you have a carbon-halogen bond, you can get precision point-to-point formation of a bond. With an alkyne or an azide, you have to build this in right at the beginning of a molecule. Whereas with a carbon-halogen bond, then you can put it in right at the end. You can tailor right at the end. So it's a late stage opportunity. And I'm going to reflect very briefly on how this precision is enabled. So these are the flavin-dependent halogenases that were previously known. Karl-Heinz van Pey with pRNA from pyrrolnitrin biosynthesis. This was the first one to be seen. And that selectively places at the 7 position. There are others that place selectively at the 6 position and at the 5 position. But this was pretty much all that was known. This little portfolio of halogenases was all that was known when we started our research for these halogenases. Their selectivity is mediated by the placement of the halide onto a lysine, which hovers over, points a finger to just the position to be activated. So this is Karlheim Van Pee's PRNA at the 7 position. And here you see it from the side, pointing its finger over that 7 position there, activating just there. But as I said, a small portfolio then, and we believe that there should be so much more. So we started to have a look at all of those halogenases that I'd shown you that work around tryptophan, and we'd started to pile them up and see whether or not we could see any additional gene signatures. And then we took this new gene signature and fired that against all uncurated space and started to find some very unusual enzymes. Here's one that I'll reflect on for a moment here. So VERX1, rather than just working around those dinky scaffolds, now we'll take these much larger medchem relevant scaffolds, placing a halogen just at 1 position. Where does it come from? VERX1, it's from a virus, a cyanobacteriophage, and one that infects the 2 most abundant photosynthesizers on the planet, Synechococcus and Prochlorococcus. So we're very interested to see what other genes phage are carrying in. They tend to bring in a lot of additional photosynthetic genes, extra copies of photosynthetic genes into these cyanobacteria to boost photosynthesis, but they're also bringing in a lot of halogens. We're working with Asaf Vadi from the Weizmann, trying to understand the natural roles, and we're also utilizing these enzymes as tools for precision molecule editing, accelerating drug discovery, and spinning out a company focused on those halogenases. We've now got a library of over 200 halogenases. This is a slightly older number, about 50 additional on top of that now, where we can start to predict and walk around many different scaffolds of CH activation. So going from what was previously known to medchem scaffolds of interest and placing halogens on them, and then CH activating. So of our original dream and target sheet, these are the ones that we can reach very, very quickly. We've recently been able to reach DNA presented substrates, showing that we can do this on members of DEL libraries, and we're working to develop that further. And we're now reaching out for bigger halogenases that can enable late-stage functionalization of aryl halides, yes, but also alkyl halides too, for diversification. So the very final thing, bringing this all together, that genochemetics. What if you could have your natural product of interest or your drug molecule of interest, and then you could tune it in a living system, maybe even in the presence of the target? And what if you could use it and conformationally shift it? So with the genochemetics, we're just at the start with this. I've shown you that we take an organism, we take a gene to add in a handle to make that tagged natural product, whether it's a halide, an epoxide, an alkyne, an azide, but our preference being the halogens, and then moving out and tagging that, so using it for product library generation for SAR, fluorescence tagging, and for affinity tagging and directional immobilization. So playing it with this antibiotic, psidomycin, we know that the key end required for activity against Pseudomonas aeruginosa, where it inhibits MRAY, is this end. So we're tagging into here and doing this in a living system, bringing in that entire series of genes into the organism. And then doing our cross-coupling upon that, we're able to, within the living system and inside the cell, make those brominated metabolites and then trap them within the cell. So just really trying to bring together a snapshot for you of why we believe blending the best from both worlds as dictated by the targeting system provides such a powerful approach for the future. I'm really grateful to my lovely and brilliant team. These are my saints in St. Andrews, or halos for halogenasing. And it truly is a blend of synthetic chemistry and synthetic biology. So members of the team quite often come in as synthetic chemists and then move on over to the molecular biology. And then molecular biologists also want to learn some of the chemistry too and start evolving in that direction. Very grateful to these guys for their generous funding. Very recently received another very large grant from EPSRC, not mentioned here, for primed precision innovative molecular editing for diversification, so new openings on that. Very grateful to these guys, and thank you so much for your kind time and attention at the end of the beginning of day one after a busy night last night. I would not be doing my job properly if I didn't flag to you this journal where I'm one of a team of 6 commissioning editors. It's got a not too bad impact factor. It's at 4.6 at the moment. We love things that are at the interface of biology, target identification, and chemistry. Please be in touch with me if you're interested in writing an article there. And this is a conference past. This sadly happened last year, Synthetic Biology of Natural Products Conference in Mexico. It will be running again in 2 years' time. It's tentatively scheduled for Mexico, but we'll be able to update you on that later. And we have scholarships, particularly for people across Latin America and early career researchers. So thank you so much for your very kind attention.
Yeah, thank you, Rebecca. It's a great overview of the work, some inspiring ideas, and I think there will be 1 or 2 questions, and surprise, surprise, Martin has a question.
Thank you very much for this very interesting talk. I have a question concerning the halogenation topic, because you mentioned these are flavin-dependent halogenases, right? And I think bromination is usually easier with those enzymes than chlorination because of the redox potential. Can you maybe comment on that?
Absolutely, great question. Okay, so in terms of redox potential, the easiest is iodination, then bromination, then chlorination. And until 2019, there weren't any of the iodinases known. That viral one, its preference, kinetically, you see it's for iodination. Why wasn't it seen before? So there were some studies that were done by the Walsh lab where iodide sources were seen to inhibit. The problem there was that the access to the active site was very small, very constrained, and of course the iodide is much, much bigger. So hence the blockage. We believe with that, with Virex-1, that first viral halogenase, because it has such a large active site, therefore there's not a problem for iodination to be mediated. The other nice thing with Virex-1 is it's got such a large active site, so very large substrates can come in, and there is a pi stack vice that substrates can be very directionally orientated, so you get that one regiochemistry. But yeah, the short answer to your question, sterics precluded, bromination had been seen to be dominant when the active site will fit a bromide.
Fascinating enzyme. Thank you very much.
Other question? Or yeah, Svetlana.
Thank you very much for a nice talk. I was just curious about the influx of these heavily modified precursors. Do you see any troubles with the influx, and also do you also work on the engineering of possible transporters?
Really great question as well. Okay, so for those substrates we have, all of the substrates I've shown you, there's been no problem with efflux. When we're including halotryptophans as precursors, again, no problem with efflux in any of the bacterial systems that we've worked with. And quite often you get a much higher level of production once you're putting that halotryptophan in, because basal levels of tryptophan availability are quite low. So you load something in that's lipophilic, it's well uptaken, and your natural product production is nicely accelerated. There are some substrates that can be problematic for uptake. Sometimes you can get away with this by just simply making an ester of an acid to increase the lipophilicity, to increase its uptake into the cell. There are some other very smart tricks that are done. People used to put a lot of time into making snack esters. Doug Hansen, when he was with David Sherman, showed very, very nicely that larger simple esters can be used to replace that. So if you're looking for a general trick, go and have a look at Doug Hansen's paper. Great for improving uptake of some of those precursors.
Thank you.
I also have a question myself. I mean, of course, bromination is a little bit nicer for later or later stage modification. And I was really, congratulations for the iodination. By the way, the first enzyme I've ever seen coming from a virus used in chemistry. But this is all on aromatic systems. So the interesting thing is we have, for instance, for fluorination, we can do that well on non-aromatic systems, but not on aromatic systems, where it also has a benefit, as we know as chemists. On the other hand, for non-aromatic systems, chlorination, bromination, okay, iodination probably not important, is not well known, although in nature we have also lots of at least chlorinated compounds, non-aromatic.
So this is one of the things that we've been looking at for a little while for the alkyl halide activation. One of the strong criticisms of current medicinal chemistry is it sits very much in flatlands and it doesn't explore the three-dimensional architectures that natural products do, and that's needed for a good medicine. There are the alpha-ketoglutarate dependent halogenases. They look very, very similar in structure to the hydroxylases, but there is a block at the active site. So there's been some very nice work by Michelle Chang, who's uncovered a few more of these alpha-ketoglutarate dependent halogenases. They tend to be a little bit more fussy to work with. They tend to, some of them you require glove box processing with them. But yeah, we are nursing a few through at the moment.
With the halogenases, which we encountered also, in vitro, in vivo, you don't see it, but it's also evident is that they are sort of self-destructive.
Yes.
So the question is, how can we improve the performance? You seem to have an answer if I see your smile.
Well, that is the nub of the problem, isn't it? So At both steps, with the flavin reductases and then with the halogenases, so that the pairs work together, there is often leakage, there's production of hydrogen peroxide. There can be leakage of that hydrogen peroxide, production of HOX, which is passed down a tunnel to that lysine for delivery. There can be leakage of that HOX. If there is leakage and if there is overproduction of those two components, then that's very much your enzyme is creating its own bleach to denature itself. But we're working on a few tricks to get around that, and we have some very high conversions and much longer longevities, but it's early days on that.
Final question, or if not, I don't see it. Of course, there is a break later. Thank you very much once again for your inspiring talk. And as the whole setup is not only international but also extremely interdisciplinary, we absolutely change to a different issues. We move, but we stay with microbiology in a way. The next speaker will be Peter Valentin Weigand. He is he's the director of the Institute of Microbiology at the Veterinary University of Hanover. And I'm very happy to have you here. Breaks old memories, later to that. He studied in Gießen veterinary medicine, and after his habilitation in microbiology, he was DFG fellow, among other things, in New Zealand. So already the second person who has been in New Zealand on the other side of the earth here today. And then became a research associate at the TU in Hanover, but he moved up the ranks, and at the moment he is the director of the TU, and also he is a full professor for microbiology and animal infections. And I'm really, how you say, ich bin, now I miss the words. I'm really looking forward to this talk, because it will tell me something which is absolutely out of my area, and that is, for me at least, is often more exciting than to have another standard chemistry talk. I'm not talking about yours, because that's not standard chemistry, yeah, but this is something from the other world. We have several such talks here, and I think that's a nice thing for the audience also, to hear totally different perspectives. Looking forward to that.
Yeah, good morning everybody. Thank you very much for the kind introduction, and thank you much to the organizers for giving me the chance to talk here. And as mentioned, we are coming now to a completely different topic, so to say it with Monty Python, and now to something completely different. But it is not as completely different as you may think when you see the title, the Porcine Respiratory Disease Complex, PRDC, because first of all, respiratory infections in animal medicine, in livestock especially, they certainly have the consequence of massive usage of antimicrobials. And you all know that the rise of antibiotic resistance is of serious concern, and that the veterinary medicine contributes to this. And if you have heavy use, then certainly the chances are quite likely that you run into this problem, and that you need bioactive compounds, new antimicrobials and natural products. So the topic which was introduced by you, or the talk before, is also important for us. And thank you again for the brilliant talk. And I appreciate your love for chemical structures. I also like them, but I like even more the microbes and morphology. So my first question is, are any vets in the audience? Okay. Me. I'm the only one. Good. So the topic I will talk about is, as you can see, these complex situation in porcine respiratory diseases, which are important globally, worldwide, and as mentioned, which can cause a lot of problems with antimicrobial resistance. And but I will, in particular, talk about our research topic, which is done at the TIHO, and I have to correct, I'm not the director of the TIHO, I'm director of the Institute of Microbiology. Our present director is Klaus Osterrieder, who is a virologist. He's just newly started his job. So at our institute we have several research groups, and one group is led by myself, and we are dealing with host pathogen interactions in this respiratory tract. And I learned that we have also an online audience, right? Anybody listening online? So I stick here and we will use this pointer here and try to, let me see, the laser pointer is here. So maybe you can see it and the audience also. Okay, and I can use this here. Oh yeah, works. So first of all, a short outline of my talk, which is approximately 20 minutes. We are a little bit late, I think, so I will try to speed up, but I think the coffee break is long enough. So I will first start with a short introduction, and then I will focus a little bit on some ex vivo models, and this is maybe the second link to what we heard before, because we need models to test chemical or bioactive compounds in conditions or under conditions which are as close to in vivo situations as possible. And we are using two such models, which are old models but are re-emerged in recent times. And then I have two examples of studies we did using these models. One is on a co-infection situation with swine influenza virus and Streptococcus suis. Strep suis is my favorite bug. And the second example is the co-infection situation with Bordetella bronchoseptica and Strep suis. And finally, I will give some conclusions on that. So starting with the introduction and models, first of all, the PRDC, and as I heard you are no vets in the audience, I have to consider this certainly. So briefly, the respiratory diseases in pigs in swine industry are most important worldwide. They're causing a lot of problems in animal health, in animal welfare, but also they have an economic impact because of the use of antibiotics which are needed to treat these diseases. And you can see from this schematic model here on the left side that it's a complex situation. Here the pigs or the piglets, young ones, are still looking happy, but when they are faced with these pathogens and this situation, they get seriously ill and they can also die of this. So we have some primary pathogens like PRSV, swine influenza virus, or some bacteria. And these primary pathogens, they certainly cause a primary damage to the epithelium. They can infect those pigs by themselves. They can cause disease clinical symptoms. But the problem is that comes into place when they have facing secondary pathogens. And these secondary pathogens like Pasteurella, Streptococcus, Bordetella, they are very often part of the commensal flora. So if you have a primary pathogen, then these pathobionts can cause an increase in severeness of the disease. And this is even more the case when you have environmental conditions which favor these situations for the pathogens, so which can lead to even some more severe progress of the disease. That is stress, husbandry conditions, climate management, and especially in intensive livestock keeping, then this is can maybe a big problem. And we are facing changes here also because we are facing transformation of agricultural systems worldwide due to climate change, etc. So it's a very big issue, not only in veterinary medicine, it's a big issue in One Health. So the pathogens which are causing problems, some are listed here on the right side in this table, and I have indicated in red those ones we are working with. That's number one is swine influenza virus, even though most piglets in Western countries are vaccinated. A big problem is PRSV. It's present in many swine. Then we have some bacteria mentioned already like Pasteurella, but we work with Mycoplasma, for example, and Strep suis, and also with Bordetella bronchoseptica and Actinobacillus pleuropneumoniae. So these pathogens are causing serious problems, and usually how this works out is that a primary pathogen, such as depicted here in this model, viruses or bacteria, can infect a epithelial mucosal membrane. It will cause some damage, will cause some induction of an early immune response, and may cause some kind of clinical symptoms, but then come into place, as I said, the secondary pathogens, and you can see it here in the model in the center part. The damage of the primary pathogens could be viruses or bacteria. They can, for example, damage the ciliated barrier, so they can be ciliostatic, or they can cause a reduction in ciliary activity. So this primary barrier is then destroyed, and this allows the secondary pathogens to invade the tissue and then cause severe disease. And this goes further on for the, with respect, for example, the modulation of the immune response, the local immune response. And we all know this situation or this problem from human medicine, where we have the influenza A virus, which is then co-infected or followed by a subsequent infection, for example, Staphylococcus aureus or pneumococcus. So we have this situation also in the human medicine. It's not only in veterinary medicine. Now, when we look for host pathogen interactions and we want to understand these co-infections or polymicrobial infections, we need models. We can do it in animal models, that's one thing, but it's quite complicated, especially when you not only have one pathogen but two or more pathogens. So it's a very complex situation, and that's the reason why quite little is known, surprisingly little is known about these comorbidity mechanisms when two pathogens or even more infect a host tissue. And in veterinary medicine, we have the big advantage. We have easy access to tissue of animals, and these are the natural hosts, like here the pig. And we can use these tissues, as I will show you in a minute, to develop ex vivo or in vitro models. And with these models in cell culture, you can use standardized and stable methods to monitor the infection, to have different kind of outcomes, to see for virulence properties, to see effects of the co-infection on virulence and pathogenesis. And this has long been done with simple cell lines, but we all know that it's far from the in vivo situation. So in the last 10 years or so, we are more and more using primary differentiated cell from the natural host, ex vivo, in vitro models, and in veterinary medicine, as mentioned, we have easy access. So it's much easier as compared to human medicine that we can use these models. And that's why we are using two kind of models since quite a few years now. And I would like to shortly introduce you before I come to some results we obtained with these models. And one is the porcine precision cut lung slice model, PCLS, on the left side. This is a model which has been invented decades ago in the 1980s, and then maybe has been forgotten a little bit, but nowadays we have quite advanced technologies for imaging, for sequencing, single cell sequencing, things like this, so we can have much better readouts of these models than before. And the second model I will introduce to you is the air-liquid interface, the Halle culture model with differentiated epithelial cells, and which is also a model which has been invented several years or a long time ago, but is now more and more used in human and animal medicine. So first to the PCLS. Briefly to show you the procedure, and I'm explaining this because this was one of the parts where our GLACIER students have been involved in, which I will shortly introduce at the end of this talk. So first of all, we prepare these tissues from the lungs of slaughtered animals. So notably, no animal has to be killed. We just go to the slaughtery, we get those lungs, then they are transported into the lab. Then we take the cranial lobes of certainly apparently healthy lungs. They are dissected, and then they are filled with low melting point agarose to keep the structure, because this is the advantage of PCLS. It's a tissue which is resembling the natural architecture and structure at the time point of extracting. And then the next steps are that we use a tissue slicer, so to cut slices. These are approximately 300 micrometer thin, and we are using a Krumdiek tissue slicer for this. This is quite important to have the appropriate equipment to do this. Then after that we have to remove the agarose again with cell culture medium, and then we can put these rings, as you can see here, in a 24-well tissue microtiter plate, and with these rings of the tissue we can then do infection experiments, and we can keep them up to 2 weeks at least. Notably, you can do this technique certainly also from lungs, from other animals, from humans, and this technique is also possible from tissue, for example, from the brain, from the gut, etc. So we are doing a respiratory infection, so that we are focusing on respiratory tissue here. Now, one advantage we have here also is that we can measure the ciliated activity, or the cilia activity, and I hope you can see this here, the activity of the cilia surrounding here the bronchus, and this is also for us a possibility to monitor viability. I think now it's, I go back, you have seen it, I hope. Let's see again. You know what I mean. The movie has been seen, right? Okay, I wasn't sure because here and here. Okay, anyway, so you can use this cilia activity to monitor the viability of the tissue and the suitability of the model, and we only use tissue which has at least 90% activity of the cilia, and then we can also use it later on for a readout. Now, if you see this tissue here in tissue samples stained with HE, for example, on the left side, an A, and here immunofluorescence, yeah, I think you can see it quite nicely here, where you can see in dark the cell nuclei, then in red the ciliated cell, that's a beta-tubulin staining, and then in green this has been infected tissue, that is, for example, Bordetella bronchoseptica. And here on the right side you see in a scanning electron micrograph of the structure of this tissue. So the PCLS is one model. It has the advantage of, as I said, it's that it has the same architecture, the structure, all the cells which are present in this tissue, so you can do an infection under closely in vivo mimicking conditions. What you cannot do is you cannot do with infection with a single cell type. You cannot do any barrier studies, so translocation across a membrane barrier. And for this we use the ALI cultures, the air liquid interface cultures. Again, I would like to shortly introduce you to this. We again take lungs from the slaughtery, then we take tissue and bronchi, which are dissected from the lung parenchyma. Then we scrape the cells from the tissue, we propagate them in collagen-coated tissue cultures, and then seeding them on collagen-coated permeable membranes. So we transfer them finally after about 1 week into transwell chambers. And in these transwell chambers, we first keep the cells under conditions with full medium in the upper part and in the lower part of this chamber, so the apical and the basolateral part, so to say. And you see first the polarization of these cells, so you can do nice translocation studies with these models. And later on, you have a differentiation. When you remove the medium, then you have the in vivo condition with air on one side and medium on the other side, and then you can also flip all the systems, so you can do all sorts of things with this. But then you can have construct a differentiated pseudo-stratified epithelium, and you can do infection and translocation studies. And here also some pictures. I show you the electron micrograph, which is shown here in A, and you can see here the situation in polarized cells. So this is an intact monolayer. And here you see, after several weeks later, you see the differentiated cell with a lot of ciliated cell. And you can also see this in immunofluorescence, here the polarized cells, and here the differentiated cells, and here you see staining for the beta-tubulin, for example, and you can see the pseudo-stratified multi-layer cell layer, and here you can see also basal cells, for example, so you can also measure, for example, effect on the thickness of these basal cells. So this allows you to do a lot of do a lot of interesting studies. And you can also measure the electrical resistance of this barrier, and these show you below here the tier measurements, so the transepithelial electrical resistance. And you can see here these are 4 batches, for example. You start with the early conditions here, and you can see at the beginning you have a lot of variation of the tier measurements here, but later on it's getting more and more stable, around 200 microohm per square centimeter, and then you can see that's a stable barrier. You can certainly use also fluorescent labeled dextran, for example, so other methods to measure this. And then you can do, as I said here, you have a nice cell layer of highly differentiated porcine epithelial cells, and can do nice things with it. For example, what we did, with this I would like to introduce you shortly what we have done with co-infection questions we had. The first one was swine influenza virus and strep suis, and the second Bordetella bronchioseptica and strep suis. And to the first one, swine influenza virus, why we choose swine influenza virus. As mentioned earlier, this is a major pathogen in swine. It's even though it's vaccinated against swine influenza, we have swine influenza infections. It's a porcine pathogen which causes high fever, coughing, sneezing, and we have different subtypes like the low pathogenic H1N1 and the highly pathogenic H3N2. And we later on used all these 2 subtypes, also the swine influenza virus subtypes, because we can compare pathogenicity and the effect on Streptococcus suis. And Streptococcus suis is more or less an opportunistic pathogen which by itself can cause some disease, but it needs some factors which enhance the susceptibility, and in a way it mimics the pneumococcus in human medicine, one could say. So it's part of the commensal bacteria, but also a pathogen. Now, when we did these co-infections, we usually did a pre-infection with SIV, as you can see here, and then after certain time points we did a subsequent infection with strep suis. You can see we always have the 2 strains here. One strain is a wild-type virulent strain which is encapsulated, and the other one is non-encapsulated. And the reason why we compared it is because the capsule is an important virulence factor, and we wanted to know how does this capsule contribute in the co-infection scenario to the pathogenesis. And then we determined ciliary activity, bacterial growth, virus titer, and some other things. And to summarize this, what we found after the early stages of infection, we found, as you can see here, in the immunofluorescence on the left side, the virus in infection after 24 hours with H3N2, the highly pathogenic virus type. This is strep suis wild type. You see the small green dots, they are adhering bacteria. Because this bacterium is encapsulated, usually you have a poor adherence. And then you have the non-encapsulated mutant, which also shows some adherence. If you quantify this, usually the adherence is higher in the non-encapsulated. And then we did the co-infection and stained this, and you can clearly see, I think here in the center, when you have a co-infection, you have a clearly enhanced attachment of the strep suis and colonization after 24 hours. And you can see on the lower part here that the strep suis seems to preferentially attach to the virus-infected cells. And this is not the case when you have a non-encapsulated strain. So what we found out here is that swine influenza virus can pave the way for the attachment colonization in the early stages of encapsulated bacteria. And we can also quantify this. You see here strain 10, you see clear and increased adherence in the encapsulated when you have co-infected with SIV. And the theory behind it is that infected cells, SIV-infected cells, express hemagglutinin at the surface at a certain stage, and that the capsule of the bacteria can attach to the hemagglutinin as a structure. And we have done some more studies on the linkage types and some specificity, but it's very clear that this adherence step is mediated by the hemagglutinin and the interaction with the capsule. And that's why the non-encapsulated strain did not show any difference in mono versus co-infection. So we also looked at later stages and looked at 72 hours. Again, you see here staining with a tubulin, and you see the infection with strep suis and a mono-infection. You can see here the non-encapsulated strain shows a much better attachment here to the lining of the cells than the wild type in the beginning. at 72 hours, so later stages. And then if you look at the H3N2 co-infected cells, so previously infected with a virus, you can see the green dots here, they are invaded deeper into the tissue, and you can hardly see any ciliated cells. So there is a damage to the ciliated cells, and then this confers a better invasion. And if you use the other virus type on the right side here, you can see that it stays the same, so there's no real effect, and the ciliated cells are intact. So that means that this virus type with low pathogenicity, with low ciliostatic effect, also has no effect on the subsequent strep suis invasion. So we have two steps here, the attachment and the invasion. And to confirm this effect of the virus on the cilia activity, you can see in red, this is the pathogenic virus, a highly pathogenic one with a clear effect on the cilia activity here, in red, and in blue, the other virus does not have any effect at these time points. So that fits quite well to this theory that the cilia were the reason or the modification of the cilia for this paving the way effect. So to summarize, we can say in this first part that the virus promotes adherence and invasion of PCLS by strep suis in a two-step procedure. The one is initial capsule-dependent effect by the hemagglutinin attachment, and the other is the favoring the subsequent invasion, which is independent of the capsule, and this is due to the SIV-induced ciliostasis. So this one thing we could nicely show with such a PCLS model. And in the second example, I would like to show you what we did with a pathogen. And here the reason was that Bordetella bronchoseptica is also a very important pathogen in PIDC. You can see on the right side here the Bordetella. And we know from the literature that this is a pathogen which is causing respiratory diseases, not only in swine, but also in many other animals, also in humans, in fact. It has a lot of toxins and factors which can cause some damage, even though surprisingly little is known about the pathogenic mechanisms. And one thing we knew from the literature for a long time is that when you have a Bordetella infection in swine, you always have a enhancement of severeness of disease by other pathogens, not only strep suis, and this has been even used for experimental infections, as seen here on this study here from the 1989 paper in American Journal of Veterinary Research, where they used Bordetella pre-infection to get a successful infection with strep suis in piglets, because it's not easy to do a piglet experimental infection with a strep suis, and they used in fact Bordetella bronchoseptica. So for us it was clear maybe Bordetella bronchoseptica, known to have some effects on cilia, have similar effects or not as SIV on a subsequent bacterial infection by Streptococcus suis. So this was a question, and what we first did, we did a setup, an experimental. We did some studies on the dosage. We wanted to have a ciliostatic effect, but not a complete damage of the epithelium. So what you see here is in the first 24 hours you have a monoinfection with Bordetella bronchoseptica. This is the cilia activity again here, and you can see that the cilia activity is reduced to, let's say, 40% after 24 hours, and then we started with the infection with strep suis, so the co-infection, and checked on the effect on cilia activity and the subsequent stage of co-infection. You can see the cilia activity goes down as long as Bordetella bronchoseptica is present, but if you use strep suis alone in monoinfection, there is no effect on ciliostatic or on cilia activity, even though strep suis possesses some toxins like the suizycin, and which we also tested here, but I don't go into detail, that's too far. So we can see that Bordetella bronchoseptica reduced cilia activity by about 50%. It goes further on if you wait longer. And on the other hand, the cilia activity was not impaired by either of the strep suis strains. So this was clear something which strep suis would profit from a previous cilia activity modification, because it cannot do this by themselves. Then we also looked for the toxicity, because strep suis, as I said, has a toxin, the suizycin, and we checked the LDH release, which is a standard cytotoxicity assay. And please just look at this indicated area here with the blue square. What you can see here is when you have the wild type strep suis strain, you have quite some cytotoxicity, even though it's quite low, surprisingly low in this PCLS system. When you have cell lines, it's much higher. And then if you have the co-infection, it goes up dramatically. So that means that obviously Bordetella bronchioseptica has a clear effect on the cytotoxicity of strep suis, even though if you do the monoinfection Bordetella in the LDH release assay, you hardly see anything. So it's another type of toxicity expressed here by strep suis. So there are two toxicities coming together: the cilia activity toxicity of Bordetella having some effects, and the cytopathic effect on the membrane measured in the LDH release of strep suis. So they are both combined. So this is an effect which is certainly raises a question: how does Bordetella increase the LDH release and cytotoxicity? And the most obvious explanation is certainly because, as shown before, it enhances the colonization, and that is what you can see here. This is colony-forming units in strep suis and Bordetella bronchioseptica, and what you can clearly see here after from 4 hours to 24 hours, again, this is the wild type here, this bar, and the other bar here, for example, is the co-infection. There is a clear increase, at least by tenfold, when you have a pre-infection of Bordetella bronchioseptica. So most likely, by enhancing bacterial growth of strep suis, Bordetella bronchioseptica can also enhance cytotoxicity, because more bacteria obviously can produce more toxin. And then we also did studies on to check whether this is all due to the suilicin. And to make a long story short, this effect is certainly dependent on the toxin suilicin. So co-infection of Bordetella bronchioseptica promotes adherence and invasion of strep suis, and the invasion and adherence of Bordetella bronchioseptica itself is not affected, so it's independent. We also did some further studies. I would like to reduce it to this picture, and this picture you have seen on the beginning also, because we found that both pathogens seem to not compete for attachment sites. So this effect is beneficial for strep suis. It does not have any effect on Bordetella bronchioseptica, but also not a negative effect. So it seems that the Bordetella bronchioseptica on the left lower part here attaches mainly to the cilia, whereas strep suis is only in close vicinity. And this has been in this colored picture by Manfred Rode. We can see that obviously Bordetella bronchioseptica attaches to different sites as compared to strep suis. So they can nicely live together, and that's what they in fact also do, because both, as I said, are also part of the commensal microflora. So the conclusion, what can we conclude? I think we conclude that these co-infection scenarios are interesting to study, because these mechanisms between two pathogens, which can either be beneficial or not for the pathogen, in fact in most cases are not very beneficial for the host. Little is known, so there's a lot to do on these aspects, and the two examples hopefully show that it's quite interesting to see how one pathogen can affect another pathogen and somehow explain the severity of a disease, even though we have not looked at any stages of the immune response. So certainly we have these initial stages which I showed in the beginning. So we have a primary infection, then a secondary infection. We have shown these effects on ciliated cell, on invasion, but certainly interesting as a next step is to look at the host response, because if you use PCLS, for example, you only have the resident immune cells. There is no recruitment of immune cells because you take away the tissue, and that's why for the future it would be interesting to see in co-culture models where you add immune cells, what about the immune cells and the immune response, not only in immune cells but also in epithelial cells. And we have done some studies on this, measured cytokine responses and transcriptome analysis, and that showed us that there's a clear effect, and that this certainly immune response plays an important role in the later outcome of the disease. But this is what is depicted below here, and this needs further studies to speak about this. Now, with this, I would like to come to an end. I would like to acknowledge people who did this. First of all, Desiree Schaaf, she did a PhD on this project, and later on now is a postdoc. Maren Willenborg, she was a PhD student, started with the model itself and did studies on the toxin suilicin and then several others. And I would like to point out here with this red area, arrow the Gloriana Castillo-Badia, and she was a guest student funded by GLACIER, and she was involved in the project on PCLS Bordetella strep co-infection. And then we had cooperation with pathology and virology, and we certainly had some external partners. We had some funding from EU, DFG, etc. And last but not least, I would like to summarize the contribution of the GLACIER-funded students. As I said, Gloriana, she did a characterization study on bacterial pathogens in our lab last year. She's from San Jose, Heredia from veterinary medicine. Another two students, Amanda and Carolina, they did different kind of studies at the TO, one on porcine pathogen interaction with neutrophils in the biochemistry department. She's also from San Jose, from the UNA, and Carolina, she did a study on Enterococcus faecalis infection in chickens. So all three did some studies on host pathogen interactions as a THO, and it was a real pleasure to have them in the lab, and thanks to GLACIER that we could make this possible. So with this, I would like to thank you for your attention, and I'm open for any questions.
Thank you very much. We have time enough. We have planned graciously with the breaks. Okay. Any questions from the audience? Yes, please.
Carlos.
Peter, nice talk. I have two questions. One is regarding the models. From your expert point, what are the limitations of the models, particularly for testing drugs? The second is related to your co-infection studies. You mentioned Bordetella. You know that I like Bordetella a lot. Since the early times, elentuonamen was mentioned in the role as bridging adhesin of filamentous hemagglutinin. Have you done some studies to look if this might be playing a role using knockout mutants or whatever?
First to the first question. The disadvantage maybe of this model certainly is that you take tissue from different animals, that you have quite a variation between animals. You certainly take only healthy animals, that's clear, but there still is a kind of variation between the different animal sources, but the variation is not very, in PCLS at least, it's not a big variation, so it's quite standardable. So we have done many studies on the effect of the toxin. We have done these Bordetella studies. We have measured ciliary activity in the PCLS, and it always in the same range, so it's quite reproducible. The disadvantage in PCLS certainly is that you do not have the possibility to do studies with single cell types. You can visualize them certainly, but you cannot do the same like with the Ali culture. And you don't have any, as I said, immune cells, except the ones which are resident at the time of taking the material. But we have done studies with co-culture of the tissue with porcine macrophages from the lung, so this is possible to do. So I think this model can be developed into a more sophisticated model in the future. So I think, and the big advantage to say this is PCLS is quite easy to produce. The method is, I would say, not very complicated. And they can kept, as I said, until 2 weeks. Some others reported for other animals even longer. So it's nice. It's difficult to freeze them. That's a disadvantage. So you need the fresh material. There are studies who try to cryopreserve the PCLS, but with limited success. And we also we are always taking fresh material. With the Ali, maybe shortly, the Ali, as I said, there is also some variation from animal to animal. You have to get rid of the fibroblasts, you know that. That's some of the problems, but that can be done if you have a good protocol, and we have developed this and established it for many years now. That works very nicely, but it's much more complicated. And there are many pitfalls along the preparation. It takes several weeks. When you then have the finalized model, you can use it for 3 weeks. That's fine. And the advantage is that you can cryopreserve it. The disadvantage, as I said, is it's very complicated, time-consuming. It only consists of the epithelial cells, again. But then you can again do co-culture experiments. So this is for the models. This question, okay? Good. And with the toxins that...
it seemed that we need to be very cautious in using this system for compounds that have a bad bacteriostatic activity, for example, but very robust.
I personally think that these models should be used in the future much more to do studies on, for example, antimicrobial resistance and things like this, because it's really quite close to in vivo, and you can do, at least with PCLS, easily do many studies to do the statistics on it. And okay, then to the second question maybe. With the toxin, that's a very good question, because we also did studies on using FHA mutants and the DNT mutants. It's another demodecrotic toxin in Bordetella bronchoseptica, and we were clearly speculating that these toxins have the effect on the cilia, and to our surprise, not. So we are not clear what the reason is. We did many studies of mutants we got from Susan I just forgot, the one who is working on the rabbit Bordetella. We got DNT mutants, FHE mutants, but there's no clear effect on the celiostatic effect and on the co-infection. So it seems independent of these toxins. And there we realized very little is known about Bordetella still, even though you did many studies a long time ago, I know.
And these toxins, is that small molecule toxins or peptides or proteins?
These are peptide toxins.
Peptides,
yeah.
okay.
And any further questions to the toxin can be probably answered perfectly by Carlos. But yeah, it's peptide toxins. And the toxin from Streptococcus is also a protein, which is a channel-forming, pore-forming toxin. That's why we measured it with the LDH release.
I really like this tissue models, because they overcome a lot of the problems which you have with cell cultures, of course. Though I wonder that yeah, you talked about a little bit about the difficulties, but still, I mean, you take real life slaughterhouse material. So don't you carry in with that a lot of not only mycobacterial problems, but also other things which are sitting in the lungs? So how do you manage then to have good cultures at the end? Yeah.
Well, certainly from the beginning on we have antibiotics in the medium, but we don't have any big problem with contamination later on. Then we remove antibiotics when we use it and infect it, so we don't need the antibiotics later on, but after preparation. And you could also argue, I mean, certainly you have all those sorts of things. You have the microbiome and things like this, depending on where you take the material. From the slaughtery, you think everything is dirty. You have to be well at the site of the slaughtery, to be there and take the lung yourself. You cannot just go there and give me a lung, yeah, because you have to be aware how this is taken. But on the other hand, you have the situation which is natural in pigs. So you look at a situation, when you do this often enough, it's much closer than would you have a sterilized material. The thing is, then once you do the infection, you can, as I said, you can remove antibiotics, do it antibiotic free, and we don't have any problem with contamination there, or hardly any. It's more difficult in Ali when you detect a contamination after 2 weeks, everything is gone. In PCLS it's easier.
The problem with veterinary drugs is, of course, when you, I mean, I'm coming from drug development, and so is of course that people say, okay, first of all, it cannot cost a lot, and then no one develops specifically or rarely specifically for veterinary purposes because of the cost involved. So you rather take the trickle out from the human drug development. But vice versa, it could be interesting because, of course, testing antivirals, for instance, is a problem because you need the cell host for the virus in a way. So the question is, can we use the systems you presented to test for antiviral compounds, for instance, and what would be the throughput which you can manage if I sent you 1,000 compounds, you know?
Well, we cannot do it. In fact, we have a small group, and in fact, I will retire soon, so I cannot do it myself. But anyway, I think that's in fact a good idea because, first of all, the pig is a very good model for human diseases, as you know, and the porcine lung is very similar to a human lung. You can, with a PCLS model, I would say it's cheap enough. who do many studies. I can't answer the question how many in a row, but the virologists who have developed this model previously, before we started with co-infection studies on it, they're doing a lot of virology or virus infection studies with these with the PCLS. The scaling up is a difficult question, so maybe one has to first screen in a different model and then go in the next step into the PCLS model and then into the animal. So it's something to refine the experiment later on in the animal, but it could be a good model to fill the gap between primary cells and animal experimentation.
If there's no more questions, I don't see any, then we will have our coffee break until 11:15, and after that we will have our chemoinformatics, AI, artificial intelligence, machine learning, whatever session. Yeah. But keep in mind, any artificial intelligence can only be as intelligent as the human intelligence generating it.
Presentation? Pardon?
Can I transfer my
Yes, please.
presentation? So, you use the mouse for a presentation, or is there?
You can do it. I miss the pointer. I have to look for it. Which folder?
GLACIER, yeah. This one.
And who else?
I think, can I remove it?
Not yet. It won't let me. Yep, you're welcome. Not yet recognized.
Oh yeah. Thanks.
Which one?
That one. The one.
This one.
Yeah.
Thank you. You're welcome.
Ja, schön, dass Sie da sind. Dann sehen wir uns mal persönlich.
Ja, tut mir sehr leid, dass ich könnte nicht länger bleiben, aber es gab ein paar Sachen im Vorfeld, dass ich ja nicht umorganisieren. Ne? Ich war schon committed für andere.
Ja, ja, es ist ja auch, August ist natürlich Urlaubszeit und so. Wir sind dankbar, dass die Leute kommen.
Nee, mein Problem ist, ich hatte ein paar Gesundheitsprobleme und ich hatte ein paar Termine zum Arzt für Check-in etc. klingt ein bisschen doof, aber ich hatte viele Nebenwirkungen nach einem RNA-Impfstoff.
This one?
Sind Sie nicht der Einzige? Also,
ich hatte, okay, wir reden nicht über etwas trivial wie abgeschlagen zu fühlen für eine Woche oder kognitive Defizit für eigene Monate. Aber ich hatte, nee, was war schlimmer? Hallo.
ich hatte was von Covid selbst. Also, das ist wirklich Sprachstörung.
A minute, I need an adapter.
Ja, die Sache, sagen wir, Post-Vac, Post-Covid, was man sich neurologisch messt, ist ganz ähnlich. Natürlich wird nicht so viel geredet, aber in der Realität. Und es gibt viele Fälle bei Covid-Patienten und bei Individuen, die sind geimpft, wahrscheinlich das Spike-Protein hat etwas zu tun, dass natürlich geben etwa, das ist im Prinzip eine myalgische Enzephalobilitis. Das ist die Realität. Aber das ist nicht die einzige Sache. Ich sage zum Beispiel Populationprobleme. Ich hatte Probleme von der Aggregation von Thrombozyten, riesige spontane Hämatoma über 10 oder 11 Monate. Ich habe die ganze Studien im Gerinnum-Labor. Normalerweise, wenn man macht eine Aggregometrie, die Thrombozyten machen eine Kurve. Ich habe 6, 7 Sachen weggeworfen. Bei einem hat gut gewirkt, bei der anderen war eine verpettete und niedrige an Bord, bei anderen war eine flache an Bord und die anderen haben absolut nicht bewirkt. Aber ich hatte richtig riesige Hämatomas, an einem Tag aufgewachsen mit einem blauen Auge. Ich war ganz komisch, weil der Kerl von dem Gerinnum-Labor hat mir gesagt, wenn Sie keine Sorge, Herr Gutzmann, wenn Sie haben eine Elektiv-OP, Sie müssen eine Infusion mit Solalarm machen. Ich sage, ja toll, denn ich habe eine Elektive-OP, was mache ich, wenn ich habe einen Unfall im Auto?
Ja, das funktioniert nicht so.
Und wenn ich habe blaue Augen und ich habe Hämatomas, die Zahl an Thrombozyten war normal, no? Aber wenn ich habe Problem mit Population überall, warum ich kann nicht im Gehirn, no? Bluten und dann bin ich vergau oder nicht gut mit.
Ihr kennt euch?
Hallo.
Hallo. Das ist eine Barbara. Sie ist auch der Chef von
Ja.
Ich muss mal ganz schnell nach
Okay.
Okay.
So.
Sorry, I need to pick this one.
Und mich kannst du rauswerfen.
Gucken wir, ob ich muss in
Second session. Here we go. Where are we?
Könnten Sie sortieren für Datum? Das kann nicht sein. Das ist nicht meins.
Intendo?
Nee, das kannst du rauswerfen.
Das ist die
Okay. Yeah. Then it is not this one. I need to stop. Zack. Halt. Ja, jetzt. No.
Ich habe mich verwundert, war fast leer.
Okay.
Okay, jetzt ist vielleicht ein bisschen einfach. Wieder bei Datum? Die zweite. Glacier, Halle, Final. Gut.
Okay. Close. And eject. Okay.
Vielen Dank, erledigt.
Bitte schön. Ja. Danke auch. Jetzt.
Sie wollte nur das Foto, aber dann habe ich gesagt, wenn schon, denn schon.
Warte mal. Ach, ich wollte ja eigentlich schreiben, ja. Ja, gar nicht. Und Hä?
Ich höre.
Wieso ist Ende? Ist da eine Animation?
Nee, eigentlich nicht.
Aber niemand hat was angemacht. Meine Frage war, wann die Teamsitzung kommt, weil da muss ich noch mal ein bisschen...
Also,
Es geht, es ist eine automatische.
Also, ein bisschen Schwierigkeiten mit der Zeit, weil Herr Westjohann jetzt eine halbe Stunde Verzug produziert hat.
Mhm.
Allerdings fällt ein Vortrag aus, habe ich heute Morgen die Information bekommen, dass er wirklich gar nicht kommt. Und das ist der letzte Redner vor der Pause. Also hier haben wir 3 Stück und das fällt aus, aber wir sind im Verzug, also das bleibt, glaube ich, ungefähr so. Ja,
Das ist der, also nach der
und
Kaffeepause.
ich habe
Super.
Herrn Hoffmann gesprochen, er ist ja hier vor Ort. Er wird die Folien hier zeigen und sie redet nur.
Das ist nicht das. Nee, das ist an sich geht das.
Genau. Also,
Ach so.
guten Morgen.
Also, naja,
Herr
gut.
Hoffmann ist schon da.
Ich bin da, aber der letzte Vortrag.
Genau. Aber wir sind ja ein bisschen verzögert.
Das heißt, es bleibt alles so.
Ich gehe mal davon aus, weil hier am Vormittag haben wir jetzt ein bisschen mit Fragen Zeit verbracht. Ne?
Also es ist der hier, mag ich mal sagen.
Genau, der kann gar nicht kommen.
Die High-Vorträge alle überziehen ein bisschen.
Also ich glaube, das bleibt dabei.
Ein bisschen länger.
Genau, also hier,
Ist das
nach
jetzt
diesem
Session 1 oder
Vortrag kann
The
ich
last
sofort mal,
The Last One? Okay.
ne, wie das aussieht.
Ja,
Ja, wie eng ist das bei Uwe Schneider, der Kalender bei dir? Oder hat sie jetzt eine Zeit eingeplant? Okay.
Ja, ich kann mir kurz anschauen.
Gut.
Perché malas informazioni.
WhatsApp ist noch einfacher.
Mandelbonies.
Bis?
Mandelbonies, Michael Mandelbonies.
Jetzt sind die
Ah, no, this is not I'm sorry. It should be Germany.
This one?
Germany, yes. Sorry.
Oh, I'm sorry.
Okay.
Yeah, so we will check.
Can I, yes. I have to check.
That.
Yes, but can I see our program? We don't have any. Okay. And can I see the, if I want to see the,
Previous.
no, if I want to see it like this, just,
It doesn't work.
it's the
No,
point.
we don't have a...
No, you can. No, never mind. So leave it. The regular. I will switch back. Okay. Excellent. And if I want to have to change to change it to the red one with the eyes.
That is... Pointer options?
Where is it? Where you can? How it? Tome, you will be here, so you will do it for me. Thank you very much.
You're welcome. Yeah, I need to eject it. So close it down. Where's it? Here. It is still busy. I need to... I don't want to unplug it.
I have a lot. Nothing will not happen.
Sorry, so I need to close it. Still, the system still has some handle on this.
Never mind. Just take it out.
sometimes it refuses to maybe we can have a different.
Thank you very much.
Du kannst mit mir. Ich komme auch gleich, ja. So, ja, Anna hatte, wo sie die hingelegt hat. Ah, hier. So, und da muss ich erstmal wieder.
Das ist diese blöde Auto-Weitermach-Sache.
alles klar? Are we ready? Maybe? Are we ready? Okay, welcome to the real virtual session. For the next speaker, I don't need any notes because Mehdi is actually the head of the chemo or computational chemistry and biology group in my department. And I'm very happy to have him here also as a speaker because he combines many positive things. So he is not only runs the standards which we do nowadays in computational chemistry. Can the music? I mean, it's nice to have some background, but he does not only do the standards in computational chemistry, that is things like SAR or docking, molecular docking, stuff like that. But we also, in our department, always wanted people who also develop own tools and not only use standard programs. And he does that. And where did he get these abilities? Well, he studied actually in Iran and did his PhD there. Then he moved to Germany to the quite famous Max Planck Institute in Göttingen on what is it? Physical. Biophysical chemistry. Yeah, we know it well. And after that, he went to RWTH, so Rheinisch-Westfälisch-Technische Hochschule Aachen, which is one of the big technical universities in Germany, where he became a group leader and sort of habilitant, which is internationally speaking something like assistant professor. And after that, and he did many fantastic projects there, and so we were really very happy to be able to win him to head the group of computational chemistry at the IPB. And so I hope you will also enjoy the work which we do and which is growing on Mehdi's turf. Mehdi, please.
This one?
Yeah.
Go ahead.
So, hello from my side. Good morning. So first, I would like to thank Professor Westion for this generous introduction. It's really my pleasure to present in this wonderful symposium. And as Professor mentioned, actually what I'm presenting today is not what I typically present. So I have been working for long in protein design, but today, because of this meeting, I decided to show what we are doing in the direction of natural product for drug design. And I would like to share with you our view from a computational perspective, what we can contribute to expand the chemical space, especially in the area of AI. So maybe I missed the point. So I think I don't need to introduce natural products. So we had wonderful presentation yesterday, today, about the importance of natural product, but I just like to give you some numbers. In principle, we know that natural products have different applications. One of the most important applications has been since decades on medicine, and we see that in terms of proportion, 70% of natural products are coming from plants. And working at IPB, we are mainly interested in let's say, natural product from plants and, of course, fungi. But if you look at natural product from our perspective, I see more than 50% of the apple drug come from natural products. And interestingly, we see that 80% of most of these clinically antibiotics are originated or traced back to natural products. So this shows the high potential for this molecule. But when we think about natural product as a chemistry or in general drugs, so we have to keep in mind the complexity and diversity that we have in biological and also, as you see in this projection, chemical space. So if you see or look at here, so the estimated chemical space is about 10 to the power of 6200. And as you see, not all chemicals are drug-like or can be used or relevant for biological targets. So as you see here, so we know already we have something about 500,000, let's say, known natural product in the literature, but the great challenge is to find those that, as you see here, are fitting to the biological targets. And the current challenge in terms of prediction is to predict protein interaction with those molecules. But as we know, this is, in most of the cases, we don't know the protein target. And of course, there are protein targets which are not druggable, and we know already that we are not talking about one target, sometimes we have evaluable targets. So, and that's why I discussed yesterday about resistance to those molecules. And I would like to highlight more in terms of the vast chemical and protein space that we have also. So if we look at, actually this, just like to impress you with the numbers that we see, what is the largest number you can imagine in the Earth? Is the drug-like molecule, the chemical space. Based on this report from Biosolvit, one of the software developer for drug design, this is an astronomically huge number. And when we think about the protein, even very small protein, only let's say 100 amino acid, this is even bigger than this space. So we have a huge space, but this offers us actually a great challenge, but also opportunity. So there has been a lot of attempts to characterize and to annotate this chemical space in terms of what kind of chemical class or molecule we have. And as this is the work actually done by Raymond Group in 2012 in past. And as you see here, we have been able to use different methods in principle to characterize this space. I think Professor Medina Franco will also, I guess, explain this more. So he has done some fantastic work recently on describing so-called chemical multiverse. So how we can characterize the chemical space, and especially in terms of which one are biologically relevant. So, but the question is that we said that natural products are a very important source for that, but when we look at natural product, why this is the case? So natural products have high degree of statistical chemistry, so they have mostly, let's say, significant portion of sp3 hybrid carbons. They are not all planar. They have this, let's say, ring system which are not planar, which is very important, especially when you think about a drug for a specific protein. But when we look at the projection of natural product and the drug reported based on FDA, so you see that majority of them have not been already tested or not described as a drugs. So the question is that how, in principle, we can bridge these gaps. So in principle, we like to see from a computational perspective, how can we understand, explore, or navigate this large chemical space to bridge these two gaps from known chemical to full-scale chemical space. But there has been a lot of effort to do that, but the question is that from computational perspective, how we can do it. So to answer this question, I would like to offer what we are doing in IPB. So in principle, as Professor also mentioned, we try to combine classical physics-based or mechanistic-based approach. As you may have heard, we use in principle molecular modeling. I will explain in a few minutes. We can use different methodologies when we have already known structure from the protein. This is now mostly solved by AlphaFold, but also the structure of, for example, target. But we know that there has been a revolution in terms of high throughput screening, sequencing, robotics, and also generation of data, and also accessibility of AI algorithm. That shifts us in principle to see how can we use data-driven approaches. So in principle, how can we use those methods to even generate a model or to design a predictive model that can be used to explore this vast sequence or chemical space. So in principle, we try here, as you see here, to use already existing data to generate knowledge, but based on this already reported data. So, as mentioned, so we use classical approaches. So, you know, computational chemistry has been developed as a very complementary tools to experiment. So we have been using a lot of different techniques, including computer-aided drug design, as you see here, to look at the interaction of the protein and target, and the ligands. So in case of change in conformational space of the protein or ligand, we are using molecular dynamic simulation. And as you see here, there are also other techniques, like from chemoinformatics, we try to develop those, for example, for a structure-activity relationship. So, and in principle, these are classical techniques, but as I mentioned, this can be still influential when we have already a known target and structure. So, yesterday, Dr. Robert Renner already explained that we have a very hidden treasure in IPB, which is a compound library of about 30,000 compounds, and we try to see how we can use this as a case study by let's say, screening, biological and visual screening, in order to, for example, work on human and plant health issues. So in principle here, we are trying to provide complementary techniques to filter. I don't need to go to details why, for example, visual screening can be efficient in terms of throughput, but as you know, so we have a lot of compounds, so in principle it's very hard to keep those library, but also, so we need sort of pre-filter in order to focus on a specific compound. So for that, we have developed pipelines, as you see here. In this pipeline, we are, in principle, has been developed by Dr. Andres Vasquez in my group. We are trying to make a platform which we have the chemical, as you see, these are in principle IPB database, but also other databases. So we have a large number of chemical databases, and also depends on the target, either from human or plants. We try to do this matchmaking by different methods, including QSAR modeling, and also in principle, when we have a development model, to do a de novo design. So in principle, those methods are relying on mostly machine learning based approaches in order to screen those library and also to develop a model for structure activity relationships. So, but I mentioned that we think that how we can explore more this exercise. So that for that, actually Andres is working on different methodologies to enumerate the chemical structure or combinatorial design, and in principle, our method is focused more on fragments-based ligand design, as you see here. So there are some advantages of doing that. As I said, we can here, in principle, favor the protein-ligand complementarity, and of course, we have the potential that to explore more the chemical space, as you see here, by choosing proper fragment, and in principle this has been shown to reduce the false positive in terms of protein ligand interaction. And this helps us to in principle adjust the ligand to the protein of interest. So for that, these are the steps involved. So in principle we use our chemical library to deconstruct the fragment, and then to use that to design a new fragment library collection and reconstruct it for a specific target. So this recombination, we believe that it offers a way to explore more this database and also expand the chemical space. But I would like to show you another project that I think yesterday has been partly presented. This has been a project that has been developed since years in IPB. So we are in this project interested in understanding and developing model for interaction between the source of the natural products and their activity. So in principle, we know that there are a lot of literature data, but these are not integrated in one specific databases, and this in principle hinders to identify new plants species which has not been explored, or in principle to look at, or for example, the molecule that has been already identified as a drug, and to do it, for example, for repurposing. So that's why we focus in this project in the frame of also GLACIER on extracting and correlating this triangle. So in principle, focus on Cuban plants, natural products, and their bioactivity using machine learning tools. But for any machine learning tools, of course, you need to have a data to predict or to train a model, and then of course to predict. So for that, of course, there are a lot of course data, but they are not mostly, they are unstructured. So this means that we have a lot of database, but they're not integrative. Most of this data are hidden in the literature, but we need in principle to generate a so-called golden set to develop machine learning model. So at IBP already a PhD Anna-Katrin Hartig, has been working on developing rule-based method. In principle, in this work, so they have used expert opinion to annotate these three species, plant and natural product, with the effect using expert people. So annotating about 100 papers, the whole full text. And in principle, this is a golden set which have been developed, and we are trying to use this golden set to train in principle machine learning algorithm. So for that, mailing soon, in principle, use mostly language model, large language model, and those methods, as we know, they have been trained for specific purposes, especially in the direction of medical, but we have to fine-tune this for our specific application. So that's why this IPB golden set has been used for training and fine-tuning the parameter from this language model. So in principle, this language model is able to query the references after the validation, and this can be fed to a knowledge graph. So I will explain this in a few minutes. So in principle there, after development of this model, we are able to extract information from different texts, and this can be used to generate structured data for our knowledge graph. So for that, we are integrating, in principle, different source of natural product, existing one, and of course the literature data. But this is actually the general workflow. As I said, we are using IPB golden set corpus to train this model, and after validation of this model, this can be used to, in principle, extract information from other texts. And this is something that we are working on it. So in principle, this algorithm is able to identify the named entity recognitions and also try to see the relation between, for example, this species in multiple sentences, in principle, to get this structured data. So, but the question is that, okay, how we can use this? Already nicely Victor presented yesterday, I don't know, this has been moved. So N1 Health, knowledge base. So as he explained already, this is a very nice general universal platform that can be used to correlate, in principle, this triangle, and also to look at the co-occurrence. So in principle, he already explained that we can look at which plant has been already connected or linked to any specific natural product, and if there is any bioactivity reported. So this is a very general, I would say, platform which can be used and expanded for already other purposes. But what can be the potential of this platform in case that we could collaborate also with other, for example, I think Professor Medina Froco will present the Latin American databases. So in principle, by feeding this to algorithm, we are able to look at these relations. And this will help us to identify, for example, first, what are the underexplored region? For example, which species of the plant have not been explored so far? Or if there is any already, like we can assign or predict the bioactivity of any compound which there is no activity reported. And in principle, this can also highlight the potential of any candidate which can further use, for example, for drug design. So we are very open, and we are happy that Victor has developed this tool, and this can be a very good framework for, in principle, in combination with text mining to extract more information for a large, let's say, and comprehensive database of natural product. So I would like to show you also another example. Actually, there was a lot of discussion about the peptide. So we mentioned the peptide has gained a lot of interest as especially like a therapeutics. So in collaboration actually with University of Magallanes in Chile, David Menera, actually our new postdoc, we have developed a very comprehensive database of the peptide. So the challenges in the peptide field is that there are a lot of databases which are not integrated. So in principle, from this database, we have collected over 4 million peptides. And if we look at annotated one, we see that almost only 100,000 are annotated. So in principle, we in this integrated platform, we have collected all the available peptide, and if there is any bioactivity, also we try to develop tools to extract this from the texts. And here, as you see, this is a very nice platform. So, and this can be used further for develop machine learning tools. I would like to show you some examples that we have been already trying to do that. So, And I think this is very detailed, but in principle, for any machine learning algorithm, so you need the data. As I mentioned, Peptidea is a very nice platform. We have over 100,000 annotated sequences, and we have actually developed a machine learning algorithm in order to represent these sequences and also to classify them. So this classification helps us to, in principle, assign the function of peptide which have no annotation. So if you look at this is actually some of them are very surprising. We had the discussion Professor Westjohan mentioned yesterday. So we managed actually to get very high precision model which we can classify those peptides mostly based on their activity. I don't know if this is visible, but many activity like antibacterial, antifungal, and many other roles that as you see here, and this is the total number, so something about 60,000 peptides, and we are assigning in principle, their functions or by activity. But surprisingly, what we see is that when we look at Rosenfeld, as is known, we can also classify their toxicity based on reported in the literature. But what is also very interesting, this is also that, for example, for peptide, we see that they don't only show one function. So many of the peptide, they might have a so-called moonlighting, and they have different function. For example, if you look at the peptide, they might have different effects. And this is actually aligned with what, for example, yesterday presented, that they show sometimes two functions. So, but the problem here is that we know that this protein space is so huge. So this offers us opportunity, of course, to identify peptide which might be for a specific purpose or maybe multi-target. But of course, this is very challenging to, for example, predict if this has toxic effect. So, but this is something that we are interested and we are working on improving that. But here, I just want to show you what's the field is going, the direction. So this database helps us to predict the function for the peptide which they don't have any annotation, but it also helps us to develop completely new peptides which are not reported or are not observed in the nature. So there has been a lot of new development in terms of so-called generative AI to develop de novo peptide. And there has been a lot of development how we can, in principle, after training a model to in principle generate a peptide which is novel. And we actually, in this work, we have tried to use different model. So I would not go to the detail, but in principle, variation and encoder have gained quite a good accuracy and precision for predicting those peptides. And I like to show some of the analysis. In principle, when you generate a peptide, of course, this is in silico. How we can, in principle, validate this, at least in silico, before going for experimental validation. And as you see here, we try to analyze several properties of those de novo generated peptide. So, and we compare those properties, as you see here. For example, these are the peptides from our database, and you see that most of the property actually have similar distribution in terms of physical chemical property with already existing peptide in the literature. So this means that this field has a quite potential in terms of exploring protein sequence space, of course, to expand the protein sequence space and to generate new peptide, maybe for specific purpose or multi-target, for example, bacterial or whatever distance. So we have also one working, I would say, project, which is actually how we can use this platform to design peptide against HIV. And in principle, our first result shows that, of course, we can classify protein based on different, let's say, activity in terms of, let's say, are they antibacterial or not? So what are the target and what kind of reported mechanism it has been already reported. But now we try to again integrate this with the classical approaches for a specific target to look at, for example, structure biology. So to look at how they are, in principle, can be docked to a specific target. This is actually a work in progress, and we hope that we can update soon about the result of this methodology. So, but in principle, I would like to show you that here we have quite a lot of opportunity in terms of protein and chemical space, but we need to find how we can navigate this. So if we have such a, let's say, large number of protein, this can enable us, in principle, to predict the function or by activity, and of course to generate new molecules. So that brings me to the summary. So I hope that I convince you that, deeper understanding of the chemical space is very important in terms of drug design. and discovery, and of course, natural product represent a promising approach. And from our view, like fragment-based design can help us to explore and to expand this chemical space. And this knowledge graph also from, for example, Cuban plants also provide the knowledge for discovery of knowledge from existing literature. So with that, I would like to thank actually the people who did the job. I mean, some of them are here, and of course, the generous funding, and also Sihan was very instrumental in this project. I started Natural Product when moving to IPB, so thanks for your trust. And of course, I would like to thank you all for your attention, and I would be happy to take questions.
Are there any questions or comments already? Yeah, there's there with Professor Guzman.
Very interesting talk, particularly for somebody that is very ignorant. And my question is probably naive, but if I understood properly, you have your in silico driven approaches to select goodies that are tested to reduce time and money. Somebody did in the past some kind of blind or unblind screening to understand if the in silico methods are able to detect all the potential goodies, or there are a particular subset of compounds that are not, let's say, given a score by the conventional tools that are currently used in artificial intelligence to perform this pre-screening. This is my first question. The second is related to your toxicity versus activity. Toxicity is pharmacologically usually a function of dosage. The same thing that treat could be given toxicity, and is also a function of genetic, physiologic, pathologic, or pharmacological predisposition that might be affecting the modification or excretion of the drug. How is this taken into account in this modeling?
So, very nice question. I mean, regarding the first one, actually virtual screen has been used intensively, and we know that in principle the hit rate is not too high. In general, the literature shows 3 to 5%, but when you talk about, for example, our IPB library with 30,000 compounds, this only provide us so-called hint what kind of class of molecule we can focus more, and to start from there to modify. So the problem is that there is no alternative. How can we, screen those library against specific targets? So that's why the virtual screen can be used only the first filter. Of course, we know, as I said, if we have a specific target, one can put more time on it, but this is more or less a brute force way to look at millions of compounds and to see where you can find promising candidates. So it's not, I would say, the final, let's say, catalog, for example, testing, So you provide a list of compounds which can be tested. So the hit rate is less than 5%, at least from what I know from the literature and our work. But concerning the second one, second question, I mean, we have to assume that here we try to learn only from protein sequences. So we don't have any other information. So in principle, the assumption is that the knowledge is already in this data based on this protein sequence. And this is based on already existing predicted or reported activity or toxicity. So in principle, in our, let's say, databases, we have quite a lot of peptide which their experimental activity and toxicity is reported. And we train a model which is very decent, and in principle, this provides us a chance to predict. But in the end, as Professor Vesian mentioned, this is a prediction, right? We don't have any information about the cell, but we know that this is somehow encoded in protein sequences. That's the assumption of all this method. So this information is encoded in the protein sequences. And we try with AI to understand these patterns and to classify them based on already existing or reported experimental data.
Other questions? I mean, maybe I want to also, from the practical perspective, it's extremely important to have such good tools. Why? If you work also in the isolation of natural products or synthesis of compounds, you know that some compounds take 3, 4, 5 or even more person years to produce a few milligrams or sometimes only micrograms to isolate or synthesize, be it biotechnological synthesis or chemical traditional synthesis. And then for one reason you don't want to throw your few valuable micro or milligrams onto targets and assays which don't give you tell you anything for that compound, at least. On the other hand, as we've heard, if I say some people in assays, and I don't talk about standard antibiotic assays, that's easy, but if we have more specialized assays and you tell them, for instance, we did about neuroactive compounds, and they can make, like, I don't know, what was it, 10 assay points per month or something like that. You cannot tell them, I give you a 30,000 compound library, you know. That's impossible. So you gotta have tools to pre-select both targets and compounds or peptides to have more value for the money and the time put into these things. And so everything which helps there is important. And if I can reduce the wet lab effort to 5% and increase the success rate by a factor of 10, or sometimes we have even higher, then that is invaluable. It saves millions, it saves time. That's the important issue here.
So, great talk. So, congratulations for all the work and your team. So, I don't know if you could please further comment on how do you deal with the three-dimensional conformation of natural products or even peptides. So, in your experience, what's kind of the role of the 3D in the activity prediction?
Yeah, this is a very good question. I mean, in principle, actually, I didn't present here. As you know, there are different ways to predict. We have good experience actually here based on the target. I mean, if we talk about specific activity, and this is based on 3D structure, right? And we know that not all our, when we look at, for example, such a large virtual library, they are all, let's say, we cannot manually check them, yeah? But we try our best based on, at least because these are already taken from the lab, and they, for example, they, in principle, assign the configuration, or all the stereochemistry is correct, right? So, and we take this has very, very, I would say, a lot of impact on many, many targets. This is, I would say, still the classical approach, but we had cases that only from the structure, if the structure is okay, we assume that, as I said, this information is encoded. For example, we discussed actually, for example, flavor, we don't have any additional information from, for example, receptor, but we can predict only based on the structure of molecules. But any prediction comes with the, let's say, accuracy, right? So in general, I believe in the field of a structure-based design, we need to have a structure. We cannot completely use, let's say, method, rely on only sequence, and just like a smile, we need to use 3D structure. And that's why we combine this. For the first screening, we try to reduce the number based on much more high throughput method, but in the end, we try to look more closely to the specific one with the structure. I mean, for protein, of course, you know, we have a limitation. If there is a good crystal, we use it. If not, we try to use AlphaFold, but for the small molecule from our in-house, they are, I would say, more trustable, but when we take it from big PubChem or something, you know already, some of them, they might have not right configuration or stereochemistry or, but it's very important from our experience.
When we talk about, well, we will see, of course, a steep increase of the machine learning or AI tools. Of course, there is new dangers involved with that. Why in ChatGPT, if you ask about some star, a standard life, it's probably not it's not a big problem to have some biases and so on, which can be overcome by some tools. The bigger problem for me as a scientist is the hallucination, because that can cost not only lots of funding money, it can at the end maybe even cost lives in our business. So how can we avoid hallucinations and have only or train the program to say, I have no idea.
Yeah, I mean, this is a very good question. I mean, I'm happy we are not like designing AI for, I don't know, autonomous driving or something, if something go wrong, but there are some matrices we always check when we have a model, how reliable is the model in terms of also hallucination. We have done actually also started to work in this direction. There are a lot of development, but to move more or to try to have more interpretable and explainable model, which you can more or less look at how this is. But everything there is based on evaluation metrics. If they are good, we can somehow trace if there is a hallucination in the model or there are something wrong. But in the end, the underlying data determines how good is the model. So that's why I was very careful with the peptide. We had a very good experience. We did this for other systems, and it's very complex to get a very good model that I would say you really trust. I didn't show the result here, but we had very good, for example, in this case, because this is like something like 100,000 peptide.
Another problem is, of course, that the information which you use for the training comes from very different sources. Exactly. And every scientist who has read papers in his field knows after some time what are the more trustworthy, let's be outspoken there, and let's say less trustworthy colleagues and papers and groups and so on, or where you get more precise data and less precise data, which may still be very helpful and important, the less precise ones. The question is, we did that at OntoChem to put some confidentiality label onto these, but that's hard to do automatically, I guess.
Yeah, this is a very difficult question. I mean, in the end, I would say the problem is that we don't have like data from a lab, one technique, one assay, even in one lab might be different. Yeah, this is a big problem, but there are some, we try our best like to see if there is any imbalance in the data, in terms of the data, but in the worst case, this is something we cannot really control in terms of like where we get the data. For example, this peptide has been reported in so many different databases, but we try to find what are the outliers, what might be, sort of contamination, like what we have in also wet lab, but this is the part that we can put some uncertainty on it, and in the end we can report, okay, you have to more or less, treat them more cautiously when you want to design, For example, there's the work I showed. This is like, I mean, when you talk about a generative model, these are like the parameters that we can check. Okay, if this peptide has a very strange charge or any strange secondary structure or anything, we try to in principle evaluate after actually evaluating model how we can analyze already the output. This is, I would say, the only, let's say, last check-in that we can carefully check if a model first is a good model. This is a big question. As you mentioned, many parameters in terms of underlying data, and then of course if it's a model, how predictive works, and if you have a prediction, we have to check, is this prediction meaningful or is it something that does not make sense for an expert in terms of, for example, a compound which has a very strange structure or a peptide which has a very strange, I don't know, charge or something which is not observed normally in the nature.
Sometimes it's especially the strange things which makes the big discoveries and the outliers. Okay, I think I see no more questions. Maybe one important message, because we have the people, I mean, this is all streamed to the internet, but it's also because there are people in Latin America probably because of the time shift, we'll watch that at some other time. The important message is also working, of course, we do in chemoinformatics since a long time, that practical people often have a wrong expectation, and that is that the computer program gives you then the compound or structure. Let's say the structure is a virtual, the compound is a real world. So it gives you the structure which is then the solution. It does not. It's more like your Google search. Yeah, you have a list, and then let's say we hope that among the first 10 virtual hits there is a real good one. That is what we want to achieve, and don't believe that the number one is really the number one in real life. That's not the case. How often do you have that in your Google search? Yeah, that the number one is really the number one. So you have to go sometimes to page 2 or 3 of the results, or refine your question. Very often that's also mandatory.
Yeah, I think also can add, it's like in most of the field, for example, for peptide, if you do like to have a novel peptide, this only provide you a very good starting point. It's not the final peptide, because we know we have to modify it in the end for your purpose. As I mentioned, it can be very specific, it can be multi-target, but this provides a better starting point. And in some cases, maybe novel one, because maybe it's for IP important, this is the point.
And then you need the chemistry for the end capping or macrocyclization to avoid degradation, to have better bioavailability, and so on and so forth. So we will not run out of work, that's for sure. Yeah. Thank you very much. So, the final speaker of the morning session will be José Luis Medina Franco, and I'm really happy that he's here, because he also made a big effort, like Rebecca, to come here. He came not from Mexico, where he is a full professor at the UNAM. I will explain you in a second for those who don't know what it is here. He came, I think, where was it, from Boston or Arizona? Because he had a talk at a big conference in the US, and then flew directly over, told he manages 2 conferences in 3 days or something like that, or 2 days even, the same as Rebecca. So thank you very much for taking this effort, and we are very happy because we have started very recently the cooperation. Actually, what is different to most other speakers which I have known before is that we really came together as a consequence of the GLACIER project. Yeah, and that is, of course, very nice. And also we have found out that we have complementary setups in our system so that we can profit from each other. So in that sense, I'm very happy to have him here and want to shortly introduce him to you. So he studied at the National Autonomous University of Mexico, the UNAM. UNAM is a really huge university, and I would say together with USP Sao Paulo and University of Havana is one of the leading universities in Latin America, and the biggest one. I think UNAM has 100,000 students or so. What? 700,000. 700,000, unbelievable. So after his start at UNAM, he moved then to Arizona, I believe, as a postdoc, and then other postdoc or career step, postdoc assistant professorship in Florida, and with research at the famous Mayo clinics, which gives also a suggestion or a connection to the medicinal part. And then he returned as a professor to the UNAM. I think in now Latin America there is hardly a better place and he became I should say simply, he's a leading computational chemist in Latin America. So we are very, very happy to have you here and to have you as a cooperation partner, and we appreciate the effort you had to take to come here in time.
So, good morning, everyone. Thank you so much for the very kind introduction, Professor Lodger. Also, many thanks for the invitation to this event, and many thanks to all of you for listening. So, this morning I'd like to share with you some of the research work going on in my lab concerning the expansion and exploration of chemical space, in particular using food chemicals and more importantly, in natural products. So here on this slide, I'm showing the main library. This is in the main campus in Mexico City from UNAM. So this is the outline of the talk. Very briefly, at the beginning, I'm going to present some introductory concepts about chemical space, natural products and drug development. This is just for the sake of completeness. And also I'm going to move to molecular databases, emphasizing natural products, food chemicals, and a major emphasis is going to be on a multinational effort to put together a unified natural product database from Latin America. And at the end, I'm going to finish with some brief summary and an outlook. Chemical space in cheminformatics, it's a really key concept from the theoretical point of view. So on this particular paper by Professor Barnek, so he really defines the role of chemical space and the role in cheminformatics and in drug discovery in general. So he basically defines as a multidimensional space where we can locate the chemical compounds, and those compounds are going to be defined by a set of descriptors. There are several other also valid definitions of chemical space. A couple of them, well, three more of them are here on this slide. One very common one is just to make a resemblance, a chemical space with a chemical universe, where it says that the chemical space is all possible compounds that could exist. And Professor Madi already mentioned some specific estimations that the number of compounds is enormous. Now, the chemical space concept has several practical applications. One of them is to study structure property relationships, to conduct structure diversity analysis. I'm going to mention some specific applications on natural products, to design libraries and to select compounds from those libraries. So the chemical space can be analyzed using the full annotated multidimensional space, but also for most practical reasons, chemical space is analyzed through visualization in lower dimensions. That can be scatter plots or can be kind of network representations. So I don't know, I'm sure I don't need to convince the audience here of the significant roles of natural products in drug discovery. I took this picture from a very nice review paper of Maria Valli and Professor Bolzani, where they kind of highlights a few of several natural products that over many decades have made extremely key contributions in drug development. On this slide, I'm showing a few or so many chemical structures of natural products that are used in the clinic. As we can see from this slide, the chemical structures of some of them are quite complex, but in some cases the chemical structures can be relatively simple. Now, in Latin America and in many other regions in the planet, people rely a lot on traditional medicine, like teas, herbal preparations, and there, as you know, there are several research groups working on the isolation and characterization of the active ingredients, or the single chemical structures measure responsible for that bioactivity profile. So here in this slide, I'm showing just a very few examples of natural products from mostly plants from Latin American countries that have major contributions to drug discovery, either as drugs approved for clinical use or as a source of inspiration to produce bioactive compounds. So among the several types of chemical libraries used in the pharmaceutical industry, non-for-profit institutions like commercial libraries, de novo design libraries, on-demand libraries. Here I want to emphasize, of course, that natural products and food chemicals play a significant role as sources of bioactive molecules. Now, concerning food chemicals, quite recently, Juan Avellaneda, actually he's from Colombia, working his master's students at UNAM. He did a cheminformatic analysis of perhaps the largest public available food chemical database. It's FoodDB, with roughly 77,000 food chemicals. And the question in this study was to analyze the chemical diversity, the contents of the chemical scaffolds, and how these chemical structures are compared to approved drugs and natural products. So among the several different types of analysis reported on this publication, on this slide I'm showing a couple of visualizations of chemical space, comparing the chemical structures of compounds in FoodDB, here in red, with drugs approved from clinical use in orange. Here in kind of greenish color we have food chemicals that are also commercially available, and in yellow we have as a diverse set of natural products. So here at the top we are showing a visualization of chemical space using Disney based on max keys. At the bottom it's the same visualization by using a different structure representation, extended connectivity fingerprints. Just by looking at the two visualizations that jointly we can call them like chemical multiverse, we can see a very nice overlap between the chemical space of food chemicals with drugs approved for clinical use and also natural products. Secondly, we can also see that there are several food chemicals basically here at the surroundings. Perhaps here is more dramatic, that food chemicals of course are bioactive compounds that are really expanding the chemical space of approved drugs. Here in this slide I'm showing part of the scaffold analysis. For the sake of formality, these are the Venice and Murco chemical scaffolds, and here we're comparing the overlap as summarized here in numbers, like the frequency of the overlap between the chemical scaffolds in food chemical databases from FoodDB, approved drugs, those compounds were taken from drug bank, and natural products. The take-home message of this analysis is that we have again a large overlap between the bioactive scaffolds of food chemicals, approved drugs, and natural products. So I'm going to move to natural product databases, and here I'm going to emphasize the contributions from Latin America. But before talking about Latin America, I'm going to present perhaps the largest publicly available natural product database. It's Coconut. So the group of Professor Schneider just published a few weeks ago the newest version of Coconut. Is completely fully curated, completely fully constructed database that currently has about 621,000 natural products. And the database, the website is here, it's freely available, can be downloaded for virtual screening or for comparative analysis, that I'm gonna kind of focus on that part right now. In Latin America, perhaps one of the first countries that start making an effort to put together the chemical structures of natural products in a single big repository, it's Brazil, as far as I know. The database has been on the public domain now for so many years. It is called Nube. The latest version was published back in 2017, contains more than 2,000 compounds coming mostly from plants, but also we can find the rest of natural products, marine, and also from animals. And as far as I know, the group of Bolzani are going to publish a new release, perhaps later this year or early next year, with many more thousand curated natural product chemical structures. So about 5 years ago, we started a collaboration with Marila Valli, Adriano, and Professor Vanderland, analyzing the contents of the chemical structures, the chemical diversity, and the physicochemical profile of the natural products in Nube. So here I'm just, for the sake of discussion, here I'm just presenting the profile of 3 key characteristics like lead likeness, paints, and protein-protein interaction like profile of Nube as compared with other natural product databases. Now, Nube, as many other natural products from Latin America and in other regions in the world, are being using a lot to do virtual screenings to identify bioactive compounds. Just one example here, it's this paper where also a group from Brazil did a virtual screening of the entire Nube database and identify using experiments active compounds against Trypanosoma cruzi. Last year, the group of Miguel Angel Chavez published the first version of Peruvian natural products. The database is called Peru MPDB. It's also a public resource. Here is the URL of the web address where anyone can see the entire contents of this chemical database. And in collaboration with the group of Miguel Angel Chavez, we did a structured characterization of the compounds in the Peruvian natural product database. Here I'm just showing just one example, visualization of chemical space of the Peruvian natural products here in purple, as compared with natural products from Brazil, from Nube, natural products from Africa, the public database AfroDB, and here we're comparing with the chemical structures of Biohackim, our natural products from Mexico. I'm going to present this database in a minute. So the main conclusion from this visualization of chemical space is quite interesting, despite the fact we don't have so many compounds in this database. I mean, this is the first version, 280 compounds. They have a large chemical diversity. They are as diverse as natural products from Brazil. Of course, we expect that as this database is growing, the chemical diversity will likely be also growing a lot, and as other databases in Latin America, it's an important resource to identify bioactive compounds. So in Mexico, we have been putting together the Biohackim database. This is a database of natural products isolated and characterized in Mexico. So there are several groups working at UNAM and other universities working on this isolation and characterization of active compounds. So one of the latest developments on this regard is the implementation of a new web interface that is called BioMexDB. So this is a work of Fernando Martinez, a bachelor's student, that on his own he built this web interface. You can see a snapshot of how the website looks like. The user can freely search the contents of each individual compound. The current version has, by the way, roughly 600 compounds, and it's also possible to download the entire database for virtual screening of other type of analysis. Few other examples of Latin American natural product databases is Systematex. It's an effort of the group of Marcus Scotti. The latest version of Systematex was published in JCIM back in 2021, where they implemented as part of the website free predictors of biological activity. One of the most widely used for small molecules is PAS, but there are several other web resources to predict the activity profile of these compounds. Again, as was previously discussed, this activity profile should be experimentally confirmed, but at least it's a very quick way to have an idea of the potential activity of these compounds. Also, quite recently, and what I mean recently is a few weeks ago, a group of Argentina by Martín Lavecchia published the first public library of natural products from Argentina, Nature AR, and also a few weeks ago, the group of Héctor Fabio Cortés in Colombia published the first natural product database publicly available from Colombia, MPDB Ejecol, natural products, especially from the coffee region, the Eje Cafetero. So as you can see, several countries are putting together natural product databases. So here the idea was, why not joining efforts and putting together a single unified chemo library? Why not building a Latin American natural product database? So this idea kind of got started back in 2020. It was kind of just a thought concept, but over the years, actually, we have been working together, and the current version of the Latin American Natural Product Database, or LANAPDB, contains roughly 13,000 fully curated, standardized natural products, and the full details of this process of data curation and standardization is published here in this paper. So this is the current work team of LANAPDB. Dr. Marvin Núñez from El Salvador, William Zamora from Costa Rica, Dionisio Olmedo from Panama, Professor Vanderland, Adriano and Maria Valli in Brazil, the ones that are building in Nube, Miguel Ángel Chávez in Peru, and Héctor Cortés and Johnny Roberto Rodriguez in Colombia. From this team, I'd like to recognize a lot of work that he's putting into this project. Alejandro Gomez, a PhD student in my lab at UNAM. He's the one working in close collaboration with other research groups, and as I'm sure you know, perhaps the most time-consuming and sometimes boring work of building this database is curating the information, but it's crucial to have reliable data in this database. Of course, we're open to join efforts with other Latin American countries to add the natural product resources to this Latin American natural product database. So these are the specific databases that are so far assembling the LANAPDB. So actually there are already 3 databases from Brazil. These are the names of the other databases from other countries. Most of the natural products comes from plants, but there are also natural products from other sources. For instance, in Mexico, as I mentioned before, in Biofakim, we have natural products from fungi, propolis, and marine animals. So here in this slide, I'm showing the kind of summary of the contents of this database. I'm going to do so in the next following slides. Here on the left-hand side, we have the number of unique and overlapping compounds in the current Latin American natural product databases. So here I'm going to highlight just the number of unique molecules. So in Brazil, if we put together all compounds in Brazil, there are more than 10,000 unique chemical structures from that region. In Mexico, roughly 1,300 unique chemical structures, and in Costa Rica, 585 unique natural products. From the structure classification point of view, around 64% of the natural products are terpenoids, and around 35% can be classified as diterpenoids. So part of the property profile analysis is summarized here in this slide. Also, just for the sake of discussion, at the top I'm showing the distribution of 3 properties, LOHP, molecular weight, and topological surface area. Comparing on the top set of violin plots the profile of the entire LANAPDB with coconut and drugs approved for clinical use, just as a reference. So take-home message from this analysis is that Latin American natural products have a very similar property profile as compared to the entire coconut, and also, as we know, there is a large overlap between the property profile of natural products with drugs approved for clinical use. Also, and this is not surprising, we see that drugs approved for clinical use have some of them unique properties, because most of the drugs approved for clinical use come from synthesis or semi-synthesis. Now at the bottom I'm showing the distribution of the same properties. log P molecular weight and TPSA of 7 individual Latin American natural product databases as compared to approved drugs. And one of the highlights of these comparisons is that natural products from some countries, for instance, El Salvador and Panama, have unique physicochemical properties. So this slide shows a visualization of the chemical space using Tmaps and max keys, comparing the coverage, the diversity, structural diversity of 7 databases. And just by looking at these plots, we can really see that the diversity of natural products from Brazil, it's the highest. They also have a large coverage in chemical space, and this is kind of expected because it's the database with the largest number of compounds, and this diversity is followed by natural products from Mexico and natural products from Costa Rica. As I said before, the chemical space is also analysis, not just presented here just for the sake of time, but is also analyzing has been analyzed using the multidimensional space. Here is just showing approximate visualizations of chemical space. So this slide shows the 20 most abundant rings that they are called, like in the literature, magic rings, is bioactive rings. So a magic ring, it's based on the work of Peter Earle, where he did analyze the chemical structures of the entire chemical database and highlighted the most common rings frequently present in the measured therapeutic targets. So by just showing this overlap between the most bioactive rings in LANA-PDB, where we can see that the most abundant individual rings are directed to kinases, proteases, GPCRs, and many other therapeutic targets. And all this analysis comes from experimental data from Campbell. Concerning the molecular complexity, we know that several metrics have been proposed to try to approximate in a mathematical manner the structural complexity of the chemical structures. In this particular analysis, we used the normalized spatial score published last year by the group of Professor Baldman. Here we are comparing the chemical complexity of LANA-PDB, again with entire coconut and drugs approved for clinical use. And two major highlights on this distribution is that one, there is a large number of natural products from Latin America that have a large molecular complexity. So from a practical point of view, they have a large potential to be selective with therapeutic targets. And also very interestingly, overall, most of the Latin American natural products are more diverse than the entire coconut database. Also, this is the last slide showing this profiling of the databases. Here we are measuring the product likeness of Latin American compounds as compared to coconut and approved drugs. In this particular study, published earlier this year, we compared the natural product likeness using 3 different metrics. One really commonly used in cheminformatics is the natural product likeness calculator, again by Professor Peter Earle. The MP Scout by Johannes in the University of Vienna, and a new score is Neural Network Natural Products Score by Professor Oliver Koch here at the University of Münster. So comparing the distributions on the panel on the left-hand side, A, and panel C, is very interestingly because according to Peter Earle's score, Latin American natural products are more natural product likeness as compared to coconut, but we use a different metric, the neural network base score. Latin American natural products have roughly the same profile as coconut. So here in the middle, we're directly comparing Latin American natural products with approved drugs. Kind of not so surprisingly, most of the natural products from LANA PDB have a high profile of natural product likeness, but what I'm going to highlight here is that, as you can see, there is a significant proportion of approved drugs that are also drug-like. As we know, this is kind of a very nice way to see the high influence in natural products in approved drugs. So in conclusion, natural products and food chemicals expand the bioactive chemical space. The current version of LANA PDB is a compendium of 10 Latin American natural product database. This is the exact number of fully curated compounds. The entire database is open access, currently can be downloaded from GitHub for virtual screening or other type of analysis. Also, the entire database is available through the website of Coconut, so you just need to go through the Coconut webpage and look for collections, and there you will find the Latin American natural products. And each compound in LANA PDB, it's annotated with entire, of course, linear notations, reference taken from the peer review paper where the chemical structure was published. Also, each chemical compound has taxonomic and structure classification that I just presented briefly before. The physicochemical profile and chirality. If that chirality is annotated, that in most cases it is, the user will also find measures of molecular complexity and, also very important, synthetic feasibility. And finally, each compound is annotated with commercial availability. I mean, some user may want to select some compound for experimental screening. Several compounds, around 40%, are commercially available, so the user can go directly to the chemical vendor. And also each compound has experimental bioactivity data as reported in the literature. So work in progress. Now, this is a major effort, but the idea here is to put together a physical collection of the compounds, meaning not only the electronic version, but also the physical samples for experimental screening. And currently going on in our lab, in many other groups, the database is being used for virtual screening with different multiple endpoints. So, very nice lecture of Professor Mari also highlight the several applications of computational tools that can be used to fully exploit. Also, Professor Rebecca highlight a very nice review. So a couple of years ago, we did publish a review of several methods of artificial intelligence that can be used in natural products, and our group are using several of these resources now to use these chemical structures to identify bioactive compounds. So with that, I'd like to really thank members of the research group. That the name of the research group is DIFACIM. This is an acronym in Spanish that stands for Computer-Aided Drug Design at the School of Chemistry. As I mentioned before, many thanks, big recognition to Alejandro Gomez, putting together in collaboration with other groups in Latin America, the LANA PDB. Also many thanks to Ana Chavez, that last week she got the PhD. Fernando Martinez, the student that built the Biomex database. Now Fernando actually in a couple of weeks is moving to Milan to pursue a cheminformatics master's degree under the Erasmus Mundus scholarship. Juan Avellaneda, working heavily on food chemicals, and Daniel Marquez, that has been updating the Biofakim natural product database. Also with many thanks to the funding agencies in Mexico and other places to fund this work. So my final slide is an open invitation here to the community to join us at the second School of Cheminformatics in Latin America. It's going to be held virtually December 4 to 6. So this is a list of confirmed speakers, several speakers from Latin America, also from other regions of the world. So this school is co-organized with Karina Martinez, professor also at UNAM. So the registration is open. You can scan the code here or just go to the website. If you are interested in presenting a talk, you can freely do so. Just in the registration, suggest or give the name of the talk and the abstract. So actually, this School of Cheminformatics is very much inspired in the efforts of Professor Alexander Barnek, that has been holding several, over the years, cheminformatics schools in Strasbourg, and also the work of Professor Funatsu, that also for several years is holding cheminformatics schools in Japan. So the idea is together now bring these cheminformatics schools to bring also students in Latin America, but again, this is just not for Latin America, it's also open to everyone. So with that, I'm going to stop. Many thanks again for the invitation. I'm open for questions, comments that you may have. Thank you.
Yeah, I see already questions. Fabio.
Yeah, thank you, Professor Medina, for your nice talk. I have two questions. The first one is something I'm really missing in most of these databases, especially when it comes to plants, is a description of the environment where these plants are. And I think this information might be important in order to figure out what the natural compounds coming from these plants are. So, what do you think, what strategies can be used to overcome this problem? That would be my first question. And the second question is, especially in Latin America, many indigenous communities, they really have a lot of knowledge about the plants in their environment. Maybe some of the properties assigned to these plants might be a myth, but I also think that some of that knowledge actually is holding a hidden treasure there. So what do you think is the role that some of these databases that you presented and your database can play in order to preserve that knowledge that is in danger and fading away? Thank you.
Well, thank you so much for the questions, and the comments are great points. Regarding the first question, as far as I know, among these databases I just presented here, the current versions in Latin America, the one the Mexican one, the Biofakim, each compound is annotated with the source. I mean, like with the state and with the city the plant, let's say, was collected. But as you pointed out, talking with the developers of coconut, in most other natural products, that information is missing. So that's something that the community should also pay attention to kind of encourage the community to report in these databases the source. And the second point is also a wonderful point, that we should really take into consideration the practical use, because as you beautifully mentioned, many of these compounds are bioactive. So, but that information is missing in the databases. So just kind of emphasizing the point. So basically, it's also like the annotation to preserve that data. So I really want to thank you for that point, that we should really take into account, include that information into the profiling of the natural products.
Thank you, Professor, for this nice presentation. So my question was that I was looking at the numbers of natural products in the Peru database, and it's a very low count. My question is, what's the criteria to be considered a Latin American natural product? Are these products can be only synthesized from a species that are endemic from Latin America, or are products that can also be synthesized from a species around the world? Maybe the level of annotation and curation also impacts the count number of the natural products in the databases.
Okay, yeah. Also, many thanks for the comment. So, like the criteria to include the compounds in the databases is actually if that compound was published in a peer-reviewed paper by, let's say, a group from Peru or from Mexico. But of course, that's also a great point. It may happen that the same natural product may be present in several databases. And kind of part of that question is addressed on this analysis, where we're comparing the overlap of like how many compounds, let's say, are in Brazil, but also in Mexico and in Costa Rica. And we have working on kind of the same overlap with natural products in other regions of the world.
I'm a bit critical about such analysis because knowing how natural product isolation works and worked in the past, it was totally biased. So people went out and they had a connection to either a region or they had a connection to a certain type of taxa, let's say Euphorbiaceae. And then these people did a lot of research on Euphorbiaceae, and then you suddenly have a bunch of knowledge about Euphorbiaceae, which grow under certain conditions better than under others, and then you suddenly have this bias, especially if you have countries like, for instance, Panama, where you had not so many people doing research. So the difference which you see in Panama, for instance, from what you see in Brazil or in Mexico, in my opinion, I mean, no one can prove that now. Neither your case or I can prove my case, but I believe that a lot of these differences you see is the bias from the researchers in the past and nothing else. So it's not that Panama has more special plants than Costa Rica. I don't believe that, honestly. I think it's just this bias of the few researchers who done this, given these results, which you now use.
Yeah, well, totally agree with your point, and actually, yeah, but thanks also for pointing this out. So these databases are not really like a representative of the true biodiversity, are just efforts to collect in an electronic database the information that is in the literature. But as you pointed out, so we definitely know that the contents of this database is biased, because since the chemical structures come from literature data, so these databases rely on that what has been published. So yeah, for sure we know that there are types of natural products that are much more difficult to isolate and characterize, that are there but are not necessarily represented in the literature.
Yeah, for me, as being on both sides, it's more the practical question: where do I find the white spots with the interesting stuff? The other thing is also with respect to the bioactivities. Of course, the correlation is still very often is weak, because, and that is the fault of natural products chemists of the past. We have all these papers where you read the compound is antiviral, antibacterial, anti-something, and it's all bullshit, of course. I mean, because it's maybe just a tanning compound which then denaturizes the protein, and then it looks like it's an inhibitor, and we have lots of this stuff. And then we have compounds like half of the flavonoids at least, which have these antioxidant properties and general things, and there you can find these plants all over the world, so it doesn't matter really. On the other hand, you have other areas, like we work on the flavor. There a single methylene group changes the flavor totally, absolutely. So this is something which is not to grasp by these statistical methods. So yeah, we need the statistical methods to give us indications, but I'm not a friend of statistics with respect to developing drugs, because there we need the single compound, and it's buried under statistics. So the question for me is, how can we develop tools to overcome this problem and getting from the statistical and general and biased data to a specific information I can use to really develop a drug? That is a I know no one has a real perfect answer yet.
Right. Yeah, kind of part of the answer to that question is that there are metrics, of course, they are far from perfect, that try to capture this like promiscuous behavior of the chemical structure. For instance, here on this slide, I'm showing like the pains profile, or there are other metrics to measure what it is called the invalid metabolic panaceas. So as you're saying,
we should explain pains. Not everybody is from the field.
Okay, so pain are basically a reactive compound. It's a kind of a frequent hitter that in an experimental screening, high-temperature screening, it's almost always active because it's a reactive compound. It's not because necessarily bioactive. So, and the same as you're pointing out happens a lot with natural products. So for natural products, the technical name is invalid metabolic panacea. And there are kind of statistical methods to assign a score for a compound. So that kind of helps to rank order what compounds may be really promising as what compounds are really just generic reactive molecules.
One last question, if there is any. Yeah. It is guest professor from Cameroon.
Okay. Thank you, Professor, for the nice talk. I was glad at the end of your talk you're trying to move from electronic database from physical database. So I have a bit of experience in Botswana as we were trying to build what we call Pan-African Natural Product Library, and you should be prepared to face two problems. The first is people care most about the publication than the products. So after publishing, they don't care about where they store the products or so. So from the number of compounds that you have in the electronic, you should not expect, I don't know, maybe even half of them. The second problem is the purity of the compound. Like from plants, most of the compounds are not fully pure. Like when we have the structure from animal and whatever, we don't care about the purity. So you should really check the purity of the compound when you're trying to build this physical collection. Yeah, thank you.
Yeah. Well, thank you so much for the feedback. So yeah, we will take that into account. Thank you.
Very important. Purity and also availability of compounds is, of course, is a filter, a strong filter. Okay, with that, I want to thank you once again.
Thank you
for the talk and all the speakers of the morning session. Please give them a hand. And we will be back here in 1 hour for the immunology session and of course for the final grade. And I can only advise you to listen to the final talk, because Bert's talks for me are always a fantastic highlight of the conference. And yeah, Bert, we look forward to that. See you then.
please, may I copy the presentation of the first day, the pandemic intelligence?
We can have a look. This probably, yeah.
Oh, it is.
It's not recognized.
And the Colombian man, I don't remember, that was yesterday or the 4 days. That is.
I can't remember, and I don't feel well. Ja.
Störe ich dich gerade?
Nee, es geht sofort los.
Den Stick.
Die spannende Frage, wo wir das jetzt machen. Weil wenn wir das... Also die sollen nicht von Remote gezeigt werden, sondern...
Anna meinte, es ist besser, ich zeige sie hier, aber sie spricht... Also am Anfang spreche ich, aber dann wird die meiste Zeit Marike online sprechen. Und ich klicke vorwärts.
Das bereitet mir Kopfschmerzen. Weil...
Wir können es auch so machen, dass Marike das von dort spielt.
Das würde mir... Also das ist nicht... Der Aufbau hier, den habe ich ja nicht selber gemacht, sondern das habe ich von dem Universitätsmenschen bekommen. Wir tun das auf alle Fälle erstmal drauf.
Einmal drauf und dann überlegen wir, wie es... Also mir war das auch nicht ganz klar.
Kommt's nicht? Nicht da, kommt
vielleicht da. Dagmar.
Can you show me how it works?
In a moment, please.
Yes.
Yeah.
Vamos a ver si...
Cierre.
If it works.
So. Which one?
Also, die beiden, das einmal als PDF abgespeichert und einmal als PowerPoint. Macht mal die PDF ruhig. Ja, können Sie beide rübernehmen, genau. Also wenn ich es hier selber mache, würde ich es in der PDF machen, sind die nämlich nicht einzeln einlaufen. Wenn sie spricht, ist es besser, wenn die ganze Seite drauf ist. Deswegen habe ich die PDF gemacht. Okay, kriege ich erstmal. Das ist wunderbar, danke.
So, it is already prepared.
Yes. It's not
So, yeah, you can navigate.
It's like my normal mouse. Okay.
You can use the normal mouse. We can also move the.
I think the normal mouse is fine.
No, the normal mouse is fine.
As you like, yeah.
And the pointer is. If I need the pointer. That's the laser pointer. Okay, perfect.
Okay.
You can also draw, if you would like.
Okay. Wunderbar. So, jetzt ist bei uns die Frage, wie dir es lieber ist. Ich glaube, wir adjusten uns. Also, Ulrike kann auch die Screen sharen bei sich.
Ja, das würde ich fast bevorzugen. Also ich habe ein bisschen Hudeleien, vor allen Dingen mit dem Ton. Der kommt ja dann auf meinem Teams an. Und wenn wir dann nicht Vorkehrungen treffen, dann gibt es Rückkopplungen und dann kommt in YouTube überhaupt nichts mehr an. Also kommt so viel Echo, dass man quasi nicht versteht.
Wegen der YouTube-Sache.
Genau, und deswegen müssten wir, wenn das Teams spricht, muss ich an dem Pult da unten Mikrofone ausschalten.
Ach, dann habe ich die ja auch nicht.
Das ist einfach, das muss dann sozusagen, das ist totaler Mist, aber wie gesagt, ich habe das eben nicht selber aufgebaut, sondern aufgebaut gekriegt.
Spielen mit dem, was wir haben.
Und wir haben das halt rausgefunden, dass wenn wir die dann stumm schalten, dass es dann funktioniert. Und das ist doch auch der letzte nach dem Coffee Break nochmal. Also wir hätten auch noch ein paar...
Ich habe gesagt, um 3:30 Uhr habe ich Sie eingeladen, dass wir das vielleicht probieren und schon mal einen Testlauf machen.
Ja.
Jetzt hoffe ich, dass Ihr Link schon um 3:30 Uhr funktioniert.
Der funktioniert die ganzen Tage.
Okay.
Ich bin sogar schon drin. Ja? Also wir sehen jetzt schon hier, was über die Kamera übertragen wird. Und wenn Ihre Kollegin das freigibt, ich brauche nur den Stöcker reinzustecken und dann sehen wir es auch auf der Leinwand.
Okay.
Das geht so raus.
Also, ich kann Sie mal anrufen und dann... Dann können wir das zu dritt auch mal kurz noch besprechen.
Wir hatten am Montag diese Panel-Diskussion und es hat zum Ende hin leidlich funktioniert.
Hi Marike. Hi. Hello. Can you hear us? Okay. I don't know if this works too well. Okay. So I'm now here with the IT guy, and he suggests that you do the presentation from your MS Teams account by screen sharing. That's right. Yes. And what we normally would have done, and the information I had before then is not that valid because they get interferences with the microphones for the online presentation. That would work for you, right? You could share.
So there's always this risk of mistakes, but I try, and then if there is a problem, then you should have a backup space.
No. I have it on the computer here, and if all else fails, I sing it from the PowerPoint here, and that would always work. But normally MS Teams is not ideal for us, but it should work, and we should be able to share the screen. We'll try at 3:30. Okay? And then we'll see how it goes. And then I probably don't have really a microphone on here, so we have to see how our idea that I do the first slide, and then we try to make it happen that I introduce us in a way the project and then hand it over to you, if not to do it all. But we will only use my two showing slides by sharing the screen, right?
Yes, you should just make your presentation and just the switch between Bert Hoffmann and you requires me. I would like to then mute or unmute the local microphones because our local setup for audio is a little bit unlucky, and because we stream the entire conference, and if the Teams is doing audio, then we get some mix-up, and the YouTube audio quality will not be well, and therefore I would like to close our local microphones when you start talking.
Okay, so when I hand it over to you, the technician jumps in and you make a 2-second break or whatever you need, and then you continue. Okay, so we should try it.
You should use either Chrome, or if you are using Windows, you should probably use Edge, and that works perfectly. There is no longer dedicated Teams app, it's just browser only nowadays. They totally cancelled. Firefox does not work as well, but Chrome and Edge should, and maybe Safari work.
Wonderful, thank you.
And the team session is already open. You can, in principle, you can already join the conference, and as soon as just to check out the link works, you could do it.
Okay, perfect. Yeah. So that was fun. Thank you, Frank.
Yeah.
Thank you, Marike.
Welcome.
And we have high expectations.
13:40. Ja, so rein prinzipiell.
seat. I guess we will start, otherwise we get too delayed. And actually, we are switching now from natural compounds, from chemistry and artificial intelligence to immunology, and I'm very happy to share this session. First of all, I have also to an announcement. Markus Meurer from the Champollion Foundation, he would not come, so that we have one talk less, and then after the coffee break we have then also one very important part of GLACIER is more the social political stuff. Okay, and for me it's a big pleasure in particular also to introduce Dagmar Garcia Rivera from Havana. Dagmar is current Vice Director and Senior Research of the Finlay Vaccine Institute in Havana and Cuba, and she was actually the one who developed with the chemistry all the wonderful COVID vaccines and also others which we will hear. Dagmar studied actually pharmacy at the Faculty of Pharmacy in Havana, and did also there the PhD thesis. Then during her PhD thesis, she moved a little bit around to Spain, but you always kept more or less in the Spanish side where you could use your Spanish. She went for a couple of months, I guess, to the University of Santiago de Compostela and spent also some time at the University of Valencia in Spain. And furthermore, as a postdoctoral scientist, she moved to the Ghent University in Belgium, and in between also, I guess, for a couple of months at the WHO Reference Laboratory at the London University College. She has published many great papers which were really recognized, and this was also that she received various prizes, in particular of the Cuban Immunology Society, prizes of the Hearts, as well as of the Cuban Academy of Sciences. And for me, Dagmar, it's a really great pleasure that you came here and give the first talk in the session. Thank you very much.
Thank you, Barbara, for this nice introduction. It's a pleasure for me to be here for sharing with you our experience from the Cuban industry for research, development, and production of vaccine. Now I'm talking about the development of pneumococcal conjugate vaccine, the Cuban experience. We will be talking about the pneumococcal conjugate vaccine as best alternative for preventing pneumococcal disease in children. Also, our experience in the clinical development of Quimibio, which is an heptavalent pneumococcal conjugate vaccines, and also what we call the race for the 11 valent serotypes. Well, 2 days ago we were talking about pneumococci. Streptococcus pneumonia is a gram-positive bacteria, and this is encapsulated bacteria. That means that this is surrounded by the capsular polysaccharide, which determine at least 100 different serotypes of this bacteria. We, the human beings, are the unique reservoir for the nasopharyngeal colonization of this bacteria, and this important cause of disease, severe disease, and mucosal disease in children and older adults. Also, there are another important challenge with this when you work on these fields, that is the antibiotic resistance of this bacteria, and this is become more relevant, the approach of prophylactic vaccine. The world now is more than 30 years ago, is very well known that if you want to be more effective for preventing pneumococcal disease, you should use the capsular polysaccharide as antigen, but you should conjugate this capsular polysaccharide to a carrier protein. The immunology explanation about this phenomenon is because when you use only the capsular polysaccharide as antigen for a vaccine, you only could be able to stimulate an important antibody response, but you will not able to induce a memory immune response, and there is not affinity maturation, and these antigens are not efficient in young infants. However, if you conjugate the capsular polysaccharide to a carrier protein, this carrier protein will or will present to the immune system the T epitopes for the cooperation of T cells, and then you will be able to organize a better immune response in order to induce antibody response of long duration because of immunity memory. The antibodies could be able to modulate the affinity, and also this kind of vaccine are very effective in young children. This is the history of the pneumococcal conjugate vaccine. This vaccine started in 2000 with the only heptavalent conjugate vaccine from White. This is a company from the United States, and after the success of this vaccine, this company was acquired by Pfizer. Also, GLACIER developed a 10-valent vaccine. Pfizer developed a 13-valent vaccines. But these vaccines were monopoly for the big pharma during more than 15 years. That means that they have a very high price for a country like Cuba. And just 2 or 3 years ago, the Serum Institute from India licensed a 10-valent vaccines, and really this is the vaccine that has been promote the decrease of the price of this vaccine and have been more available for the rest of the developed country in the world. These are very effective vaccines for reducing disease and death caused by this bacteria, but also it's very important that these vaccines are able to induce herd effect. That means that you could protect those population, older population that you didn't vaccinate, because the vaccine reduced the colonization and also cut the transmission of the bacteria, and then you could protect it in indirect way the population that you didn't vaccinate. Of course, WHO recommends the use of this kind of vaccine in all routinary immunization programs around the world. But at the end of 2021, the WHO recognized that only 51% of the child who born every year in the world receive the complete schedule of this vaccination. This is considered as unmet global need, and for this reason, we decided some years ago to start the race for develop our own pneumococcal conjugate vaccine. We start around 2008, 2006. This is the most complex vaccine that we have developed in Cuba, even when we have huge experience for more than 30 years developing our own vaccines. The first challenge was to decide which serotypes we will include on the vaccine, taking into account that there are more than 100 serotypes of pneumococci. So you should include more than one. You should design a multivalent vaccine, and at that moment, we decide by the 7 more relevant serotypes that cause pneumococcal disease in Cuba and Latin American countries. Our composition, the composition of the vaccine is this that is shown in the slide, but also we focus on trying to cross-protect using a special conjugation method. I'm not going to talk about the conjugation method of the vaccine. I will most focus on the clinical development, but we include 7 serotypes of the vaccine, but with the aim of inducing protection against 9 serotypes, because 6B could induce protection versus 6A, and 19F could induce protection versus 19F to 19A, and I will show you the main results. Okay, the timeline starts with the research stage. We were focused on that stage around 2012, and at that moment, we were so far away from the final vaccine, because at that moment is when really you start to develop a vaccine, when you finish the research stage. So we were working on development stage. Later, the clinical evaluation and epidemiological study, we were focused for more than 10 years. This is the main presentation of today. And later, we should scale up and transfer technology. We should accomplish with all regulatory requirements from the national regulatory agency, following all the WHO recommendation. And finally, this year, we licensed this vaccine, and this is now introduced in our national immunization program. Okay, we designed a clinical strategy of research and impact evaluation for this vaccine that was supported by 4 main lines. The first were the epidemiological studies, the second, the clinical research of the clinical trials, also economical evaluation of cost and cost effectiveness of the vaccines, and the last one was the effectiveness and health impact of the introduction of the vaccine in the National Immunization Program. We published this strategy some years ago, and the first things that we did was to conform a sentinel network as platform for PCV development. In pneumococci field, it's very important the epidemiological surveillance of the bacteria, because all the time could appear new serotypes, the incidence of the serotype could change, and also when you introduce a vaccine, against some serotypes, these serotypes will go down, but for sure there will be other serotypes that will increase the incidence. For this reason, we create this platform in accordance with the National Health System. This platform is conformed by 6 pediatric hospitals of the island, because the pneumococci surveillance is very expensive. It's not possible to do it as universal surveillance. We need to do as a sentinel surveillance. And this has been important for describe the baseline of pneumococcal disease before the vaccine introduction, and later the impact after the vaccination. These are some of the results that we collect from this surveillance. For example, during 2019 to 23, there were more than almost 200 isolating of the bacteria in children, and you can see that there were 22 different serotypes isolated, but the 79% of the isolated strain were vaccine serotypes or vaccine-related serotypes that are included in the vaccine. That means that our vaccine could be effective around 79% of the pneumococcal strain that we have circulating in Cuba. This is another important study that we conducted in infants around 2 to 18 months old. It was a cross-sectional study where we studied the colonization rate in the nasopharyngeal of the infants, and you can see that more than 20% of the infants since 2 months old are already colonized by pneumococci, and also the 60% of the pneumococcal strain that we isolated from the nasopharyngeal of the infant were related with the vaccine that we were developing. Of course, we published these results. Also, we did another study of prevalence of pneumococcal nasopharyngeal caries in daycare attendance children, because in Cuba, normally the first year of the life, the baby is at home with his or her mother, but when the children become 1 year old, move to a daycare attendance centers, so it's normal that in this daycare center, the prevalence of pneumococcal caries will be higher. And of course, we show that in these children, the prevalence of positive for pneumococcal caries was around 32%, and around 73 of the isolated pneumococcal were related with the vaccines that we were developing. The second line of our clinical strategy were the clinical trials. The clinical trials for the case of pneumococcal disease is very well defined by WHO what you should do. You should demonstrate the safety of the vaccine in adult population before children, of course. You should demonstrate the safety of the vaccine in children and also in infants. You should conduct a non-inferiority immunogenicity study compared with the gold standard of pneumococcal vaccine on the market, that is the Prevnar 13 vaccine from Pfizer. Also, you should demonstrate that there is not interference of the new vaccine with the routinary vaccine that is used in the National Immunization Program, and also we should demonstrate efficacy of pneumococcal disease and carriage. These are the endpoints that immunological endpoint that any new vaccine should accomplish for be licensed in terms of induce immune response specific for each serotypes. That means that the immunological evaluation is by 7 because it's a heptavalent vaccine. We developed in our institution an ELISA assay following the WHO recommendation, but we did a bridging study for the validation of this assay with the International Reference Lab in London under the supervisor of Professor David Goldblatt. And then we decide to start the clinical evaluation in 2 different age groups, children between 1 to 5 years old and also infants below 1 year old. This is the timeline of our clinical development. We started in 2012 and we finished last year the clinical development. And of course, we have conducted phase 1, phase 2, and phase 3 clinical trial. And now I will summarize the most important results of that. At the end, we have vaccinated more than 36,000 children during the clinical development of this vaccine in Cuba, and this is very important because these children are our children, the children of the Cuban people that are that trust in the Cuban science, and they are available for conduct clinical trial with them. This is the non-inferiority comparison between our vaccine with Prevnar 13 for the 7 common serotypes. You can see in red or in pink, maybe the result of our vaccine compared with Prevnar in blue, and the first pre and post results, and you can see how the immune response in terms of antibodies IgG is similar for both vaccines for the 7 common serotypes. And also in the table below, you can see that there is an important response against those 2 serotypes that are not included in the vaccine, but we were focused on cross-protection because the similarity of the molecular structure of the capsule polysaccharide for the similar serotypes. With this clinical trial, it was a phase 2, phase 3, we got the criteria for the licensing of the vaccine in preschool children. These are new results from GLACIER in this year because we have a PhD student here in the Professor Selinger labs. This was a clinical trial not with healthy children. It was a requirement of our national regulatory agency that we should demonstrate that the vaccine works on children with comorbidities or chronic disease of the infancy. So we conduct this clinical trial in more than 7,000 children that suffering from bronchial asthma, diabetes, immunodeficiency, kidney disease, and so on. And you can see here pre-post the immune response in terms of IgE against all the serotypes included in the vaccine, also IgG1, and also IgG2, that's means the vaccine works even in those children who suffering from other chronic disease. Now we move to infant. Infant means baby of 2 months of age, because this is the final target for this kind of vaccine. We start this clinical trial, it was a phase 1, phase 2, in 2019, just the year before of the pandemic, with 3 different schedules of vaccination, concomitant or not with other vaccine of the national immunization program in Cuba, and we could finish the phase 1, but when we started the phase 2 with 5 children already included, we should stop this clinical trial. But the results were very good. These are the results in terms of safety. This vaccine is very safe in children, also in infant. The profiles of safety is similar to control vaccine. The adverse event that we found were what you expect for this kind of vaccine, local adverse event, some fever, but any serious adverse event related with the vaccine. And when we saw the immunological response, this bar represents the percent of children who accomplished with the correlate of protection established for WHO. You can see in the upper side that the schedule called 2+1, that means that you immunize 2 doses at 2 and 4, and the first dose at the year of life of the children, is better than when you immunize the 3 doses 2, 4, and 6 months of life of the baby. That means that we will move forward with this schedule, and also the better or the best difference is just in the case of these 2 serotypes that are not included in the vaccine. You can see for these 2 serotypes that are not in the vaccine, when you use the schedule 2+1, you have almost 100% of protected children. The same with the commercial vaccine that has these 2 serotypes included, but for these 2 serotypes, when you use the scale 3+0, the response is worse. And this is another important result of this clinical trial, because when you introduce a new vaccine in the national immunization program, you should demonstrate that there is not interference with the antigen that are contained in the other vaccine. So we should demonstrate for the full sample of the clinical trial that the immune response versus tetanus, diphtheria toxoid, Haemophilus influenzae type B, hepatitis B, pertussis, meningococcal V, and tetanus toxoid is the immune response that you expected even when the vaccines are not concomitant. We published most of these results some years ago. I'm not talking about cost effectiveness study. Of course, we perform this kind of a study, and of course, these studies say that this cost effective the introduction of the vaccine in the national immunization program. And the 4 line of our clinical strategy were the effectiveness and health impact evaluation study. Cienfuegos is a central province of the island, and during 2017 and 2018, we conduct a community interventional study in which we vaccinated more than 70,000 children that represent more than 95% of the children of this province. This was the first huge clinical trial where we immunized the vaccine in the real condition of the program. It was a controlled clinical trial in a special site. These children were vaccinated where they live, in the mountain, in the coast, in the city. And the vaccine was very safe, and we demonstrate as early impact only 1 year after, because later, unfortunately, appeared the pandemic, and we cannot evaluate the effect of the vaccine longer. But we demonstrate 1 year after this vaccination a reduction in 62.9% of the hospitalization burden by invasive pneumococcal disease, and 72% for IPD and 84% of meningitis, which for the serotypes include in the vaccine. And this is, for me, this is the most important slide of my presentation, because you can see this is the circulation of pneumococci in Cienfuegos before vaccination. You can see in red are the vaccine serotypes. In blue, the relayed vaccine serotype, that are these two serotypes additional by cross-protection, and in green, the non-vaccine serotypes. These are the previous year of baseline of the circulation of the bacteria. In 2018, we applied the vaccine and we got more than 95% of coverage. And 1 year after, completely disappeared the vaccine serotypes and the vaccines-related serotypes, and there are still the persistence of non-vaccine serotypes. Unfortunately, because of the pandemic, we cannot measure this effect as long as we want, but this early effect is similar to, has been reported for other pneumococcal vaccines. So now we are starting the race for 11 vaccine serotypes, because this is the 3 vaccines that are recommended by the WHO. And we now are developing 11 serotype vaccine, which we are including 4 more serotypes. This is a very stressful race because this is the map of the manufacture of pneumococcal conjugate vaccine around the world. 10 years before, there is nothing here. And now you can see how many manufacturers of this kind of vaccine appear in the Asia region. 10 years ago, it was just Pfizer, GLACIER, Merck. We, of course, Finlay Institute were in the map 10 years ago, but this is new, and this is an important competitor for our capacity in the world of pneumococcal vaccine. So where we are with PCV11, we have finished the preclinical evidence of safety and immunogenicity in rabbits. We have demonstrated no interference with other vaccines, and also we have demonstrated an immune response in rabbit is similar to Prevnar 13. We have manufactured the first batch for clinical trial, and we have applied for a phase 2, phase 3 clinical trial. We are waiting the authorization of our national immunization program for start this clinical trial. So at the end, always everybody said thanks. For us, our major thanks is for the parents and for the kids that are able to be involved in this huge clinical trial, because they trust in the science that have done in Cuba. And also, during this year, we have been involving several medical doctors, nurses that now are part of the project, and without them, it would be impossible to have the results that we have presenting today. Thank you very much.
Thank you very much, Dagmar. It was a great talk, and I really admire what you are doing in Cuba. Okay, are there questions? Yeah, please. Yep.
Two questions. One is related to the potential risk of replacement of the vaccine serotype by the non-vaccine serotype. Are you expecting something occurring in this regard? How you can confront it? Is easy to adapt the vaccine formulation? And the second question is, here you were talking about the pediatric, the infant population. Are you also planning to implement this vaccine in elderly?
Yes, the replacement of the serotypes that we are expecting is the same that the world has been suffering during more than 20 years of these vaccines. For this reason, we decided to add 4 more serotypes, because are the serotypes that are more relevant now in the circulation in Cuba, but also are resistant of antibiotics. We know that these are relevant. In the world that this vaccine has been using for almost 20 years, the variety of serotypes that are now circulating is very high. That means that you can never have a vaccine against all the serotypes that you detect on the circulation. But the main element for selecting are the incidence, and also the antibiotic resistance, and are the prevalence for causing pneumococcal invasive disease, because some of these serotypes you could detect on the circulation but are not invasive serotypes. Also, in the case of adult vaccines, this PCV11 we will also evaluate in adults, both in infant and adults, and we know that we should move to 15, 20 serotypes. And we will decide, it's not clear right now, if we will develop 2 different vaccines, like Merck strategy that has a vaccine for infants and a vaccine for adults, or the Pfizer strategy that has only 1 vaccine with 20 serotypes, both for infant and for adult. But of course, when you add more serotypes, it's more complex in terms of analytical, in terms of technology, and also the vaccine is more expensive. And we should take into account all these elements for the decision of the final vaccine. But for sure, more than 11, we should go for more than 11.
Dagmar, as you know, I'm not an immunologist, but when I hear 100 serotypes, that's an immense number. And I ask myself, if all these vaccines target, let's say, the 10 or 13 same serotypes which are prevalent, don't you select then for the other serotypes to come up in some future waves, so that then you have to make new vaccines? And from my perspective, and even more serotypes will probably emerge at some point. And then the question comes, is there not some domains in antigen domains which are not so variable, which one could use for developing a vaccine which is broader and not so dependent on the variations in the glycopart?
Good question. Yes, the problem is that, the first problem is that the capsular polysaccharide is the major pathogenic structure in this bacteria. And each serotype changes, sometimes the chain is a bridge between 2 atoms of the, it's something like nothing, but this is very relevant for the immune system. So there have been several approaches for looking for a universal vaccine against pneumococcal based on the protein of the bacteria. GLACIER have developed during more than 15 years vaccine based on recombinant protein that is clear which are the better conserved protein of the bacteria, but all this strategy has failed until now. The only effective strategy on pneumococcal vaccine is the conjugate vaccines, and this is a huge challenge because of the change of the serotype. And when you saw the data of London, for example, of UK, that has a very good surveillance data, after PCV20, for example, that they are applying now, they could have 40, 30 new serotypes on the circulation, but actually with very low incidence of disease, not with huge incidence of the disease. I think that we have a need of pneumococcal vaccine for the next century.
Okay, there's the last question over there, the back.
Thank you for your great presentation. I just wanted to ask that how did you make sure that your vaccination will be impressive or reliable on the infants? As you might know, infants are the most vulnerable population of people, and if I'm not mistaken, we saw that the effectiveness was just 60%, I think. And were there any side effects on the infants, or in overall, how did you make sure that this vaccine will work or not? Thanks.
Yes, of course, in terms of safety, these vaccines are very safe. We have vaccinated more than 38,000 children, and now the vaccine is introducing in the National Immunization Program in Cuba. It's a platform of conjugate vaccine that have more than 40 years of use in pediatric population. That means that we don't have any concern about the safety of the vaccine, and the data show that. About effectiveness, of course, if we have only a 7-valent vaccine, we cannot expect a 90% of effectiveness, because there are a lot of serotypes circulating and causing disease. But WHO recognized that, not now that for Pfizer, for example, it's too easy to have, well, it's not too easy, but they have a 20-valent vaccine. But from the beginning, Pfizer also started with 7 vaccine, because with 7 serotypes. Because WHO said if you should have a composition that accounting for at least 70% of the serotypes that you have on the circulation. And then we accomplished with that, and we are expecting now when the vaccine will be introduced in whole country, we are expecting to be able to show this kind of slide on where you see that how the vaccine serotype completely disappear on the circulation and some other serotypes start to circulating.
Okay, if there are no other questions, I would really like to thank Dagmar again for the great work, and I'm looking also forward to our further collaboration. Now, actually, I'm very happy to introduce the next speaker, and I'm very happy, actually, I was hoping that you come till the last minute, and you did come. Alles flying, and actually, Mikhail told me yesterday evening that was the last 2 nights, these were the last 2 nights where she could really deeply sleep. Okay, everybody knows the situation in Israel. Mikhail is a good friend and the director currently of the National Center of Influenza and Respiratory Viruses at the Central Virus Laboratory of the Ministry of Health, and she's also associated as an associate professor at the Tel Aviv University. She did her bachelor and master degree in life sciences at the Bar Ilan University. And then she moved actually as a researcher to the Veterinary Institute, and then together with her husband, who's a great tumor immunologist, over to Harvard to Jack Strominger, and I think everybody knows Jack Strominger. She came then back and started her PhD, and she worked actually on RNA processing with trypanosoma, and since 2005, you are at the National Institute of Viruses, and thank you very much, and we are everybody looking forward for your talk.
So, thank you very much, Barbara, to organize this meeting and for inviting me, and I will show you what I'm doing in my lab. So I think that you feel my work, so I will start. So I will talk about the dominancy among influenza and SARS-CoV-2 viruses. Where is the from here? So, start introduction about the SARS-CoV-2. I think that all of you know already. So, it's acute respiratory syndrome coronavirus 2. It's called the coronavirus disease from 2019. The family is Coronaviridae. It's positive single-strain RNA virus with single linear of RNA segment. And the envelope contains a transmembrane, the spike protein. So as you know, there's a lot of emergence of variants of concern that appeared during the time. It started with the wild type, and then we got the alpha, beta, gamma, beta, and the lambda and the omicron. And like you know, 2 of the VOC you can see from the South Africa, and so maybe it's because of the high number of immune suppressed HIV infection individual in these countries. So we were the first lab that showed that patient after they recovered from the wild type, you can see it here. So 6 months after that, the amount of the antibodies it was really, really low. And we said that they should take the vaccine, and this what happened. You can see the amount of the antibodies now is much better. But when we checked the amount of the antibodies of this patient, these people that recovered from the SARS-CoV-2 against the alpha variant, it looks almost the same because the alpha is really close to the wild type, but let's see how it looks like against the beta. The amount of the antibodies is really, really low, even after they were infected with the virus. You can see it's almost zero, and it's much better after they got the vaccine. And the gamma is something between the beta and the alpha. And we were the first lab that showed what happened when the Omicron appeared in the world. You can see here that we saw that after the second dose, this what most of the world had. The amount of the antibodies, even again the wild type, is really, really low, and of course that again the other variant. But let's see what happened after the third dose. You can see the amount of the antibodies against the wild type, it looks much better, but the Omicron is worse even than the beta. And we know that the beta already is not good, and we were the first that showed to the world that the vaccine is not good enough against the Omicron variant that start to be the dominant strain in the world. So this is the Omicron from November 2021. The Omicron variant started to be the dominant strain around the world, and we started to see a lot of new branches from the Omicron, the BA1, BA2, BA3, BA4, and BA5. And you can see, and even this variant, we start to have more and more new branches from this variant. So, as a new variant appeared, so the vaccine also changed accordingly. So this is, for example, the Pfizer vaccine that we gave in Israel. So the first vaccine was in December 2020, and then the second one and the third one, and one more booster is the fourth one, and then they said that we need to change the vaccine, so they changed it to the fifth one, that it's a bivalent of wild type and BA4, BA5 together, and then they understand that it's not good enough, and they change it again to the sixth vaccine, that it's just the XBB1.5 variant. So this is an example from a work that we did about a patient that are heart transplanted patient that got all the 6 vaccines, so we know it because we know this patient. And you can see here that the amount of the antibodies against the wild type is nice because they got 6 vaccine already, and the BA5 is good enough because they got the 5th vaccine, and this is the 6th vaccine that they got really nice antibodies. But let's see how it looks like after against new variant that we have, that they didn't got the vaccine, the JN1, and you see the amount of the antibodies is low. You can see it here that is significantly lower from the other variants. So it means that even if you got 6 vaccine, maybe it's not enough for you. And if we will split this group for 2, one that infected with any SARS virus and then vaccinated, or uninfected, so you can see that the uninfected, the amount of the antibody is really low. So we know that maybe it will not be enough for them to protect this patient, that it's really important to protect them against the new variants. We next check the monoclonal antibodies. It's called EvoShield, that they give it to a patient with CLL, that it's a immunocompromised patient. And so you can see here the antibodies here before they got the EvoShield, that it's really low. And let's see how it looks like after the EvoShield. You can see against the wild type and the Delta, it's looking good. But against the Omicron, it didn't help them, so in the same moment they stopped to give them these antibodies because we see that it's not do nothing against the Omicron. Here we checked it in culture, so you can see the EvoShield again, the wild type, alpha, delta, and a few type of Omicron. So you can see that the amount of the antibodies that can protect the cell from the virus against the wild type is better, like we saw before, and against the Omicron is really low. And this is again to show us that these antibodies that we gave to a patient in our Shiva Medical Center where I'm working, it's not work against not help them against the new variants. So this is the variant in Israel from July 23 to July 24. It's a really nice picture, if we can put it in a lot of colors, but each color it means that it's more and more variant, new variant that you can see. You can see all the color all the time, a new variant. So what can we do? The vaccine all the time, we can change the vaccine all the time. You can see XBB and KP, and I think that some of you know already the new variants. So we conclude that the vaccine against these variants are not efficient against the new variant. So what can we do? So most of you know this is the ACE2 receptor. This is the receptor that the SARS-CoV-2 can enter the cell when the spike binds it. So you can see here this area. This is the binding interface. So this is the RBD, the receptor binding domain of the virus, and most of the vaccine that we have, it was against this area, against the RBD of the spike protein, but like we saw, the problem that the RBD change all the time because we have new and new variants. So we thought maybe we will try to find antibodies that can bind to the ACE2 in this area, in the binding interference, and then it can help us against all type of SARS that we will have, all the variants, because this is the same area. So what we did, we cloned the extracellular portion of ACE2 frame with Ig. It looks like this. This is the ACE2, and with Ig1. And then produce it and provide it, the protein. And we did also the same we did the same construct for RBD with the IgG. So to test whether this protein is functional, so what we did, we transfected cell with the spike, and then we checked if the ACE2 Ig can bind to the cell. This is the ACE2 that we prepared. So what can we see here? We can see really nice binding of this of the ACE2 Ig to the spike protein on the cell. So we can see that our construct can bind to the spike, and we did the same with the other construct. We transfect the ACE2 to the 293 T cells, and then checked if the RBD can bind it, and we see really nice binding of the RBD, so we can see that both of the protein functional. Then we immunize mice with the ICIG, and then we collect the monoclonal antibody on all the work, and then we screen 100 of antibodies to check which antibody can bind to the ACE2. So you can see here a lot of antibodies, not a lot, few, but one of them, the 16, it's look much better. You can see it here, and this is the antibody that we choose to keep work with it. So we know that it's bind, but now we want to know if it can block the binding between the ACE2 and the RBD. So what we did, we extract the cell with the ACE2, and then we incubate it with our antibody and with the RBD, and we checked if we can block this binding, and we can see really nice that our antibody is a dose-dependent manner, and we can block the binding. Next, you know that the ACE2, the virus used this receptor, but this is not the reason of this receptor. The receptor is really important for keep the part of the activities to keep the blood pressure of check. So next we checked, we use a commercial kit to check the ACE2 activity, and we can see that our antibody that we check, including the number 16, is not effect on the ACE2 activity. So now we know that it's binding, that it's not effect, but we want to know if it can block virus infection, well, this is the reason. So this is the callus cell. We can see that our antibody can bind to the callus cell. And now we can check if it can block, and we can see that it's really nice block infection of all these variants that we check, wild type alpha, beta, gamma, delta, and Omicron BA2 and BA1 and BA2. And we can see that it can inhibit the more variants, XBB, XBB1, BQ1, and etc. So now we said, okay, maybe we'll have really different variant, that the spike will be, the RBD will be really different, and maybe it will not block it. So let's check the NL63 coronavirus. It's human coronavirus. I will just say that it's a prevalent human respiratory virus. It's caused upper and lower respiratory tract infection mainly in children. And while the NL63 is alpha coronavirus, the SARS-CoV-2 1 and 2, they are beta. It's really different, and they don't have any homology in the RBD, but we know that both of them bind to the ACE2 receptor. So what we did, we check if yeah, fine, did it. Sorry, thank you. So we could see that if we will see the cytopathic effect of the NL63 on the cell, you can see that destroy all the cell. But if you can see that we put the antibodies here, it's protect from the protect on the cell from the virus, and you can see that it does depend manner inhibit the even different virus of corona, the NL63. So we develop here a blocking anti-ACE2 antibody that block infection of all VOC tested, including the NL63, and we hope that this antibody will be effective against a future VOC. And in the second part of my talk, I will talk about the dominancy. We know that each virus has different time that is infected. For example, the RSV infected during November, December. It's in Israel, maybe in your country it's different, but you know each virus has his own time. So RSV it's about November, December, and then the influenza arrived in around January, February, and the HMPV arrived in March, April. Each virus has his time. But what happens when a new variant emerges? What will happen with the other viruses? So this is the beginning of the SARS-CoV-2 in Israel, and you can see the wave of the SARS-CoV-2, and this is before the SARS-CoV-2, the amount of the patient in our hospital with that infected with influenza. It was really huge. A lot of patients we had in this winter season, 1920, before the corona. And you can see a lot of patients with H1N1 PDM and flu B and H3N2. And then the SARS-CoV-2 arrived, and we couldn't see in the winter season any influenza in Israel, but it's not happened just in Israel. In all the world, we couldn't see any influenza at all. And then the Delta arrived, and this is the time that nothing, not corona, not influenza, no RSV. I know it. You know, if I am influenza, this is my time, and this is exactly what happened. In 2 weeks, we had a huge of cases of the people that were infected with the influenza. But what happened, what is this gray area in this time? It's the Omicron that arrived, and in 2 weeks, the influenza gone from Israel. So we can understand that the dominant of the SARS on the influenza is really important. Let's see what's happened with the other viruses. The RSV, instead of the arrive in the beginning of the winter, we can see it in the summer, during July, August. The same the HMPV, July, August, viruses that it's not their time, because it's between the 2 peaks of the SARS. But look at the adeno all over the ear. So some people say maybe it's the mask, maybe it's there's a lot of reasons why it's happened, but it's the dominant. It's not just the other things. And I'm in the lab many years ago. I'm sorry, but I was in the last pandemic from 2009, really bad, but I'm still here. So this is the swine influenza. The family is Automyxoviridae. It's a 8-segment of single RNA genome with lipid envelope. So it was a combination of swine, avian, and human together. We got this assortment virus, and this is the pandemic. And let's see what's happened in this time. So this is the pandemic of the H1N1 PDM, and the influenza, the same. We didn't see any influenza in this winter season. We saw just winter season after that. And the same we saw when we can check the RSV and the HMPV, and you can see the distance here because the RSV arrived later, and for any reason the HMPV arrived earlier, and we can see here the distance between the peaks, and you can see the distance in the other years. So it means that again the influenza was dominant on the other viruses, like we saw with the SARS. And this is the adeno. We saw already this type of picture because the adeno is all over the ear with or without the influenza PDM. So what is the mechanism? There's maybe a lot of things, but we did a lot of work about influenza and the other viruses. So if we infected mice with influenza and we take the lungs and do a RNA-seq, we can see 2 waves of protein that most of them belong to the IFITS protein. You can see one peak when the mice was sick, but then after they recovered already from the influenza, we could see one more peak of this protein. So this protein belongs to the interferon system. It's there's a few functional, especially inhibitor of viral process and viral replication. So we think that maybe this is part of the mechanism of the SARS now, but we are working about it, and I will not going to show it, and just want to thank my people in my lab and the collaboration with the Israeli CDC and the collaboration, a lot of collaboration with people in the Sheba Medical Center, and one collaboration with one from Jerusalem University, and that's all. Thank you for you.
Thank you very much, Mikal. Okay, there's already the first question coming up from Daniel.
Thank you very much, very nice. So I was in doubt regarding the immunization of mice with the AC2 antibody, with the AC2 AGG. So you immunize mice, and then you make a lot of neutralization, say, but the purpose is not to be used in humans, right? Because one cannot use mouse antibodies to use in humans. And on the other hand, if you make an immunization of humans with an endogenous ACE2 sequence, you will have to use very potent adjuvants or something to elicit actually a protective immune response, because many of the examples that there are in therapeutic vaccines for cancer or something that people try to generate antibodies against, for example, EGF receptors and several things, because they have human sequences that are endogenous proteins, the immune response is very weak. In humans, of course. In mice is one thing, but then we cannot use the antibody from mice in human, but if we use the human sequence in humans, the immune response will be low. What do you think about it?
We use the extracellular of the human ACE2, and we inject it to the mice, and then we take the split, and we did all the monoclonal antibody with impridomas, and this all the work. It's the same for. Yes, we immunize the mice with the IG ACE2 that we did.
And do you think that will elicit a full immune response? Not immune response, but you want it as a
Yes, we hope so. It's the same like the Evo shield that they gave against the spike protein. And we will do it in mice. Now we will start to do a very big work with mice, with all the variants. So I'm sure that this will be one of the important things that we are going to do. Because it's hard, because we need to do everything in BSL-free, and now we have the BSL-free for the animals, and we will do it.
Yeah. Very nice presentation. You were focusing the initial part on the antibody titers against different lineages. I think that there are very nice recent paper published by the Cleveland Clinic where they analyze protective efficacy of booster based on the XBB lineage in 50,000 medical health care personnel. And the thing that puzzled me is that when they were looking at the time in which XBB was circulating, they have just 54% of protective efficacy. That is a far cry from the initial postulated 1995. And this dropped to 19 when JN1 arrived. Even more concerning probably is the fact that they have, by multivariate analysis, a clear correlation between the number of boosters and the likelihood that you are getting infected, but this is a different story. What is your take from this poor efficacy? Something that has to do with the unrelatedness of the strain? Of course, this can be explained in the dropping, but the initial poor efficacy have to do with immune imprinting. What might be involved based on your experience with the different lineages?
This is the problem. This is the problem, because you can see all the time new and new. You saw all the colors, and it will not stop. And of course that you have cross-reactivity, but it's not enough. We saw it, and there's a lot of, I don't know, we need to check it, but it's not good enough against the new variant, and we know it, not just in the lab. You can check it in the patient that's infected with we have a lot of groups that we can look after them, you know, that you can have we have a really big cohort on our Sheba Medical Center where I'm working. It's the worker in the hospital, not the patient, it's the worker. And then it's healthy people that we know exactly in which viruses they infected and which vaccine they got, and we can have sample from them 1 month after the vaccine, 2 months after the second vaccine, third vaccine, so we can check this patient if they infected and after which viruses, and we have a lot of work about this too, to show that even if you got the vaccine, you could infected with the virus, and it's awful for a while, but we are waiting for the new vaccine from you.
Okay, now
maybe it will be better, because mine is really expensive because it's a monoclonal antibody, so we will give it just for specific people, but we are waiting for your vaccine. And
yeah, my question is related, is from the molecular point of view. Because there are some, of course, it's crazy, this amount of variants, we cannot follow them with vaccines. Otherwise, we will need to change the vaccine every year. That is what is recommended now by WHO, but this is very complex for the industry. So my question is, because there are some data coming from Moderna experiments that they demonstrate that when they use RVD instead of the spike as target antigen, they have better cross-protection against the new variant of the virus. This is the question. What's your opinion? Because I know that you have studied very well the interaction between the RVD and the human receptor. What do you think about these key mutations that are located more on the RVD, but also the virus cannot change completely the RVD, otherwise they cannot find the human receptor for infected cells. What's your opinion about that?
So of course it will be much better, but we will see. We will. Of course that it's not changed so much, and maybe it will be better than the spike. But until now in Israel they gave just against the spike. We work a lot with Pfizer vaccines and with Moderna vaccines, and we are waiting, and we are waiting for you, of course.
Okay, I think I would like to thank Mikael for this great talk, and I guess based on the discussion, I think there will be developed a couple of new collaborations. Okay, now it's a great honor to introduce the next speaker, that is Professor Carlos Guzman. He is currently head of the Department of Vaccinology and Applied Microbiology of the Helmholtz Center of Infection Research in Braunschweig, and he has studied in Argentina at the National University of Rosario medicine, and then he became also medical doctor of the University of Genoa. He has also two nationalities, I think Argentina and Italy. And then he also even did his PhD then in microbiology sciences in Genova. And after this, he moved to the former so-called GBF, it's now the Helmholtz Center, to Braunschweig, and then also I think you went for a couple of years to Hanover, and then back to Braunschweig, and he's really very much interested, you will hear it, on the vaccinology, in particular with this goal to prevent and also treat various infectious diseases with focus on immune responses of the development of adjuvant systems as well as delivery systems, and also the development of novel vaccine candidates for testing. Thank you very much.
Okay, thank you very much for this nice introduction, and I want to thank the organizer that are giving me the chance to present part of the work that we are carrying out in our department.
Laser pointer or normal mouse? Pointer or normal mouse?
Normal mouse.
Okay, then please go.
Okay, we have many diseases for which there are no vaccine available. We have diseases for which we have vaccine, but the vaccines are not so good. But what is needed to develop a vaccine? And probably seems trivial, but we need an antigen or antigen that are able to confer protective immunity. But this is not enough. We need also to develop technologies that allow us to promote the type of immune response that is conducive to protection. Because if we are stimulating a strong immune response, but it's the wrong one, we might be causing more damage than good, as was the case for the first vaccine that were developed against respiratory syncytial virus. How can we stimulate what is needed? Okay, there are different means. One of them that is prototypical for subunit vaccine is the use of adjuvants. Adjuvants help us to improve the strength of the response, but they also help us to modulate the response according to the clinical needs. And they help us to reduce the amount of antigen that is needed, that is particularly important if we have a restriction in the batches that can be used and the doses that can be generated from these batches. We can speed up responses that sometimes is trivial and necessary, but in many cases, like for travelers or in pandemic, is of the sense. And of course, we can promote long-lasting memory. The problem is that we have only a few adjuvants that are approved for human use. Indeed, the adjuvants are not approved. The vaccine containing the adjuvant are the ones that are approved. And we are having a lack of adjuvant that help us to evolve mucosal vaccines. There are also another bottleneck, that is that they are more or less a virtual monopoly on the adjuvant that have well-defined molecular market that makes sometimes a bit difficult for the academic group or the small biotech to have access to these, let's say, new generation tools. These were the driving forces for us to establish a few years back a program focused in the identification of entities that have this activity, immunomodulatory adjuvant activities, but we want to focus our search in adjuvant that we are somehow filling the gap. So adjuvant that were not only active by parenteral route, but also by mucosal route, adjuvant that were not only modulating or promoting humoral responses, but also very effective cellular immunoresponses. And we have different candidates, among them the cyclic D nucleotides, and the one that I want to present some data to you today is the cyclic D AMP. This molecule has a very well-defined molecular target. It's an agonist of STING that is triggering a TBK1 IRF3 cascade, leading to the production of type 1 interferon and TNF. It's activating different key immunocells for the initiation of an adaptive immunoresponse, and interestingly, is promoting different effector function. Antibodies, different T helper subsets, cytotoxic T lymphocytes, and one of the very interesting features is that it's also active in situations where you assume that there are poor responsiveness to vaccination, like in the very young, the very old, in comorbidities. And another important feature is that in contrast to other pattern recognition receptor agonists, like for example poly-IC, cyclic D-AMP is able to promote an immunoactivation that is restricted to the injection site and is self-limited in time. That, of course, is good in terms of reducing the likelihood of reactogenicity. And is also amenable for combination with other technology platforms that you can use for antigen delivery. I want to give you a couple of examples. Several years back, we started a program in cooperation with Crucell in which we were looking at, for example, sublingual vaccination against the avian flu virus, and we have virosome-based formulation. This is basically like a liposome, to make it simple, but the virosome per se are immunogenic, are promoting an adjuvant effect. And here you can see that when we vaccinate with virusomes that were generated with the NIVRG14, that is a vaccine virus, and H5N1, Vietnam-like, you have antibodies that are able to prevent the hemagglutination by the virus, so they are biologically active, not only against the Vietnam-like strain, but also the Cambodia-like strain. More striking, and something that we have not observed with the benchmark adjuvant, is that we have also activity that was able to neutralize the H9 and the H7 virus. This was also the formulation that promote, the only one that promote multifunctional T cells recognizing the antigen. And this is very important because there are several studies showing that this type of responses protect more efficacy, with more efficacy to vaccination. And this probably explains that when we test these formulations in a murine model, in the case of the adjuvanted formulation with cyclic D-AMP and GMP, the animals that were vaccinated after challenge maintained their weight, but we observed the weight loss in the controls and also in the mice that were vaccinated with the H5N1 virosomes alone. This was more or less the information that you use, and we are exploiting now in a consortium that I'm coordinating that is aiming, among other goals, to develop next-generation vaccine against flu that are broader. One of the candidates that we are aiming at is based on COBRA technology. COBRA technology has been developed by Terros in the States and is basically an in silico approach that based on antigens, in this case the hemagglutinin and the neuraminidase, use this algorithm that makes some kind of multi-consensus building that allows the incorporation of all minor and major neutralizing epitope of all subtype of influenza strain, historic and modern strain, and hopefully future emerging strains. And this kind of approach leads to antigens that have a more broader, superior efficacy in terms of cross-neutralization than the native hemagglutinin or neuraminidase. And we are adjuvanting with cyclic DAMP because we would like to protect not only against disease, but also against infection, reducing the likelihood of horizontal transmission from an infected individual to susceptible host, like for example happened with the parenteral vaccine against COVID. We perform a bunch of studies in mice that show us that the adjuvant is essential, show us the cyclic DMP outperform benchmark adjuvants, that the best responses were obtained by intranasal route, and that the cyclic DAMP was able to promote broader IgA subclasses and also secretory IgA in vaccinated animals, and that these antibodies were biologically active because they inhibit the hemagglutination, they neutralize the virus, and also by ELA test we observe activity against the neuraminidase. We also stimulate multifunctional T cells, and we confer protection against virus challenge. But we need to keep in mind that mouse are not the optimal system in order to predict for vaccine efficacy in humans. And the system that is used is ferret. And again, we need to keep in mind that with the exception of infants, vaccination against flu is not a priming, it's a boosting. We are all in contact with the virus or have been vaccinated or both. So one of the models is naive ferret for infant, the other model is immune-experienced ferret that are pre-infected with different strains. This vaccine candidate promote protection in naive ferret, and here you are showing you what's happening in immune-experienced ferrets. We have different formulation, pentavalent, octavalent formulation, high, low dose. What we define is that considering seasonal strains, we get efficient protection with a low base dose pentavalent formulation. And we also got faster viral clearance in the nose of the animals that were challenged. Now what we are doing is we are going in the GMP manufacturing. We want to perform the pivotal tox studies, and this should lead to phase 1 and 2 studies later on. The second example that I want to show you is focused in a different bag. It's not a small one, it's a big one. It's Trypanosoma cruzi. It's the etiologic agent of Chagas disease. This is a disease that is endemic in 21 countries. Is not in non-endemic countries. We are seeing this in 19 countries. And we have, and this is a huge underestimation, at least 10 million people that are chronically infected. From these 10 million, at least 30–40% will evolve with time, very invalidating like creatinine, cardiac or gastroenteric forms. This is costing in terms of quality of life daily, it's costing in terms of the money that need to be put to treat these patients or their sequels. And they are drugs, yes, but they are only useful very early on in infection. If you are prolonging the infection, you cannot cure with the drug, and there are no vaccine available. So what we decided is to put together another consortium, Crucivac, that was bringing from bench to vet a candidate against Trypanosoma cruzi. What we were in principle confronting is that in contrast to other diseases, there are no clear correlate of protection. But we know that there are certain effector mechanisms that are playing a role in vaccine efficacy or in protection against disease. The problem is that it's a bit complex. Antibodies are helping, but not too much, but need to be of a Th1 pattern. We have cellular responses that are the main drivers. We need, of course, T helper 1 cells. We need polyfunctional CD4 and CD8 cells. We need also T helper 17 cells. And last but not least, cells that are producing interleukin 10 to reduce damaged by the parasite. So this is not an easy pattern to get, and we try different adjuvants. At the end of this preliminary study, we set up for cyclic DAMP. And the antigen that we were using is a synthetic trivalent antigen that encompasses domains from crucipain, from the transhyalidase, and the amastigote surface protein 2, because the bugs, the parasite, have different biological form, and you need to be able to tackle all of them by using antigens that are expressed by all biological form or a mixture of them. So the initial studies were performed in mice. We compared different administration strategies, mucosal, parenteral. We did studies in different sexes. We did studies in different ages. In a nutshell, what we got is that the vaccine was extremely well tolerated, that we were able to stimulate the pattern of response that we would like to stimulate, and there were no age or sex biases. And we also defined the dose that we would like to use in further studies. We also performed an initial proof of concept in a model that is lethal of Trypanosoma cruzi infection, and you can see in green that the vaccinated animal survived, that the parasitemia is in principle under check, and if you are looking the heart of a non-vaccinated and a vaccinated animal, you realize that there are no inflammatory infiltrate. Not only that, in zero you don't have many enzymes that are pathognomonic of muscle damage. This was good, but not that good. Why? Chagas is not killing. Chagas is usually a chronic disease. The infection from Chagas is usually asymptomatic or oligosymptomatic. So we evolved a model of early chronic phase in which we test our vaccine. And what we observed there is that, again, in the vaccinated animals, creatine kinase as marker of muscle damage was low in the vaccinated animals, that we have lower load of the parasite post-challenge, that we don't have infiltrate or more modest infiltrate in the myocardium, and that we have a reduced fibrosis of the heart, and something that is pathognomonic for Chagas. Chagas affects the heart, and you have electrocardiographic alteration. In these animals, the electrocardiogram was normal. We don't have a prolonged PR interval. Then we move at another species that is also a natural host of Trypanosoma cruzi, that are dogs, particularly in endemic area. You are seeing on the right that this animal that is vaccinated, the fur is normal, there are no alteration. Again here, creatine kinase low, we have a PR interval in the electrocardiogram that is normal, we have an area under the curve for parasitemia that is more modest, and we have, again, in the upper panel, in the vaccinated animals, lack of inflammatory infiltrates. So, what are we going to do now? We are performing studies in non-human primates, and we already perform some toxicology studies that allow us to select the animal species to perform the pivotal tox that is currently running in rabbits, aiming at completing the preclinical package for applying for a phase 1 trial. The problem of Chagas, if you remember what I said very early on, the drugs are useless when you are in the chronic phase of the disease, and this is a huge problem. And there are these antiparasitic drugs like nifurtimox and benzimidazole that are not really working unless you give in the very early phase that usually is asymptomatic. It's a conundrum. So what we want to check if the vaccine, particularly in combination with the drug, will help for the therapy of these 10 million people or more that are chronically infected. And we evolved a model, or we use a model in which the animals were infected, then after 120 days they were vaccinated, or vaccinated and treated, or only treated with benzimidazole, and we were looking at what happened after 240 days, a hell of a lot of time for mice. And this is what you are observing before vaccinating, post-challenge in the heart of these animals. What you are seeing is that you have a prolonged PR, QT, and QRS interval. So you have electrocardiographic alteration before your therapeutic intervention. And this is what happens if we have animals that are controlled and we treat with benzimidazole. This electrocardiogram comes with certain improvement, but it's not like at basal. But if we are doing the combination of the drug with the vaccine, we go down to what we are expecting in animals that are healthy and challenged. Then we are looking in animals that receive this combo therapy at the stain sections for inflammatory infiltrates, and again, a considerable reduction. We are looking by Pachyrocereus stain section at the fibrosis. This is again down. And to give an optical hint, this is the histopathology of a normal tissue. This is what we are observing post-challenge, and this is what happened in the animal that received the vaccine and the drug. In terms of immunoresponses, what we are seeing is that in control animals that were just challenged, we have very few multifunctional T cells recognizing the pathogen. If we are having benzimidazole, this is not changing too much. When we vaccinate, we are getting an increment in cells that are producing 2 or 3 different cytokines. When we have the combined treatment, we have the maximal stimulation of multifunctional T cells that are producing 4, 3, and 2 cytokines. We are coming from 25% to 74%. So, in the last few minutes of my presentation, I want to show you what you can get in terms of versatility of this technology. First of all, this is also working in model of poor responsiveness, I mentioned before. Here is the kind of responses that you are getting using a golden standard adjuvant. In young, in blue, and in aged animals, of course, responses are poorer. When you have cyclic DAMP, you have an improvement in young animals, and you have responses in aged animals that are at the level of the responses that we are getting with the golden standard adjuvant in young animals. And these are animals that are 1.5 years old, real grannies. This is a different model. It's an ADCO knockout mice. These are animals that have a knockout in a transcriptional factor that is important for lipolysis. These are becoming huge. They have a lot of adiposity. They have glucose intolerance and insulin resistance, showing more or less the pattern that we are seeing in diabetic patients. If you are seeing here in animals with this phenotype that are vaccinated without the adjuvant, we have under low-fat diet very poor responses and extremely heterogeneous. If the animals are on a high-fat diet, there are no response at all. Once we have the adjuvant, we have very robust, identical and high responses in animals that are on low-fat diet, and in high-fat diet, we have 3 out of 5 animals that have titers that are considered seroprotective. The last example is based on neonatal vaccination. Here, it's difficult to get responses for subunit vaccine. One of the adjuvants that have been promoted is Resikimod, but as you can see with this model antigen, the responses are not that good. When we have CDA, we have a good response, and these responses are in a mock system in which we have an influenza virus that have one protective epitope of OVA clone in a permissive loop of hemagglutinin. We are seeing that if we have Resikimod, the animals drop weight. If we have our adjuvant, the animals are protected against challenge. You can also combine it with nanocarriers. We have an example based on polyphosphacines. In this case, we immunize by intranasal route, and what you are observing that the antigen alone is very poorly immunogenic. When you combine with 2 polyphosphacine, this improved, but the true jump in immunogenicity is obtained when we co-administer this with cyclic DAMP, and this is even better than what we obtain using as golden standard, the polyphosphacine with poly-IC and antibacterial peptides. And we induce also secretory IgA, different T helper success, multifunctional T cells, and CTL. The animals are protected, again, as you can see in the model that I already explained with you, with the 2 different polyphosphacine formulations. And then we move at a different route, not mucosal but transfollicular. This is a route that is not breaching the mucosal, the skin barrier, and allow us to vaccinate. And you are seeing by two-photon microscopy the root of the hair in green, and in red the polyphosphacines. What we used here was a hemagglutinin, and you are seeing here the weight of the animals that were non-vaccinated, and you are seeing what happened when we have the polyphosphacine with the hemagglutinin without the adjuvant, and here you are seeing side by side with the adjuvant by transfollicular route, non-invasive, and in comparison subcutaneous route, exactly the same. And by in vivo imaging, with a virus that is expressing a GFP, you are seeing that in non-vaccinated virus we have animals, we have in principle viral replication in the lung. When we have the vaccination without adjuvant but with the antigen, nothing changed. And in the two groups that I mentioned before, with the adjuvant, transfollicular and subcutaneous, we have a good protection. So where are we at the moment in terms of bringing this into the clinic? There are a clinical trial running for a next generation vaccine against papillomavirus that is thermostable and have a broader pattern of a subtype that can be neutralized, 18, in a cooperation with the Cancer Center in Heidelberg. Is starting this month a phase 1 trial for a therapeutic vaccine against hepatitis B by a prime boost protocol. We are evolving a mucosal formulation as a prophylactic vaccine against hepatitis C. I already mentioned Chagas and flu. And we also evolved different studies in cooperation with the Center of Genetic Engineering and Biotechnology in Cuba for COVID-19. And we then also, there are studies running for a dengue vaccine. More recently, we started with a consortia that is focused in the development of Helicobacter pylori vaccines under the leadership of the Technical University in Munich. So in conclusion, cyclic DAMP is an adjuvant that promotes humoral and mucosal immunoresponses, that is active by systemic and mucosal route, that is active in model of poor responsiveness to vaccination, and that promotes this self-limited immunoactivation restricted to the inductive site. How? Is agonizing with STING, but is targeting STING to proteolytic degradation, in contrast to other agonists of pattern recognition receptor. Improve also the efficacy of nanocarriers, and I have not presented also RNA vaccines, and we are developing at the moment CDA adjuvanted vaccines against different indications, 8. The acknowledgement, I put the people with whom we collaborate in the slide that correspond, but I would like to highlight here our cooperation with the team in Cuba, since I have not presented the specific part of the work. And the big recognition is to my team. I'm presenting the thing, but they are doing the work. And I'm very proud of them, and they are the vital links that in principle lead to this data. Thank you very much.
Thank you very much for this great talk. It's open for questions.
Everything was very clear or completely unclear?
No, it was everything very clear. I would say, okay, Ludka.
Yeah, about the adjuvants, bringing them to the market. So the requirements are fully the same as for a normal drug, with all the clinical phases and everything and the costs.
The thing is the following. The adjuvant, you are not going to get an approval for the adjuvant, you get an approval for the vaccine that you are developing, and one of the components is the adjuvant. The adjuvant, according to PI, is considered an excipient. The thing is, if you were never in a clinical trial with an adjuvant, the level of astringency increases. You need to deliver much more data. Because they want to be sure. It's the same, no? Novel antigen is an issue, novel antigen and an adjuvant, novel antigen, novel adjuvant, and novel delivery system increase even the threshold. But I think that at least in Germany, the regulators are extremely helpful. They are performing research themselves, so the scientific and early scientific advice, I would say that is a must, because they are telling you, for example, things that are related not only to the preclinical development of your vaccine candidate, but also, for example, is the way in which you are planning the manufacturing of your GMP grade material for subsequent GLP talks and trials is the right one, is the kind of QA that you have associated with it is the right one. So if you are more or less posing the right question, they are instrumental in helping you to develop it. And we did it for the adjuvant itself, and for each of the indications that I mentioned, we have scientific advices in Belgium, here in Germany, and was instrumental really to help. And we have a lot of data. For example, we have done also PK/PD studies to understand how long, what is the half-life of the adjuvant, where it's present. For example, we know that have a half-life of 18 minutes. Given by parenteral route. By mucosal route, we are not even able to detect it. So, interesting feature.
Are there other questions? If not, thank you very much. And I think we have now a coffee break for, I don't know, half an hour or? Okay. Then we meet in half an hour for the last session. Thank you.
Das ist aber auch im Moment okay, weil das momentan wahrscheinlich zu ist, unser Mikrofon.
Also nicht jetzt, sondern beim Vortrag sagte sie das.
Ja, also das Teams, ich habe hier das gemutet, einfach, unseres.
Aber doch jetzt erst.
Naja, vor...
Ach so, okay. Also sie hatte das so um 2:30 geschrieben von der Stunde.
Can you hear us? No. Is there now audio? In Teams? Okay, I can't hear you, but I will change that. Yes.
Genau. Und
Soundcheck.
Also ist das im Anstand. Da brauchst du nicht zu gehen.
Aber ich finde sehr interessanten Lebenslauf. Finnland und dann ist geboren, danach Kanada.
Gab halt nur Konflikt mit der so das Präsenting. Gucken wir mal, ob wir das also, aber insofern, das interessiert alles nicht.
Okay, Input 1.
can hear you. Okay. And you also clearly understand. That's it. Okay. So then half an hour, so, no, not half an hour? How much? How many minutes?
In 10 minutes we're to make the test, but we can make the test perhaps now.
Is she? No, she's...
I can call her.
Please.
Nice to meet you.
We need to close our microphones when you start talking, otherwise we would get a mix of internal sound and team sound and so on.
Okay, can you try to share your screen and then talk and we turn off our mics?
Okay. Share. So I go to a window here, and here we go.
That looks
and
good.
I go to the beginning. Okay, how do I go into presentation mode? Yes. No, wait a sec. I'm sorry. Let me redo this. Stop sharing. Share again. And list PDF. Here we go. Okay, I'm in the PDF. How would I go now in the
You probably need to talk to the microphone, otherwise the sound is very much reduced.
Oh. Here. So okay, first, I'm not sure how to share now the presentation in screen share mode, and secondly, I have the volume on here. I don't know how else to.
For screen share, you need to press Control L.
Okay. That's the way it works in MS Teams, right?
Sometimes you're clearly to understand, and then your sound vanishes. I don't have an idea why that happens.
Okay. computer, so I have no idea what that could be.
But do you have a head? Do you happen to have a headphone with a microphone like this?
Sorry, because I
Now it's pretty good, actually, isn't it? Now it was okay.
Okay.
I'm asking the technician.
I can't see because our monitor was not working properly this morning, and if I would do too much switching and plugging upstairs where the technical equipment is, then I would risk to end the YouTube stream, and therefore I'm a little bit hesitant to do that. But the normal audio signal is from your computer. I don't know which part of the electronics is responsible. So it could be on your part, it could be on our side. I'm really sorry, but I don't have an idea how to resolve the situation. So you should probably direct your talk directly to the microphone and maybe with some pitch in your voice, or how to. I'm unable. So I have also poor command of the English language to explain. Because if you really start talking, it is understandable, but as soon as you turn your voice turns a little bit lower, some algorithm probably kicks in, and then we can't understand you.
Okay. I should talk slowly and slowly, like this.
Yeah, that was okay, and yeah, in the end we'll have to see. Yeah, it will be difficult, and it will be probably not the best, but unfortunately, I'm also not an expert for the entire setup here in the university, and that is the problem. It's too many variables.
And you should look happy and unconcerned and looking forward to going to the beach after this.
Oh. I don't have an idea. Could you just try once more, because it obviously is some local problem.
Oh, I can do maybe presenter mode. So.
Just continue talking.
Okay, I'm talking now, and I'm gonna put Control L. Okay, so is this is kind of, is the presenter mode better, or should I don't know. Sorry, I am also kind of unfamiliar with the way it works in MS Teams. Is this good now, the way I'm doing it?
Yeah, looks good.
And the way I'm talking now, because the first slide I can say this is our project.
It varies. Sometimes you're very silent and sometimes you're very loud. Maybe we can try one more thing. In the Teams UI overview, you have 3 points in the top menu, at least for me.
Okay, I need to go to Teams here. Let me, stop sharing so I can see. So where do I go?
In the top menu, you have 3 points. I have the German version here. It's called Weitere. I think it's others.
More. I got it.
Or more,
okay.
yeah. And then there is some audio properties.
Language and speech, but I'm not sure.
No, that the one above it.
Okay.
For me, it's Audio Einstellungen.
Here, to me, it's video effects.
Oh.
It should be audio settings.
Because for me, it's audio settings.
I have no idea how to deal with it.
No idea.
If I'm just the only person online, would it be possible for me to zoom in? Go through Zoom instead of MS Teams? See if that worked better with the audio, because I was on a Zoom earlier and there were no problems with the audio, so I don't know if that's the factor here. I mean, I've had other Zoom meetings and I haven't had a problem here, which makes me think that it's maybe on that end or it's MS Teams.
Phase-wise, it is understandable, but so I didn't get really. I was listening and thinking about what is going on. Were you supposing an alternative? So if you
I can't hear anything. I don't know if somebody is talking, but I can't hear anything right now.
We could try also Zoom, but that may come with other problems.
So it seems to be a problem of the room.
Fabian, du musst dein Mikrofon zumachen. Das ist alles verkehrt, aber ich meine, ich habe nicht die Zeit hier eben, ich müsste das komplett umbauen und habe keine Idee, ob das wirklich helfen würde.
Sollen wir versuchen, nochmal rauszugehen und reinzugehen? Try to turn it off and on again. Marika, okay, no, okay. Maybe we just try to turn it off and on again, so just leave MS Teams and try to get back in. I'm really sorry about this, it's a mess, nobody knows why. But just try to leave MS Teams and try to join again, okay?
Okay.
So viel halt Rauschunterdrückung und sonstige Technik dahinter und ich beherrsche es nicht, ja, weil mir ist das quasi so benutzfertig übergeben worden und man müsste eigentlich das alles mal durchtesten und dafür hatte ich nicht die Leute.
Naja, es ist wie es ist, also notfalls singe ich es von hier. Okay. Hello, Marike. We see you.
Hi.
We see you now.
Okay, great. Can you hear me?
Yes.
Yes. We can hear you.
Can you hear me well enough?
It is but the system may have some algorithm which kicks in only after 2 to 5 seconds, and then it reduces your voice, and that is my impression. Yeah, your first words always are clearly understandable, and then after it is regulating down your voice.
But let's try. Let's talk a bit, Marie. Talk a bit, Marie.
Okay. Yes, I'm talking now. How does it sound? I'm talking in a loud voice. Okay, can we try Zoom?
It is some algorithmic thingy which prevents it.
Okay, can we try Zoom?
Zoom, yes. Can you paste the link in the chat?
Just a second.
Okay, my colleague tells me that you're clearly understandable on his computer upstairs, and then I have to I have to get an idea how we can get your voice to the room. It could be also something upstairs. I need to ask my colleague to just maybe the camera or our yeah, this is difficult to tell. I'm hardly able to explain it to him. I will try to look upstairs.
Okay, if you think we can then no problem, right? Or if I can do this.
So this was which camera? This one is we should have this one. Maybe take this one off. Maybe we have now, yeah, because this entry, ja, also Eingang 1 ist quasi diese Funkbrücke, da kriegst du die ganzen Signale von mir unten. Und dass du jetzt quasi das ausmachst, nee, das hilft uns auch nicht, weil dann sehen wir die Präsentation nicht. Ja, ich sag ja. Aber vielleicht kann man das Audio, keine Ahnung. Gut. Verflucht. Hier ist. Das ist der Teil. Ja, okay. Möglicherweise Glück, dass wir das. So. There is now here again the audio signal. Okay. I will go downstairs and let's see.
This is the test from the low keys. CCB. Could you say something again?
Yes, now
in Teams it's okay, but we need to check if the room audio is also okay. Could you please continue talking?
Yes, so now can you hear me in the room? How is it working?
Yeah, it's much better now. The only question would be,
how the YouTube sound is, but I would probably sacrifice the YouTube sound.
Yeah.
Oh, it starts. No. It went away. I wonder if I'm really out of ideas. So, I'm not sure because it is obviously not a Teams problem. It is a problem of our machinery here in the room, and so there's just voice or noise cancelling kicking in, and yes, I will try one more thing. The link doesn't open. I get just Zoom international phone numbers. You could try, and I will follow. If it's that link here you copied, then I will try to open it. But currently I cannot open it.
Okay, well, I'm also sending it to Bill's email because I don't know why it's not opening. So I'm also going to send it to Bill's email.
You probably dictated to me and then... yeah, this time your voice also was okay, yeah, but now she's too late.
Werte von einer Woche.
Ich sage ja immer,
Funktioniert es? Ja? Funktioniert es?
Sonst müsst geht? Jetzt geht vielleicht. Ich lasse die Techniker, die verstehen mir davon Hello, I just on.
Musst du unten anmachen.
Anmachen.
Da ist irgendwo so ein
Also ich
muss man hochschieben unten.
Ja, du kannst das auch nehmen.
Hello. Is this on now?
Ah ja. Danke, was das ist.
hear us?
I can hear you, yes.
And we can hear you loud and clearly. So I just need Fabian's indication whether the YouTube is well. Okay, then probably everything is in place now. But we need to share the you share the screen. You share the screen for presentation.
Muss das hier ran?
Ja, jetzt woanders.
Andersrum?
Flipped.
So?
Okay. Because here that small
okay.
hole is.
Okay, we will start now the last session and move now from immunology to social sciences. And I will introduce two people. The first one is Maria Bloomfield. She is actually from Finland, but has a very international CV. She studied then at the University of New York in Canada and performed her bachelor as well as her master degree at the York University. Then she moved to Chapel Hill in the US, performed there a PhD. In between she moved to many different countries and worked all in different countries until 2020. She worked at the Miami University, then moved to Germany, to the Institute of Hamburg, and now she is currently director of the Institute of Political Science at the University of Hamburg. And the second person whom I would like to introduce is actually Bert Hoffmann. Bert is member of our GLACIER consortium, and he is a political scientist, and in particular he studied in Berlin political science, performed also there his PhD, and already moved into the days of his PhD into politics of third world countries. And currently he is actually head of the German Institute of Global and Area Studies, the office in Berlin, and he is since 2023 actually president of CISA, that is the European Council for Social Research in Latin America, and he leads his research, or both research focuses actually on political and social sciences in Latin America, and I just would like to give you words, and we are looking forward to your talk.
Okay, thank you very much for the nice introduction. I'm not sure if you have been aware, we've been struggling the past half an hour with the audio. I hope now everything is working. This Marika, can you hear us?
I can hear you. Can you hear me?
Yes, and we're not at the stage for the last half hour, so this is really just in time. and I'm very happy to be here. Thank you very much for organizing this great GLACIER get-together symposium. And we are aware that we are on the third day and the final presentation, so I hope we will make it kind of smooth, and it's not a topic that will be easygoing, and we will do it together. Basically, Marika will share the slides, and Marika, you will see on the first slide that we've been a team doing this. From the German side, it's Marika and us and me, and we have very much of the work was done by the Colombian team. You have the names Rafael, Doris, and Magali, and we also had a collaborator in the US, Andrea. And most of the work really was done by those persons and by Marika, and that's why I will pass most of the time speaking to Marika. I hope it works. What I would give is a little bit the context, what brought us there. So could you, yes, the context is, most of you are well aware of it, of course. We have this huge social crisis in Venezuela with an outflow of migration which really has reached tremendous proportions, and in neighboring Colombia this is being felt very, very severely. And with this immigration is coming infectious diseases along, and we've been focusing in a way on that and on one specific one. So the next slide, and then I will already kind of turn it over, the disease. So we are in the region, up at the border region between Venezuela and Colombia. I don't know if you can read it, Norte de Santander, the city on the border is called Cúcuta. And in that border region, we have this rise in congenital syphilis in a very severe, in a very high rise of a very severe health consequence, which brings terrible human suffering and which of course also has immense costs associated with it. And with that, I would already pass it on to Marika. You take over, Marika?
Okay, you can hear me okay? Okay, I pass it then. Yes, exactly. And so precisely one of the key areas where a lot of people cross Venezuela to Colombia is in the north of Santander and Cúcuta. And as a result, indeed, you've had a significant rise of babies being born with congenital syphilis in the public hospitals that provide emergency care. And of course, that is reflective of larger levels of latent syphilis in the population. And this level is high. It was increasing dramatically. It's maybe plateaued or even declined a little, but it's way beyond what it should be. And it has severe health consequences for those newborns as well as the human and economic costs. And so this drove us to investigate this with a grant from the German Institute, the GLACIER, through a bigger project with the Charité involved as well and other institutions. And so we wanted to investigate sort of the social determinants of this issue. And first, to put it into context very briefly, I mean, syphilis, and you probably know this, but it's an incredibly preventable or treatable problem. Since penicillin was discovered, there's been a very easy solution to syphilis, and so it's one of these kind of tragedies that do not need to exist. The infection is completely treatable and curable by a shot of penicillin, or in most cases for syphilis of unknown duration, it is 3 shots of penicillin 1 week apart each. And so the key here really to basically eliminate syphilis is through early detection. And of course, given the migration, given social collapse of the healthcare system in Venezuela, the underlying conditions are creating conditions for the transmission of infectious diseases. And so there's those kind of social conditions, but also in terms of just addressing syphilis, the issue is one of early detection of syphilis. And one of the problems is that when you get syphilis, you don't necessarily present the symptoms right away, or you can actually have latent syphilis without knowing it for quite a long time. And so the problem is detection, and after that, access to this treatment of 3 shots of penicillin, and not only access to the treatment, but adherence to the treatment. So actually going back, first having that shot and going back twice 1 week apart. And of course, addressing this has significant human and economic benefits. So what we did was, this was a development aid project, and so we had 2 components to our project. Importantly, a community health services component that was kind of a practical component, which involved a community health pilot program that we got funding. That's what we got the funding for, and that was implemented by an NGO, Care Colombia, last year, between March and July. And what they did was, with our supervision, is go out into vulnerable areas outside the Cúcuta, basically settlements where Venezuelans would often come, but also that also had vulnerable Colombians living there, and create these workshops focused on prevention, diagnosis, and treatment of syphilis. Now, they weren't called syphilis workshops, they were called workshops on sexual health, something very broad to, because people won't necessarily go to a syphilis workshop. And the reach of these workshops was almost 2,000 people. It involved 83 group events, so basically what they set up were these half an hour to 1 hour interactive group workshops where they went through material related to, sexual health, gender relations, and then really zeroing in on the issue of syphilis. Following this, all the people were, with their consent, rapid tested for syphilis. And then also a big component of this was in general sort of education about STDs. And so CARE also focused very much on delivering condoms. Through this, and basically everybody who attended a workshop consented to being tested, and the positivity rate was 6%. This is really very high. The 6% was also rapid tests. They were followed up by conformed laboratory tests, and that was the 6% reactivity or the positivity rate. And those people who got it, they got free access to treatment and counseling and kind of a follow-up treatment plan. So then, in here, we wanted to actually embed studies to find out more about the social determinants to inform better public policies on a national scale, right? Not just in a pilot project. And so our goal here was to investigate the relative weight of different social determinants. And our hypothesis was, well, there are different factors that can cause the rise of syphilis. One is simply people don't know about syphilis. If they find out what it's all about, they will, avoid it. Second, well, for example, women might know a lot about it, but be unable to sort of say no to unprotected sex that might, possibly help with the spread of syphilis, and so it might be machismo. Well, perhaps it's not neither machismo or lack of knowledge, but people might know they have syphilis, but they simply have no access to healthcare in these conditions, and so they are unable to go and get tested and get treatment. The fourth hypothesis that we posited was that given the conditions of social vulnerability of these people, many left Venezuela because they simply couldn't feed their families. They face a critical lack of basic needs, and so even if they know about syphilis, and they might be able to somehow access healthcare with the great difficulty, they simply cannot. The opportunity cost of going to deal with it, especially if they don't have active symptoms, are higher than prioritizing basic needs to, for example, engaging in income-generating activities in the informal sector, etc., or the cost of access to the healthcare clinic by having to pay for a bus ticket. And on this basis, then, we designed 3 studies. 2 were qualitative studies involving interviews. The first one here was interviews of risk groups, and we interviewed 30 adolescents and young people who showed up to the workshops, but of course, importantly, interviewed before the workshops took place to find out about basically their lives and experiences. The second was interviews of those who kind of the system had failed them, mothers of newborns with congenital syphilis, and we did 20 interviews of mothers at the main public hospital that received people for emergency treatment regardless of whether they had official access to healthcare or not, and so they treated a lot of Venezuelans as well. And then the third was within this project that was the community outreach project, and among the 6% who tested positive in syphilis, we embedded a quasi-experiment with conditional cash transfers to see whether cash transfers like small monetary incentives might increase adherence to treatment to test that fourth hypothesis.
process that I mentioned. And here's just the attention, kind of the path of attention, ruta de atención, that was provided to all the people who tested positive, regardless of any other factors. So the logic with that, I'll first explain the cash transfer program and some of the preliminary results, and then summarize the qualitative interviews. So the conditional cash transfers for syphilis treatment. Here, the logic is that, well, we had like all those 114 individuals. They had all received an educational workshop and had access to a treatment plan free of cost with kind of comprehensive care as outlined by the Colombian government, kind of under the best of circumstances, and Care Colombia provided that care. So we had the control group that got information and free treatment, and then we embedded to like almost half the people were in a group that received information on free treatment plus a small cash incentive that was the equivalent of about what one would earn in one day in the informal sector plus the cost of a bus ticket and a snack. So 11 euros twice, because they had to, first they got their first shot and then they had to return a week later for the second and the third shot. And the logic was that if there's no difference in the level of adherence between these two groups, cash transfers don't make a difference. And if there is, then they might make a difference. Here's some data on the characteristics of both all of the community workshop participants as well as those who tested positive. And what it shows is that, you know, these are high levels of vulnerability among this population. One interesting thing, and I think a very important thing also politically to mention, is that these people who came to the workshops, and also those who tested positive, they were not overwhelmingly Venezuelan. Many of them were, about half of them were Venezuelans. However, there was a significant share of people who were also Colombian. And this, in terms of the politics of it, it's important to stress that kind of migrants don't cause, or they're not the only transmitters of STDs. There's a little bit of a worry of this kind of a discourse arising in Colombia, but rather the host communities. These problems exist in the host communities as well. And in fact, internal displacements of Colombians and external from the civil war over the last 2 decades is a part of the problem. You can see here a category, Colombian returnee. Many of the vulnerable people are actually Colombians who went to Venezuela to escape the civil war and came back, and now find themselves also living in difficult material circumstances. Okay, so the logic here was that we had these attendees. This is kind of the logic of the actual quasi-experiment, very briefly. We sequentially assigned people to these two groups. If anybody has questions, I can explain why we felt a random assignation was unethical and impractical for certain reasons. And so we sequentially assigned people to either the control group or the CCT group. And then we also engaged in propensity score matching to basically have two groups of 50 with the cash transfer and 50 without the cash transfer. Briefly, the preliminary findings that we're now writing up into a manuscript that we're about to submit is that, well, descriptively first, just off the bat, those who didn't get conditional cash transfers, their completion rate to the 3 shots was 45%. Those who did get the cash transfer was 80%. So there was a clear difference. And what that also means is that educational campaigns and free access to healthcare might not be enough, right, to actually eliminate syphilis. And the second also is that with propensity score matching, logistic regression, the odds of completing treatment among those receiving CCTs were more than 4 times higher than those who didn't. So it seems that in these conditions, CCTs could be a very effective strategy to reduce the spread of a virus, of syphilis, particularly among people who have latent syphilis. Then the interviews of the women with the newborns. This is the hospital where the interviews took place. Very briefly, some of the socio-demographic characteristics. In this case, 18%, almost all of them were Venezuelans. Many of them come across the border to give birth. They don't have access to prenatal health care because prenatal health care is not considered an emergency treatment, but giving birth is, so they can come to Colombia to give birth. The problem is access to prenatal care in Venezuela, so they show up here to give birth.
births. So many of these indeed were Venezuelans. Their migratory status was quote-unquote unregularized. What means is that they don't have legal access to preventive health care. The ages, 17 to 41 years old, low education, low socioeconomic status, very low monthly income. They all basically, if they worked in the informal sector, and the level of knowledge of syphilis was extremely low. And here's a quote, "I'll be honest with you, nothing, I don't know anything about syphilis." Also a sense of guilt about because these are women who gave birth to a baby with congenital syphilis, and you know, that's a traumatic event. It's one's own fault for not knowing anything, said one woman. Some of the social context and behavioral characteristics, very high levels of vulnerability, unsurprisingly. Psychosocially, these interviews were marked by the absence of support networks. 85% of them did not have access to health insurance. Also, importantly, 85% of them had less than 7 prenatal controls. The protocol is between 7 to 9 prenatal controls, is what you should have. 7 if you have an easy, not first pregnancy, up to 9 if it's the first pregnancy. And so the vast majority had not. About half of them had been diagnosed only in the very late stages of pregnancy. Their lack of access to prenatal controls had to do with the fact that there were no public prenatal controls in Venezuela. You had to pay for them and they had no money. Also, the problem we can see there in risky sexual practices and being reinfected by their partners, who were basically, some of them were helpful and some of them were very unsupportive, and here's 2 quotes about that. About telling the men that they had been diagnosed with syphilis. He didn't take it very well because, like any macho man, he put all the blame on me. I haven't told him because men, they'll believe it's me, that it's not their fault. And also cases of violence when women, and a fear of violence in even bringing this issue up. Then, these are now the interviews of the 30 vulnerable youth. Okay, so we don't even know if these people had syphilis, right? These were just part of the risk group that we were reaching out via these workshops. Some of these characteristics are that, in this case, they were also a much higher percentage of Venezuelans without regular legal status, low levels of education, and everybody working in the informal sector. And what was really marked about these migrants was that basically the majority of them left for sort of extreme economic circumstances. I left because of the difficult situation in the country. I couldn't study or have anything to eat. My mother brought me by telling we are going on a vacation. 4 years have passed and we have not returned to Venezuela. This is a picture of a settlement where people who come from Venezuela set up shacks to live. And it's very poor, but still it's much better than what they had in Venezuela. And here's a quote. "In Colombia, I have been able to improve in terms of study and work opportunities, and it is easier to get food." And here another quote. "Here in Colombia, you can fight to get somewhere. Back in Venezuela, when one worked, it did not even guarantee food." And so really kind of the lack of very basic needs was driving immigrants to cross over into Colombia. Other characteristics, almost 90% did not have access to healthcare, and this is of course a problem if you're looking at like early detection and treatment of syphilis. It's not included in this slide here, but I also just want to say that 95% of basic, I think like 29 out of 30, actually 29 out of 30 had never heard of syphilis before. So like complete lack of knowledge of it. Age of first birth in the teens, sexual debut also in the teens. And here you can see on this issue of STDs, there was a strong resistance. We also interviewed men to using condoms. They all said we should use condoms, and many of them said they simply didn't like to use condoms. And as you can see that half of men do not use protection to avoid STDs, 26% of women don't. And many of them don't because their partners don't want them to. So you can see this kind of machismo and the unequal power and gender relations being an issue as well. And now I actually conclude. I mean, what these results show are the important kind of the social determinants of health and the mixture of lack of knowledge combined with machismo, combined with lack of access to healthcare, and really the critical lack of basic resources. And then very importantly, I think it shows the importance of combining, like in terms of policy, the importance of combining social protections with health and medical measures. We can't just have public health information campaigns without providing people with the means to feed their families and actually be able to go and get treatment. And here, I won't get into this too much because this is perhaps more an academic conference, but we also had a list of very concrete policy recommendations that we presented to the municipal government in Bogota as well as the national government in Bogota, Colombia, about information campaigns, expanding access nationally to undocumented immigrants for prenatal care as well as testing for STDs. It exists in Bogota, the capital, but not nationally. And also considering conditional cash transfers as incentives to ensure adherence or increase adherence to treatment. And also importantly, none of these policies should be directed only at Venezuelans. This is a problem that is Venezuelans as well as Colombians. And I stopped talking here. I'm sorry if I went on too long, and very much look forward to your questions. And I do want to say that this is very much a collaborative effort, and fair to list the people that have been involved in this project, and we've been working on it together for the past year and a half. Thank you.
Okay. Thank you, Marika. Let me just add that we did have those outreach workshops in Bogota and in Cucuta, and in Bogota it was also the Ministry of Health present there, so we did have this message really, and let me just repeat the numbers. It's congenital syphilis, perhaps that is not clear to everybody, is really very serious lifelong consequences for the newborns with congenital syphilis, and the cost of this monetary incentive is 2 times $11. So it's completely out of proportion what you win with very small amounts of money, and you get a much better adherence, and you really prevent a lot of human suffering and a lot of economic cost, which is something, of course, policymakers will be or should be attentive to. So this is why we do have this policy. Just the last slide and concentrate more on the medical and social side, but we have this policy implications with workshops, and we got the Colombian team over to Germany. We had workshops in Hamburg and we had workshops in Berlin, because of course also we hope that this is something that development cooperation or other health international actors can pick up, because it really is I think it's a very, very clear case where you can make a big difference with very rather limited input. And perhaps also to say that the study was 100,000 euros. It was no more than that for all these results. And of course the numbers, the interviews are not that high. It's not the large N perhaps, but still numbers are clear enough to really make a very convincing, we find, call for action on that. So with that, thanks again, Marika. Thanks also to the Colombian team in absence, and thank also all those who volunteered to be part of the survey, because of course it's an issue people don't, very tabooized situation to talk about. So we also have to be very thankful to all those who participated in the study. Okay, without further ado from my side, the floor is open for your questions, comments. Am I doing moderation? Yes, I do. Okay. Then I also do the microphone guy.
Thank you very much for the nice talk.
I was wondering why syphilis, although you already point out some of the reasons. Regarding other sexual diseases, if you have any information, because definitely not to use the condom means transmission of syphilis, but some other sexual transmitted diseases. And if there is any kind of, for instance, condom providing for this vulnerable population as part of the health politics. And the thing with syphilis is that people can get reinfected even after being treated. So is there any policy or any approach or perspective in order to really prevent, beyond the education of this population, to really prevent the transmission or reinfection?
I was going to say we collect some questions, but these were some questions which we collected, so go ahead, Marike.
Yeah. So the first one was why syphilis? Because for the empirical reason that, I'll give you the context. So this was actually a side money left over from a multi-million-dollar development project between the German Development Cooperation, Charité, and Colombia to help Colombians get vaccinated during the COVID crisis. And so basically, Colombians at the border region, and including Venezuelan refugees, got vaccinated, got tested and vaccinated through German development money. At some point, basically saturation had been achieved, i.e. everybody who wanted to get vaccinated had gotten vaccinated, and they had a little bit of money left over, and meanwhile there was this invisible crisis that the doctors at the public hospital were talking about all these babies being born with congenital syphilis. And so we managed to redirect a small amount of money to address this crisis, and it was simply because of the births taking place. Of course, there are other STDs that are very important as well, HIV, and some of the studies even related to this broader project are looking at diagnosing like a package of, chlamydia, HIV, and syphilis. We only had the resources to do syphilis, and syphilis allows for these rapid tests. So we don't know what these populations we targeted. They could also be carriers of HIV and chlamydia, and there's evidence that a certain share of them are. We didn't have those resources to look at anything except syphilis. The thing with syphilis, of course, is that it is so very treatable, and so there's a good argument to be made for the cost effectiveness of zeroing in on syphilis if you have low levels of resources. Also with HIV, there's significant international funding for HIV, and so you can get tested for HIV with international donor money, but the money for syphilis isn't there. So that's why we chose syphilis. The other issue about reinfection, well, no, we are trying to, part of the advocacy is to try to get more attention to these policies. Of course, all the public health workers working on this issue know the problem is very much, there's really resolving this requires, a more holistic perspective that also addresses the issue of gender relations, because the reinfection often happens to the woman getting treated and the man not getting treated, refusing to do so, and then reinfecting the woman again, and she might be pregnant, and we had cases of that in our interviews, and so that is a broader social problem. And it's an issue also of limited resources. So no, clearly there is no holistic national policy addressing some of these vulnerable groups. For example, in Bogota, they had more more targeted policies, and I think actually apparently, and this is very nice news, we presented these results in Bogota last February, and they are taking some of the findings apparently and using them in some of their municipal policies. I can't remember if there was a third question.
I think it's we make a stop because we have other people also with questions and many people who probably want to go into at some point. But just briefly how it came about was also, I was speaking with Felix and he said, well, I saw this drama of those kids born with congenital syphilis, and as a medical guy, I know what there is to do, but how do we reach those people? That is a social question. Bert, in that your field from the social sciences, that's how we can collaborate on that. So that's also the link to the GLACIER part, because it kind of came from this discourse community that we are, which is interdisciplinary beyond what normally is the narrow disciplinarity. So I think that's a good example actually of what GLACIER can do. I got Ludger and I got Michael. Do I have somebody else? And you. And after those three, we take the three together and then we have to close, I'm being told.
Okay. Yeah, I was talking about two numbers. First, that every 18th person has synthesis and migration in the area.
I can't hear us.
Okay. I was shocked that every 18th person had syphilis in that migration area, but more shocking for me is that without the financial incentive, only 45% responded favorably to a free treatment for a possibly even life-threatening infection. I mean, that is for me unbelievable how only 45% would respond to that. You would definitely not expect 100%, but if you have no money and you get a free treatment for such a disease, I would expect it 90% to say hello. Yeah. So what is the, and their social sciences coming, what is the reason? Why is that so?
I have to get there.
Thank you very much. Oh, very loud. Thank you very much. First of all, I'm very grateful for your presentation because you looked so much on the determinants of health, and in the beginning you said migration brings infectious diseases, but you have shown that it's not migration that brings infectious diseases. It's the conditions that makes migrants vulnerable to infectious disease, and migration itself is not something like a risk, or migrants don't bring infectious diseases to other countries. There are cases, yes, but in general it's the conditions and the lack of access to healthcare, the living conditions, and the violence. And I would have expected to listen a little bit about the violence, sexual violence, for example. These women are suffering, so I think we have to name these things very clearly. But I have two questions. The first one is, when you think that there's no therapy adherence, or they don't get the treatment they can get, did they mention something in the interviews? Why don't they go to get the treatment? So why the therapy adherence is so low? Did they tell something about the reasons why they don't? And the second question is about the consequences. That's about syphilis, and you says conditional cash transfer would be an option, but people have tuberculosis, have sexual reproductive health issues, so a lot of health problems. You cannot give for every disease a conditional cash transfer system to increase a little bit the coverage of tuberculosis or whatsoever. And all the recommendations from WHO, from the Lancet Commission on Migration and Health, from international conferences, is always that we need strong health systems that are inclusive for all people. That would be my recommendation. And not only punctual cash transfer for syphilis, but actually create health systems that are really capable to treat the diseases and provide the health care that people, the host population, and also the migrants would need. Thank you.
Thank you, Michael.
Thank you very much. I was wondering a bit on the maybe ethical questions of giving people money to cure them from a disease they have, and maybe a bit controversial, but might it not even be an incentive for the people to get the disease, to get the money repeatedly? Although I do not believe that this is the case, but maybe you can comment on that as well. Thank you.
Thank you. Marika.
Thank you. Yes, very excellent questions. I wish I was there and I could have coffee with all of you afterwards. On the issue of the surprisingness of the low level of adherence to treatment, I think what that shows is the importance of doing this kind of on-the-ground work and understanding the lived realities of people who live in the context of extreme scarcity of resources. So many times people said that I went for days without eating. You know, I watched my kids without food. And in that context, the hypothesis that we posited that you can either have a menial job in the informal sector to make sure your kids are not starving, or you go and you focus on getting treatment for a disease for which you might not at the moment have any symptoms for.
And so kind of trying to understand the calculus of people who live at the margins of survival, I think that's an important issue. I think there's a lot more to explore in this, and a lot of people have explored much more this issue, but it's understanding the conditions in which people, like in the calculus, an opportunity cost that you deal with when you have immediate needs, and then you have critical but not as urgent needs, right, with long-term kind of payoffs or costs. But if you're not sure if you can survive the week, then that might be kind of a further issue. And then I think there's also a gender element, because clearly there seems to be more concern about this with women who actually suffer the costs and have children who, and as primary caregivers to those children. So I think those need to be looked at together. The second issue of violence, yeah, that's a very good point. And in fact, I'm on another commission studying the issue of sexual and domestic violence. There were some, we considered it unethical to directly address the issue of violence in interviews, given that like the ethics are such that you should only address violence if you have resources to offer people who then kind of come out and say they're experiencing violence. So we didn't have those resources. And so following the ethics, we had a question that referred to it. And yes, clearly there is violence here as well. There's also sex work going on for survival that increases the spread of these diseases. It should be stressed that it's not just kind of sex work and violence, that there are broader issues, but these are two of the nodes where you see, and the fear of violence, so you see that the issue of violence is a very important condition that exists in and of itself and is aggravated by the breakdown of the conditions that cause for this migration, total collapse of the state in Venezuela. And then there was a question about why no adherence. So this is we would love to do more research. If anybody wants to collaborate or has funding, there's so much more to do. Please reach out to me. This was a very small, limited project that was actually a health services project that we managed to kind of throw these little studies into it. But we didn't interview the people who got the cash transfers versus not. We interviewed people, the at-risk groups, before they even knew about the symptoms. And so we didn't ask them those questions. If I were to go and do a follow-up project, I would like to kind of embed more qualitative interviews with the people who were in the quasi-experiment to explore that. Then there was a very excellent question about, well, we can't just give conditional cash transfers for people to, come back for treatment for diseases. So on one level, I would say, why not? I mean, it's incredibly cost-effective, and it helps people meet basic needs, and it's very humanitarian in that sense. And if it helps prevent contagious diseases, why the heck not, instead of dealing with the results? However, I am a very strong advocate, and I've written policy papers on universal basic income for all children, for example, or even all adults. And so one of the speakers there mentioned the importance of strong and inclusive universal healthcare systems. I couldn't agree more. I would say that we also need some basic minimum income for everybody who is born into this world, and so that they can actually feed themselves, which then prevents a lot of diseases from occurring in the first place, and doesn't have to face the choice between feeding their kids or showing up for treatment. So there's different ways of doing this. But of course, in a policy proposal for the Colombian government advocating a universal cash transfer for all displaced people, is not. They won't listen in the same way. They think, why don't you just target these people with the cash transfer? You can spread combat the spread of disease. Finally, ethically, well, I mean, we adhere to ethical guidelines. I mean, cash transfers, first of all, conditional cash transfers have become a very broad and accepted development strategy in the last 25 years. They're most well known for mothers receiving them for kids going for health checkups and keeping, staying in school. So there's that. Of course, the way you do it, you have to do it in an ethical and appropriate way. And in this case, nothing was withheld. They got kind of the comprehensive healthcare treatment offered by, the protocols of Colombia. And then in addition, we gave people cash transfers under clear circumstances, and there is a significant kind of
expanding literature now that has been, and policies that have been doing that. Whether there's a moral hazard, I would say that in this case not. I think in general, there's a sector of society that kind of I'm not saying you do that, but sometimes moral hazard argument is made, I think, a little too loosely. I mean, in this case, it's hard to imagine somebody would try to get infected with syphilis to get 10 euros. In other cases, for example, there's some evidence that, well, if you offer like very generous cash transfers for people who are unemployed, it might be a disincentive to go and look for a job. Those kinds of arguments, I think, can be, you can see both sides. I think I covered them all.
Thank you, Marika. I think it was very complete. And it just prepared to remind you the 11 euros were calculated by what is the loss of income you could have if you don't travel there. It's a bus ticket and the loss, or the compensation for the loss of income. It's very much what we have also with healthcare. When you cannot go to your workplace, you still continue to get paid. I mean, and so I know that we're over time. It's the third day of a long conference. It's been a lot of input on very different themes. Thank you all for being here on the third day of this conference, also to the online audience. Thank you very much, Marika, for joining us online. Thank you for the IT colleagues who managed to get the voice of Marika into the room. Thank you everybody for also for the whole organization team here at the Martin Luther University in Saale, the last speaker. I think it's in the name of all of us really a huge thanks. It was a wonderfully organized conference and we all know how much work goes into that. Thank you so much.
Okay, Bert, I think you already made some nice closing words, and I think we had a great time here, and everybody enjoyed it. And I think what, from my point of view, which fascinates me in GLACIER, is really this one house concept, where we have, as you could see, social sciences, political sciences, which were really very heavily discussed, and I think we could have stayed here another half an hour or hour, and then together with all the chemistry, the vaccination, all these different aspects from animals to humans to plants. I think this fascinates me, and this did also fascinate me that I was saying, okay, I would like to join, and also together with Ludka to really be part of this, and we are representing here also Halle. But first of all, I think this would have been not possible if we would not have great help from Anna. Where is she? Come on here. Stop. Yeah, I think Anna was really the one who organized everything, put us into ties and writing emails, and actually sometimes they do not arrive, but this was not Anna's mistake. These were a couple of mistakes at the different universities. And thank you very much, Anna. We really enjoyed you, and hope for the next time that you are with us if we are getting to another meeting. Okay, just stay.
You will have a picture afterwards. Also, of course, the conference is the biggest disaster you can have after that the general organization of ANNA would have failed is the failure of the technical organization, and we didn't have that at all. And for that I would like to thank Frank Broda and Herr Mautz, who is there in the background, hidden. So I think for Frank Broda can take also for Mautz, because he's up there and cannot come down. I don't know if he can get you on the screen here. But also for you, there is a little thank you. Of course, not enough to compensate for what you have put in here, which is beyond the normal work. Of course, we know that very, very well. And there is a number of other persons to thank which are not here today, but we will deliver them. What?
Is Robert there?
Robert, ist Robert noch da? Nee, Robert, ja, da ist er. Robert, please, Robert, come here. Robert is also a coordinator, and he's a lab coordinator in Halle for GLACIER. He is, as we know him, he's a quiet person in the background, but very, very important and instrumental, especially for all the students who come here and have to do the work. And he also took care of our guests. Thank you, Robert, also for your input into GLACIER and the conference. Okay, and then last but not least, we have our secretaries.
But Maria Heise is not here, and also your secretary, Frau Stein, is not here, but we will bring it to the Institute.
Yeah, so we would like to thank them also and at least mention them to you, because most of you will know them and or have met one or the other of them. So we also would like to thank them for their support. And that is the team, these 5 people who did, let's say, 95% of the work. Thank you very much for that.
Yeah, thank you.
Maybe we could just stand together and have a little picture taken. I don't know, Esmeralda or someone is... Michael is not here anymore. Yes. Or but Bernd. We have to go here. And of course, remembering the magician, you have to juggle with these things. Thanks, Bernd. Thanks, Esmeralda.
Okay, and I think the last words which Anna reminded me, which is always doing this, which is good. Please take all your belongings with you. Don't forget anything, because this building will be closed, and then you might run into problems to really get the stuff. And last but not least, I would like to tell everybody have a really safe trip. I know that some people are coming from far, and have a safe trip, and if you have time, enjoy a little bit Halle. Bye.
Goodbye. Danke dir.